Good morning, good evening to everyone. Thank you for joining us today. Excited to kick off the 2025 CCDS virtual conference this year. We have a wonderful lineup of speakers for today and tomorrow. For our goals for these 2 days, as with every year, it's to bring people together, bring researchers, families, clinicians, anyone who wants to learn about CCDS together in this space. We'll learn about the advances in CTD, GAMT, and AGAT deficiencies, 3 different cerebral creatine deficiency syndromes. We'll hope to hear inspiring talks and be able to leave with some ideas of hope and really look forward to move the field ahead through interesting ideas in the next 2 days. Okay, so if this is your first time attending this symposium, it's probably a good idea to start over here, who we are, who is ACD. ACD was founded by parents of children with cerebral creatine deficiency syndromes in 2012. Since then, we've grown from a rather small organization to really a full-fledged team consisting of 7 staff members, 8 board members, and 2 board advisors. We are united in our mission to provide patient and family support for those navigating with cerebral creatine deficiencies syndromes, as well as to play a role in education and advocacy for newborn screening for early diagnosis and to promote and fund medical research for our community. And we're supported in this vision by our amazing scientific, medical, and advisory board. Many of the folks in our SMAB are stalwarts in the fields of CCDS. We owe the discovery and the diagnosis and a number of advances in the field to them. And they keep us grounded in our vision to have effective treatments and newborn screening for all 3 disorders while also providing community support. So what are the cerebral creatine deficiency syndromes, and why do we have 2 days of talks dedicated to just this category? So CCDS represent 3 single gene disorders that impact the biosynthesis and the transport of creatine. And deficiencies in these genes lead to a profound lack of systemic and cerebral creatine deficiency. So just for a little bit of maybe background, the first step in the biosynthesis of creatine is the creation of GAA, which takes place through this particular enzyme called the AGAT enzyme, which is primarily expressed in the kidney. AGAT utilizes these precursors, arginine and glycine, to synthesize guanidinoacetate. GAA is then carried through the blood, reaches the liver, where it's acted upon by the second enzyme in the biosynthetic pathway, the guanidinoacetate N-methyltransferase enzyme, which results in the formation of creatine. Creatine is then transported through the blood to different parts of the body, and it's thought that a defect or mutations in any of these genes result in the profound lack of creatine that you see. Early studies have shown that while the lack of creatine throughout the body presents a significant challenge, it's the lack of creatine in the brain that is associated with a large amount of the pathophysiology associated with the disease. And therefore, this category of disorders is more broadly called as cerebral creatine deficiency syndromes. To give you a little bit more about the flavor for these three different diseases, AGAT deficiency is considered ultra rare. We know of less than 50 patients that have this disorder, and early diagnosis is key. GAMT deficiency also is ultra rare, with the incidence being 1 in about 500,000. We were successful in the past several years in ACD played a role in enabling putting this disorder on the RUSP, or the recommended uniform screening panel, which enables newborn screening to be possible in all 50 states of the of the US. And this, we hope, will lead to early diagnosis and interventions, resulting in better outcomes for patients with this disease. And then finally, CTD, which is the least rare of these three disorders, but still falls by the classical definition, still falls under the category of rare disease. It's thought to be underdiagnosed. It's an X-linked disorder, primarily diagnosed in males, but also affecting females and often going undiagnosed in females. It's thought to be the second leading cause of intellectual disability in males. And the unifying theme for these three diseases is that there is no treatment that corrects the underlying cause of the pathology. For AGAT deficiency and for GAMT deficiency, supplementation with nutrients, with the missing key agents is thought to have a positive outcome. But what we hear of every day from talking to patients in our community is that a treatment is still needed for better outcomes and management. So what does ACD do, and how does it spur or initiate advances in these diseases? Here I'm showing a typical maybe timeline of preclinical and clinical drug development. So for most common diseases, really you see that about 5 years are spent in identifying a molecule that is ready to go into the clinic, and then 8 years or so, give or take, is spent in the clinical development or clinical trial phase before it actually hits the market. And this is true largely true for the more common disorders. For rare diseases, this is a little bit flipped, because in rare diseases, since the patient population is small, the clinical trial phase can sometimes be shorter. Patients get access to therapies during that phase in certain cases. But where we spend conversely more amount of time, where we have a liability, is really in the early parts of discovery. And this is because the population affected by these disorders is small. There is a dire need for better understanding of the disease, for tools and reagents that can enable prosecution of the disease early. And this is really where most of ACD's efforts, research strategy-wise, where our efforts are focused, right? So we have spent a fair amount of time supporting initiatives that fall into each of these buckets. So I already alluded to diagnosis, early diagnosis. So how can we diagnose faster and quicker, maybe more patients that are going unnoticed? We did this by addition of GAMT to RUSP, and similar efforts are underway for CTD as well as GAMT. To learn more about the patient population, we initiated a patient registry. We'll hear more about that through the symposium. Through this and some focus studies on outcomes that are important to patients, we really try to bring forth the patient voice and cascade that information to stakeholders in the field, whether it's clinicians or investigators, industry stakeholders, and the like. Clinical trial readiness, in order to enable success in that final phase, when we get there, it's important that our community is brought along for what should you expect from a clinical trial? What does participation in a trial really look like, and what can you expect? And we do spend some time in educational webinars to bring our people along with our understanding of where we are in this odyssey. And last but maybe not the least, the two critical pillars, therapeutic discovery and biomarker discovery. So ACD invests in promising innovative therapeutic approaches to identify a molecule that is ready for clinic. We support initiatives in this area. And then importantly, to understand if these therapeutic opportunities actually translate to actionable improvement, you need to have a sensitive biomarker that you can use to read out progression of the disease. And some of our efforts are focused in understanding more of these as well. So since 2019, ACD has made extensive research investments. We have supported members of our consortium in gene therapy efforts, many fellows over the past several years. This year we have a rather large fellow class of five fellows. We have also simultaneously supported sponsored research collaborations with CROs as well as academic institutions, and in total we've spent upwards of $1 million in all of these initiatives over the past several years. Simultaneously, we've also invested in our own registry, and you'll hear about updates from our registry advisor, Emily Reinhardt, later through these next few days. And then for new families in the audience, please do stay tuned for that talk. If you know if you want to know what is the registry, how to get involved, then please do make sure to attend that talk. There's a lot of good information over there that will be useful. And then really, I think it's important to underscore that it's partnerships that drive our progress. Over the past couple of years, we've been incredibly fortunate to get this type of support from all of the universities in the US as well as elsewhere. And together, these efforts will accelerate discovery of therapies for CCDS. These are just some of our partners. It's not comprehensive, but just a reflection of the diversity of folks we work with. A key component of ACD's efforts is to really bring new researchers, young researchers into the fold. And this is what our fellowship program enables us to do. By virtue of bringing in young researchers, supporting them in their pursuits, we really hope that we will be able to retain them long in the field of CCDS research. And I'm really excited to know these are just a few papers that I pulled in a short amount of time, not comprehensive, but the efforts that we have supported in the past couple of years have led to tangible movement of the field. And over this slide and the next one, I'm calling out papers that did acknowledge us in their support. It's one thing to call out in this slide particularly is the ones highlighted with the box are papers published by our own staff members on understanding on early diagnosis and understanding of important outcomes for our patient group. These efforts have been really made possible through some of the funding that we received from CZI, PCORI, as well as the funds raised from our own community, from the Walk for Strength and Holiday Heroes. I realize I'm at time, but I'll just take maybe a minute or so to quickly wrap up. But the one thing that I did want to mention is that capitalizing on all of the progress that we've made so far, we did announce earlier this year our most ambitious initiative yet, Race for a Cure. We're really fortunate that one of our board members, Jeffrey Allen, enabled us to utilize his platform to raise awareness for CCDS. And this is our most ambitious call yet. We put out a call for letters of intent and are in the process of evaluating proposals with the goal of supporting many more innovative translational therapies. And so really all this to say that together we really are changing the future of CCDS. I hope through these next 2 days, I can leave everyone with a feeling of optimism and confidence on the amazing work that members of our community will present. And with that, I'll take you to what to expect today and tomorrow. We have 4 sessions on both days. Our sessions are a little different this time around because we indexed higher on longer times for talks so that we can dive deeper into the topics. We will have Q&As, panel-based Q&As moderated by members from our community, as well as times for break. I do understand that these 2 days of symposium get to be pretty intense, so feel free to step away if you need to, but do come back because there's a lot in here that is truly exciting to hear about. If you make it till the very end of day 2, then we do have a treat for you. We have a special session at the end of day 2, an interactive panel that we're really excited to get into for what's the future of therapies for rare genetic diseases. A few house rules on your Zoom panel. There is a Q&A box. Throughout the talks, please do post questions in the Q&A box, and where possible, please specify which speaker the question is addressed to. We will also have an opportunity for you to interact with us, for us to get to know you better, get to know what your interests are better. There are polls. Please watch out for them and please do participate in them. It really helps us design our content better for you in the months that come. We love acknowledgement on social media. So whatever your preferred platform, please give us a shout out and tag us. And with that, I'm happy to invite Colleen to pull up the first talk for today. We'll be hearing a recorded talk from one of the parents in our community, MAAIKE. MAAIKE lives in Netherlands and has, with her 3-year-old son, Oscar, who has CTD, she will be sharing her experiences navigating CCDS, a CCDS diagnosis. And so this is the first of a number of talks of this manner. And I hope it really brings us together and sets the tone for what is important and why we are here today. Front and center, we put patients first, and I hope that this will be inspiring to everyone.
I'm going to tell you something about our son Oscar. We are a family of 3, so myself and my partner Desmond, and we have one son, Oscar. He's currently about 3.5 years old. We live in the Netherlands, but my partner Desmond is Irish. So he's been living in the Netherlands for about 12 years now, but does not speak Dutch. So I speak Dutch at home. Desmond speaks English also to Oscar, so we're a bilingual family living in the Netherlands. And yeah, so Oscar is 3.5 years old now. He was diagnosed when he was just over 2 years old, diagnosed with CTD. And I would like to tell you a bit about the background of our history with CTD and Oscar's disease and what we've been through the past, 3.5 years really. So Oscar was born in February 2022, and it was an uncomplicated pregnancy and birth, but he did have a very low expected birth weight. So I was induced at 40 weeks because we just wanted him to be born because we thought he was very small, and he indeed was. When he was born, he was 2.6 kilos, but he did really great just after birth. He was feeding well. He was just a happy baby. Although we quickly sensed that something was not normal, even though I didn't know many other babies. Something just wasn't right. He seemed different, and it was always difficult to pinpoint what exactly was wrong. We noticed he was experiencing delayed milestones pretty much from the start. Like, even the first ones, like smiling and turning around and sitting and crawling, everything was delayed. What also was noticeable that he had quite a lot of reflux and vomiting. I actually wanted to find a picture where he was enjoying tummy time, which he hated, so he didn't really enjoy it ever. And then I saw that this photo was not just a photo of his tummy time, but also of here where he just vomited. He would leave traces of milk everywhere he went. But we always thought, or we were always told, it was normal for babies to give back milk. He seemed to have a lack of interest in his surroundings, where other babies were really experiencing everything around him and were curious. Oscar was, we always described him as content. He was just a happy baby. He was happy wherever you would put him. He was happy with whatever he was doing or what other people were doing, but he didn't really venture out himself. And what was noticeable that he had limited weight gain. He was small from birth, and he still is very small. But for the first months, we just dealt with it. Everyone told us, okay, all kids are different. We'll see what happens with Oscar. Even though his milestones are delayed, let's wait and see what happens. And that changed a bit after a while. He also had breath holding spells, so where babies cry so hard and they can't really get out of themselves. They pass out, they turn blue, they turn stiff. or become very weak and then pass out usually for a few seconds. And it happened for the first time with Oscar when he was just 6 weeks old, but it became more frequent. And he had a very severe one when he was about 17 months old. It actually happened in daycare, so we weren't there ourselves, so we didn't really know what happened, but he cried for some reason, and then he passed out, and he remained unconscious for a good 20 minutes. So they called the ambulance came, the trauma helicopter came, it was all a big fuss. And by the time we were in the hospital, it was about 20-30 minutes later, he was just smiling as if nothing really happened, even though he had been unconscious for 20 minutes. So we wanted to follow this up. So we did neurological tests. We did a 1-hour EEG, but it didn't show any epilepsy. And he did have these delayed milestones. So we continued with regular checkups, but we then did not do any further tests. But we were definitely not confident it was nothing, but we also didn't really know what to do at that point. So we just kept doing what we were doing. And then he had another one of those severe spells at 20 months, and that was for us the point where we said, okay, we really need to dig into this because we don't have a good feeling about it, and we need to look into it further. So we ended up with a pediatrician neurologist in the local academic medical center. We did a 24-hour EEG, which showed no epilepsy. Also did some blood work that was normal, and we thought, well, just to be sure, they offered it to us, let's do some DNA tests to see that at least he doesn't have any of those rare disorders, because, you know, even though we thought something is wrong, we never expected to find something that rare. But then the test did show a mutation in the SLC6A8 gene, and so expected CTD. And then this was just after his 2nd birthday, and then it was just a roller coaster for us and for Oscar. This is a slide with lots of things, but that's also what it felt like to us. We just started a roller coaster of all the medical tests and everything that came with the diagnosis of CTD. So we did blood and urine samples to confirm the diagnosis also clinically, and we're following that up every 4 months. We did an MRI and an MRS to check creatine levels in the brain, and did lots of developmental tests. And it was shown then also what we already expected, that he was delayed in all areas of development, so cognition, fine motor skills, gross motor skills, and speech communication. We did a skin biopsy to test if there was any remaining functionality of the creatine transporter in vitro, which came back negative. We started supplements, so pretty soon after the diagnosis, we started with arginine, glycine, and creatine for Oscar. That's also why we're doing the regular blood and urine samples to see if it has any side effects on his kidney function. And it's also why we're repeating the MRI, MRS, and the developmental test to see if there's a difference before we started the supplements and 1 year. We actually just had the 1 year tests, and unfortunately, we did not see any creatine improvement in the brain, so no rise in creatine levels after the supplements. But he's been making big progress on the developmental aspects, so we're currently just continuing with the supplements. We did cardiology tests, 24-hour Holter, even a 2-week Holter, an ultrasound of the heart. And even though he's not diagnosed with prolonged QT syndrome, they do say they see signs of prolonged QT, so we're treating it as if he has it. So we're very careful with medication and keeping an eye on him. But it's a bit vague what it exactly means, so we're following that up. We did audiology tests because it has been described it can come with hearing loss, and since he was behind in speech, we just wanted to rule out there was anything ongoing there, but fortunately not. We followed up with a dietitian because of his very low weight. That's still ongoing. We went to the eye doctor because he is a bit cross-eyed, so we gave him glasses, which really help, and he's really happy to wear them. So that must mean something, that it helps him. And we intensified therapies. So we hadn't started yet with speech therapy before he was diagnosed, and then we started that once a week. We already had physiotherapy, but we continued that for once a week. And he used to go to a normal daycare or a regular daycare, and we switched to a special medical daycare where he goes to 3 times a week, which is a great place for him because he gets lots of one-on-one attention there. There's smaller groups, only 6 kids, and the physiotherapy and the speech therapy come to the location. So yeah, it's been an absolutely crazy year the past year, just from the practicalities of all these medical tests, but of course also emotionally. It was a very difficult year for us to come to terms with the diagnosis and not knowing what the future holds. Now a year later, I think we're a bit better and more able to just enjoy Oscar who he is, because, you know, Oscar didn't change, and he is a very happy and content boy who is always smiling. He's always full of energy. He never sleeps, unfortunately, but that doesn't hinder him in just being happy and active all day. So I also wanted to show a bit of show some videos of the progress he's been making and where he's currently at, and some challenges we're currently facing. So his motor skills have developed greatly in the past year. In June last year, he was walking on his knees. He walked on his knees like this for a good 6 months before he started walking. And the physio always said, well, he's really working on his core strength, and as soon as he figures out that he can walk on his feet, he will be going like crazy, which happened because he took his first steps in September last year, and he immediately could walk long distances with pretty good balance. And now, this was just 2 weeks ago, he is fully confident walking or almost running, which is great to see. It's difficult to bring him for a walk because you're only going in a direction that he wants to walk to, and he does not listen to you. He does not listen to stop, and definitely doesn't want to hold your hand while he walks, but he really enjoys it, so that's really good. And he's becoming more confident in his motor skills as well, so we're trying to challenge him as well. He's climbing, he's sliding, and we gave him this nice tri-bike recently that he loves. So he's making huge steps there, and that's great to see. The same for his communication skills. So he started with supporting sign language, which he picked up a little bit, but we also noticed that he just picks up words, approximations sometimes, but he spoke his first words around 2.5 years old, and currently we think he has, well, definitely more than 100 words in active use, and he's now starting to make 2-word sentences, and it's great to see because he makes progress every day. There's like a new word every day, and I have this little clip because I think it shows really well who Oscar is and how he is. Opa means grandfather, and oma is grandmother, so those are my parents, so it's Dutch grandparents. And Ollie is their dog.
Opa! Opa! Oma! 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 Oma!
En ook Ollie?
Hoi! 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 Hi, guys.
I enjoy watching this video because I think it also shows a bit of who Oscar is. He is just thinking about his grandparents because they're nowhere near. We had visited him that day. So he's just thinking about them and being happy about it. And when I mention Ollie, so the name of the dog, you can really see him like process that information, thinking, oh yeah, Ollie is our dog, I like Ollie too. You can just see him process the information, and that's typical for Oscar to really need a bit of time before he understands what you're asking of him or what you're telling him. And then he's very easily distracted because then all of a sudden he points at the window and says outside. And because he's bilingual, it's sometimes a bit challenging. He says, he mixes up the words in Dutch and English. So in Dutch it's buiten, and in English, of course, outside. So it becomes something like buitside. So it's always tricky to translate what he actually means. But together we usually make it work. So it's great to see that he's making so much progress, and life gets easier as well. Now we can talk to him a bit more, and he can make himself a bit understandable. We notice that the breath holding spells are really decreasing in frequency also, because he can make a bit clearer what he wants or what he needs from us. And when he's sad, sometimes he doesn't cry when he's sad, but he just says the word huilen, which means crying in Dutch. So he's trying to give words to what he's feeling, which is great. But of course, we still face lots of challenges. Sleeping is a major one. We haven't really slept through a night in 3.5 years. As a baby, he already didn't like to nap. Never really napped during the day, and he still doesn't. He wakes up very frequently during the night, and usually for a long period. So we're trying to tackle that with a sleep coach and trying all types of tactics to get him to sleep. And it is getting better, but it could be improved. So if anyone has any tips, please let us know. He is becoming an increasingly picky eater. He basically eats either spaghetti or fish fingers or bread. He has a good appetite, but he's picky, and he is not gaining a lot of weight, so that's worrisome. He's currently not getting any extra high calorie stuff, but we are considering to see what we can do there. But he always has lots of energy. He's never really ill. He's always happy, so it doesn't seem to hinder him really. But yeah, he's very much behind in terms of weight. We try to increase his focus or attention span. He has a very short attention span. He's also just a toddler, but that could definitely be improved. And he shows interest in potty training. So sometimes he tells us that he wants to sit on a potty. He always tells us if he has a full diaper. So we think he could be open for really starting potty training. So that's something that we are trying to do without too much pressure at the moment. But most importantly, Oscar is just a very happy boy, and he loves his slides and tractors and chilling. And we're trying to take it day by day since we got his diagnosis, and with time that goes better. So that's what I wanted to share with you very briefly about Oscar. I could talk about him for hours, but I'll leave it at this. So if you have any questions or remarks or suggestions, or if you just want to chat, please reach out to me. So, thank you very much.
Your story and letting us know about Oscar. I see that there's a question from Dr. Longo. Is MAAIKE in the audience? Maybe we can pull her into, if it's okay, Dr. Longo will pull her into the Q&A after the sessions to answer this. But MAAIKE has also graciously shared her email, so I encourage other parents and members of the community to reach out as well. With that, we'll kickstart our next talk. I see Amanda is already on here, so I'll just give a brief introduction to Amanda while she pulls up her slides, maybe. So Amanda is the Director of Molecular Diagnostics at the New York Genome Center. She's board certified by the American Board of Medical Genetics and Genomics in clinical biochemical genetics and laboratory genetics and genomics. All that to say she knows a lot about genes. Amanda has been involved with ClinGen since 2014, as well as the CCDS VCEP, or the Cerebral Creatine Deficiencies Variant Expert Curation Panel, since its inception. Today, she's going to talk about a collaboration update between the ACD and the Clinical Genome Resource. Hand it over to you, Amanda, for your talk.
Great, thank you so much. Do my slides look okay? You see the actual slides and not the presenter? Okay, fantastic. Yeah, so thanks to the ACD for inviting me to give you a collaboration update for the ACD and the Clinical Genome Resource. And I'm going to talk about a couple of different topics, but we are going to focus a little bit on SLC 6A8 today. So these are the things that I want to talk about. First is the collaboration between the Clinical Genome Resource, ClinGen, and the Cerebral Creatine Deficiencies Variant Curation Expert Panel. And in the interest of time and to save myself from tripping over those words, the CCDS VSEP is what I'll call it. So we'll talk very briefly about what we are, who we are, and then I'll talk about the collaboration between the ACD and the CCD VSEP and CreatineINFO and why that's so critically important, not only for current, but potentially for future individuals with CCDS. And then I want to talk about specification updates that we have been working on for SLC 6A8. This is version 2, and I want to talk about that because much of the work that has been now accomplished was really initiated after we attended ACD 2024. So I think it's another example of why this collaboration is so important. So first, let's talk about ClinGen and the CCD as VSEP. So who we are and what we do. So the Clinical Genome Resource, or ClinGen, is a National Institutes of Health funded resource, and it's really dedicated to building essential public database for depositing variants that have been curated and observed in affected or unaffected individuals, and really figuring out the relevance of genes with regards to a gene-disease relationship and specifically for variants. Are they causing disease or are they not? So the CCDS VSEP is under the umbrella of the Inborn Errors of Metabolism clinical domain. And you can see us here. Sorry, let me get out later. You can see us here. So this is the Cerebral Creatine Deficiencies Variant Curation Expert Panel. Our status is approved, and you can see just an example of some other VSEPs that are affiliated with ClinGen. So a VSEP in general is made up of a group of individuals with expertise in a specific disorder, and in this case, the CCDSs. So this is who we are. Most of the members you see here are volunteers who volunteer their time, expertise to the curation of variants in the 3 CCDS genes. Many of these faces and names you'll recommend. Jenny Goldstein is our fearless leader, and I'm honored to co-chair with her the CCDS VSEP. So the goal of the CCDS VSEP is to accurately classify variants that are observed in the 3 genes, GAMT, GATM, and SLC6A8. But to do that, the first thing that we had to do is we had to make variant or gene-specific guidelines for each gene to tell both our VSEP and any geneticists, researchers anywhere throughout the world who come across a variant in one of these genes how to classify it so that we're all consistent. So once we come up with these gene specifications, they have to be approved by the Sequence Variant Interpretation Work Group of ClinGen, and these are experts. It's a group of experts in molecular diagnostics who make sure that everything that we're specifying makes sense and it's all cohesive and it's not, it's consistent with what other groups are doing and how gene or variant classification should occur. So you can see here, we have all 3 genes have specifications released and available to anyone in the world who wants to take a look at them. These are held in something called the C-Spec, which is publicly available. I've highlighted here SLC6A8 because I'm going to talk about it a little bit later. So we have released version 1 of SLC6A8, but that was released in March of 2022. So there's been many updates since. The idea is that once we have these specifications, then we can accumulate a variety of published and unpublished evidence to curate these variants. So that begs the question, what evidence do we need to curate a variant? So this is a table that was originally published by Richard et al. In 2015, and this is the basis for variant classification. And you can see there are essentially two main types of evidence. There's benign evidence, so evidence fitting in this category would argue that a specific variant in a gene does not cause disease. And then there are different levels of pathogenic variants, pathogenic evidence. And this type of evidence would argue that a variant in this gene that meets some of these criteria might be leading to the disorder. So the general concept is that you go through all available evidence for a specific variant and you see where it fits in this grid. And by Richards et al., what you would do is you would combine. So let's say you have a pathogenic strong and a benign supporting. That would lead to one classification, whereas multiple lines of pathogenic evidence would lead to a different classification. But where exactly does this evidence come from? So some of the evidence is just available publicly or given to us by the type of variant that we're looking at. So this evidence, for example, these is population evidence that comes from public databases. So there's a very large database called NOMAD, for example, that is compiled of individuals without a known genetic disorder. So for example, there would not be any individuals with GAMT deficiency in this database. And then in silico algorithms are essentially variant predictors that can predict how severe a variant will be to protein function. I'm going to put functional studies here, but there's a very exciting talk coming up next, so I won't spend too much time on functional studies. And then this type of evidence is really just specific to the type of variant. So is this variant predicted to lead to a complete loss of the protein? Or is it just a single amino acid change, so the length of the protein doesn't change? So this is the evidence that's available for any variant. But that leaves, if you notice, all of these classes of evidence that are not publicly available. So this evidence, everything I've kind of highlighted here in green, we either need to get from the literature, so a variant that is published in the literature in an individual with or without a disorder, or we need patient data. So in the absence of all of these green boxes, we really don't have much evidence for pathogenicity of a variant. So we accumulate all this variant evidence, and it leads to a final variant classification. So what does that mean? So pathogenic and likely pathogenic means that if a variant is pathogenic, that means that there's a greater than 99% chance that this variant in this gene is leading to a genetic disorder if it meets the inheritance pattern. So for example, if it's an autosomal recessive, you need 2 variants. If it's X-linked, like SLC6A8, you just need 1 if you're a male, or 2 if you're a female. Likely pathogenic is also very strongly arguing that this variant leads to a disorder. On the other end of the spectrum is benign and likely benign. So these are variants that we are greater than 90% sure are not leading to a disorder. And that leaves everything in the middle as what we call a variant of uncertain significance. This is the bane of genetics existence. Clinicians, molecular genetics, cytogenetics, we all very much dislike VUS because they're not helpful. They just leave patients, it leads clinicians in this kind of limbo of what do we do with this? But hopefully from the previous slides, you can understand that in the absence of either a publication, a published variant, or patient data, most rare variants are going to land in this variant of uncertain significance category, just because we don't have evidence that it's pathogenic and we don't have evidence that it's benign. This is especially true for variants that don't lead to like complete loss of the protein, like nonsense and frameshift variants. That's why collaboration, especially between the CCDS VSEP and the ACD and CreatineINFO, is critically important for accurate variant interpretation. So if we have patient data, if you're willing to share your biochemical data with us, your variant data, inheritance information, that can result in a variant that would be a VUS being pushed to likely pathogenic or pathogenic. That not only helps the patient or the individual that we observed that variant in, that helps future patients as well. And overall, it's beneficial to the variant carrier, the entire CCDS community, the genetics community, future patients, and their families. So I just want to expand on this a little bit to let you know what kind of information we get from CreatineINFO and what this collaboration, how important it is for everyone involved. So this is kind of a general work flow of how it works. So we have CreatineINFO who gives us variants, say, hey, we've identified this variant in a patient through Genome Connect. And then we, as the VCEP, use the information that's made available to us to make a classification for that variant. Once we classify that variant, we put it into a public variant repository called ClinVar. So we reach out to CreatineINFO. If we just come across a variant as well, we reach out and say, hey, do you have any information on this variant? Just to let you know, this is all done through an IRB approved protocol, and all of the information that we get is de-identified. That means we don't get your name, we don't get your date of birth, we don't get where you're living, anything like that. We literally just get the variant and associated biochemical evidence. So these are the classes of variant evidence that we can get from CreatineINFO. So first of all, we get the variant information. So what variant or variants were detected? So for GAMT and GATM, there are two variants that are typically detected because this is an autosomal recessive disorder. So that can give us some evidence here, or for segregation evidence for PM3. If it's a variant in SLC6A8, that can also give us evidence. And the original Richards, when Richards came out with this, you could only have variant evidence in one category. So for example, this evidence was only strong. But since Richards came out, and especially currently, this is now on a sliding scale. So if we only have one patient, that might be a supporting level of pathogenic evidence. But if we have 5 patients, that may be very strong. We also ask for inheritance information. So for example, is an SLC6A8 variant inherited from a mom, or is it de novo, or meaning of new? Is it new in the individual with creatine transport deficiency? That can give us evidence down here. And then also for GAMT and AGAT, that can give us evidence down here. So you see, again, this is a sliding scale. So depending on the nature of the variant and inheritance information, it can be either supporting or a very strong level of evidence. We ask for other affected or unaffected family members, and do they have the same variants? That's evidence for here, and evidence for here, which again can go all the way up to very strong. And also, if it doesn't segregate, this can be counted towards benign evidence. The most important thing is the biochemical data. So the CCDSs are a bit unique in that, well, not so unique, but they are special in that the biochemical evidence for AGAT, GAMT, or SLC6A8 deficiency are highly, highly specific for that disorder. That means if we have MRS data and then plasma and urine creatine, GAA, things of this nature, we can actually upgrade this, which used to be supporting, to strong. And we're one of the only VCEPs who have been approved to use this line of evidence at strong. And that's only because it's very highly specific for the gene. And then not only does the variant information and the information we get from CreatineINFO allow us to curate that variant, it becomes important for future variants or even past variants, because there's other criteria that says, for example, okay, you found a missense variant, let's say tyrosine 313 serine and SLC6A8, and you've classified that as likely pathogenic. That means, you know, a year down the road, if we find a tyrosine 313 glycine, the fact that we have a different change at that same amino acid that is pathogenic is evidence that the new variant is pathogenic as well. So not only does it help the current individual, it can also help us reclassify variants in the future or in the past to lead to diagnoses for these patients. This is just a quick example of how the data that we were able to receive from CreatineINFO and the ACD helped us reclassify some variants. So here's 2 variants in SLC6A8. You can see they were originally classified as variant of uncertain significance. Again, this is population, and this is that in silico predictor I mentioned. That's all we had for that, so it was a VUS. But this variant was identified in someone who submitted their data to CreatineINFO. So then we got MRS data and biochemical data. So we were able to apply that at a strong level of pathogenic evidence. And then the inheritance information we were able to get also presumed that this was new in the patient. So with the CreatineINFO information and the ACD collaboration, we were able to upgrade this from a variant of uncertain significance to likely pathogenic, which provided a diagnosis for that individual, but also now this missense variant will deposit into ClinVar, so anyone who comes across this variant again will know that this is associated with CTD, and any other, glutamine 114 variant will also have additional evidence. So we were able to upgrade several of these variants, and this is just an example. This is ongoing work, and we're still collaborating and doing additional variants. So the ultimate goal of this is ClinGen's sort of overall arching theme, is that it's a collaboration between patients, clinicians, laboratories, and researchers to classify variants, aggregate all this genomic and health data, which helps us further kind of solidify gene disease validity, variant pathogenicity. And then through that, we disseminate this expert curated knowledge with these collaborations, which in turn helps the patients, the clinicians, the laboratories, with the ultimate goal of improving patient care through these collaborations and through genomic medicine. So now I want to turn the focus to our ongoing or our almost complete updates for SLC6A8. This is going to be SLC6A8 specifications v2.0. So this all started, I was honored to be able to speak at the ACD 2024 symposium, and we had a lot of feedback from patients, families after my talk, and then I was able to meet and discuss a lot of things with other families throughout the symposium. And we took away from that, I took away from that we really needed to update the SLC6A8 guidelines because they've been published now for 3 years. There have been many updates in the field. And then there were some other kind of things that we became aware of. So we started working on these V2, some of the major updates. So the Richards et al. System was set up so that you had to combine like strong, moderate, supporting to come up with a classification. There's a new publication that's come out since that has adopted all of this into more of a Bayesian point scoring system. So now instead of like variant classes, we assign points. And then at the end of a variant curation, the total sum of those points are what lead to a variant classification. It allows for more granularity. It allows for Richards originally said if you had benign and pathogenic evidence, it was automatically a VUS, which now we know through the field is not always the case. So now we can just combine those positive points with those negative points that you would get from benign, and it can still result in a likely pathogenic classification. There are updates to population frequencies. There's a new version of NOMAD called NOMAD v4.1 that just came out that has a significantly increased number of individuals. And on top of that, Jenny and Sadet specifically are working with a genetic prevalence subgroup of ClinGen to work out carrier frequencies for the genes. There's new mouse models that have become available that we've added. And then there's updates to phenotype specificity to allow for counting females. We realized after presenting SLC6A8V1 that our guidelines were really heavily weighted towards being able to classify SLC6A8 variants in males. And we've relaxed and modified some of that, so that's not the case any longer. This is just an example of some of the updates that have come out since the SLC6A8V1 was approved. So we have new evidence involving splicing impacts. This is the one that I was telling you about moving Richards to more of a Bayesian classification framework that's a point-based. There's more guidance for segregation and co-segregation. And then this really fantastic review about phenotypes and phenotypic variability in females with SLC6A8 variants. So we've incorporated all of that, and I want to kind of specifically talk about some of the codes that we've updated. So this is our original PP4, which is the code that we use for phenotype specificity. So this is the code that we use if we have biochemical evidence And for like a urine to creatine to creatinine ratio, the MRS, and then any kind of functional studies that are performed in patient cells. But you can see here that this is highly weighted towards hemizygous males. So for example, we know in females, they might not have biochemical anomalies associated with CTD, even if they're symptomatic. And females also can have highly variable phenotypes along the spectrum, and they might not always meet criteria for this code. So what we've done is we've actually expanded this. So we've separated into males and females with this SLC6A8 variant. They're largely the same. The males, the criteria that we're using there hasn't changed. In females, though, we've added this genetic testing modality. So this is essentially saying if an individual is presenting with CTD phenotypes, and she's had a very comprehensive genetic testing, including genome or exome sequencing. And the only variant that was considered probably causative is this SLC6A8 variant. We're now going to award points for PP4 for that. And so the way that we've set it up, a female with an SLC6A8 variant and no other pathogenic or likely pathogenic variants can be awarded enough points for at least a supporting level of pathogenic evidence. And then if there are any other biochemical anomalies, this can be upgraded to at least moderate, if not strong. So observation in a female with an otherwise negative exome is now evidence towards pathogenicity, whereas in V1 it wasn't. So we've also made some other updates. We've included this splicing table. This came out from Walker et al. So this is a little more granularity for canonical and non-canonical splicing variants with regards to their predicted effect and if any other variants around them were identified in affected individuals. We've also included now that affected females may be counted if they meet this phenotype specificity code, but that asymptomatic variant positive females should not be used as benign segregation evidence. So really, we've just added a lot more granularity that makes it easier to classify, easier and more consistent to classify variants in SLC6A8 in females. So we worked on this in the kind of interceding year. We've actually submitted this to the sequence variant interpretation work group. They sent it back and requested some pilot variants. So when you finish specifications, you actually have to test them on a group of at least 30 variants to make sure that known pathogenic variants are being scored and curated as expected, and benign variants the same. So we've done this. And we're ready to resubmit. These pilot variants are completed. We didn't see anything we didn't expect. The CCDS VSEP, the experts on our VSEP have approved these, and we plan it should be resubmitted with this updated pilots by the end of this month. So what's next? We want to continue the collaboration, obviously, with ACD and CreatineINFO, because this biochemical evidence that's being provided is incredibly important. We're recurating all of the variants that are not pathogenic or benign. So anything that's likely pathogenic, VUS, or likely benign, we're recurating. We're curating any new variants that come through CreatineINFO or pop up in literature or in ClinVar. Once SLC6A8V2 is approved, we're going to recurate all of the SLC6A8 variants with this updated guidance. And then something that's kind of looming in the field is the general variant interpretation guidelines. This is field-wide, not just for the CCDS. The last kind of major update was Richards in 2015, but they're kind of rehauling all of it. Adding more granularity, incorporating some of the things that we're already incorporating in the CCDS VSEP. But this is going to be a pretty huge change. We've updated SLC6A8 and GAMT kind of with the information we have. So those should be fairly easy to switch. But essentially, when this is released, we're going to have 1 year to update specifications for all 3 genes. To the new field-wide interpretation guidance. So that's coming, and we're preparing for it, so we'll be ready. So that's all I have. Thank you all for attending and logging on and hearing about our updates. And then we want to thank, obviously, the Association for Creatine Deficiencies. This collaboration has just been phenomenal on both a kind of a personal and a ClinGen wide scope. We want to sincerely thank the participants and families of CreatineINFO and Genome Connect for sharing your data with us. It's because of you that we're able to accurately classify these variants for the rest of the field for future CCDS patients. The ClinGen Community and Sequence Variant Interpretation Work Group and the ClinGen Stanford Informatics team who have helped with these specifications and similar. So that's all I have. And thank you again for your attention. And I can, I guess, hand it off.
Yeah. Thanks, Amanda. That was wonderful. I think this has been such an amazing effort and amazing collaboration between VSEP and ACD. And I think we'll get to a little bit of the maybe impact and questions during the combined Q&A panel. We'll bring you back on for that session. Next up, I think we're going to bring up a talk from Filippo INGOGLIA. Filippo couldn't make it live, so he has submitted a recording of his talk. While Colleen brings that up, I'll just give a brief introduction to Filippo. Filippo is a biochemical geneticist, assistant professor in the Department of Pathology at University of Utah, and a medical director at ARUP Laboratories. Dr. INGOGLIA completed his PA, MA, and PhD training from the University of Parma. During his training, he had the opportunity to visit University of Utah as a visiting fellow, and worked in the lab that was focused on rare inherited metabolic disorders, which piqued his interest in biochemical genetics and led him to apply for the Clinical Biochemical Genetics Fellowship. In 2021, he obtained the American Board of Medical Genetics and Genomics certification in clinical biochemical genetics, and has been affiliated with the University of Utah and ARUP Laboratories for a number of years now. In 2023, he joined the CDRC, or the Creatine Deficiency Research Center, funded by ACD and launched at the University of Utah. He will be providing an update on the study that he's working on to develop a functional test to confirm the diagnosis of CTD. Colleen, if the slides or the recording are ready, we can go to this talk.
Good morning, everyone. I'm Filippo INGOGLIA. I'm an assistant professor at the University of Utah and a medical director in the Biochemical Genetics Lab at the RUP Laboratories. It's a pleasure to be here today, although virtually, and showing you some of the data and results we obtained from the creatine transport assay project, a project funded by the ACD. And in particular, I'll show you some data on immortalized fibroblasts from patients with creatine transporter deficiency that are retaining minimal creatine transport. As I said, the project funded by the ACD, and I'll start introducing and describing the creatine transport assay, and I will conclude describing the creatine functional studies results on the immortalized fibroblast from patients with CTD. So probably, so I'm sure that most of the audience here does not need an introduction to the creatine transporter deficiency syndromes, but maybe some of you will find this short and quick introduction useful. So the creatine transporter deficiency is an X-linked disorder caused by variants, mutations on the SLC6A8 gene. It's a gene coding for the creatine transporter. Patients are presenting with developmental delay, intellectual disability, hypotonia, seizures, and autistic-like behavior. There is not therapy yet for this syndrome, and some patients, although can respond, however, can respond to creatine, arginine, and glycine supplementation. To fully understand the reason why we started the project, the Creatine Assay project, and we need to start talking about CTD diagnosis. So the diagnosis of CTD relies on the measurement of the urine creatine-creatinine ratio. If elevated, the diagnosis can be confirmed showing the lack of the creatine peak into the brain by the magnetic resonance spectroscopy and demonstration of presence of variants, pathogenic variants on the SLC6A8 gene. However, the path is not so smooth sometimes. So the MRS is not present in all institutions, but just a specialized institution have that type of technologies, and it is also technologies that require sedation of the patient. Regarding the gene analysis, the identification of VUSs and also identification of the presence of variants of the variants on a non-coding region can basically give an inconclusive gene analysis. So in those cases, a functional assay can definitely be useful, supporting the diagnosis of the creatine transporter deficiencies. So we started this project for developing these non-radioactive assays, not only to support the diagnosis of CTD, but also to support creating this assay to also support the classification of the variants. This is the workflow of the creatine transport functional test we developed. Is a 4 days assay, starting once we played the CTD fibroblast and normal control fibroblast in a 24-hour plate. Once they reach the confluence, we start the incubation using a stable isotope of the creatine. And after specifically 1 hour is the timing for the accumulation, we end the assay extracting the intracellular content using the ethanol. So the intracellular content is going to be prepared for the LC tandem mass spectrometry analysis, and the cells attached to the bottom of the plate, so of the wells, will be used for the protein quantification, and that quantification is going to be used for the normalization of the transport values. We start the development of the assay approaching the kinetic analysis. So the kinetic analysis showed a linear net creatine uptake up to 2 hours, with an optimal performance also at 1 hour. And 1 hour is the time point we select for the creatine functional assay. We also performed the KM, and we obtain a value of the KM of about 22 micromolar, that is absolutely in line with KM values previously published for the creatine transporter. So this was a first indication that the assay was promising, so actually was working. So the creatine uptake in a normal and CTD human fibroblast, this was the experiment that was proving the clinical utility of the assays. So we tested the normal fibroblast and CTD patients fibroblast. As you can see, the patients presented with significantly reduced activity of about 1% of the normal control. So this was confirming the CT1 deficiency and demonstrating the assay's clinical utility. As I told you in the introduction, so the creatine transport assay, the idea of developing these assays was also to support the classification of SLC6A8 missense variants. So to do that, we approach creating an immortalized CTD fibroblast. So we obtain from Coriell the primary CTD fibroblast with a null and absence of the creatine transport activity, and we transfected with using a lentivirus particles. Transfected those cells with a vector carrying the SV40 antigen gene. The SV40 antigen was conferring the immortalization to the CTD fibroblast. So basically, the SV40 what it's doing is disrupting the function of the P53 and RB proteins. So those are two proteins that are regulating the cell cycle and cell proliferation. So disrupting the function of those proteins basically is like creating a sort of tumor cell lines. Okay, so thanks to the CTSI lab at the University of Utah, we created through this procedure, so using the lentivirus vector and the SV40 gene, the immortalized CTD fibroblast. Once we obtain the immortalized fibroblast, the idea was transfecting those with a specific plasmid carrying the variants of interest, let them the immortalized cells expressing the protein of interest, and testing them with through the creatine transport assay we developed. So the first experiment, once we received the immortalized fibroblast, was comparing the creatine transport activity between primary CTD fibroblast and primary normal fibroblast with their immortalized version. And here on this chart, you can see to the left the primary cells, normal and CTD patients, and to the right, after the immortalization, this is the creatine uptake, so the creatine transport activity. We notice a 5-fold increase in creatine transporter, in the normal fibroblast, whereas those from CTD patients retain a minimal creatine uptake. And confirming and that and proving that also in the immortalized after the immortalization, so the creatine transporter deficiency was basically maintained. So the second experiment, after we tested the activity in the immortalized cells, so the second experiment was to transfect those cells with the plasmid vector carrying, where you see here the open reading frame. So we have the SLC6A8 gene, so carrying, so transfecting the immortalized cells with the plasmid carrying the variants of interest. So here it is important pointed out to the neomycin resistant gene. So this is a gene that is conferring the resistance to the neomycin antibiotics. So, and is the gene through, so the function of the gene will allow us to stable transfecting and selecting the cells that receive this plasmid. So The intention was immediately starting with a stable transfection and testing those cells after 20 days of selection in presence of the geneticin. So the problem was that we immediately noticed a very high mortality rate in the immortalized cells, and a proliferation rate drastically reduced of the immortalized cells. So it was impossible for us to test the creatine transport in the stable transfected cells. So the stable transfection is under investigation, and we are trying to troubleshoot those and overcome those difficulties. But in the meantime, we noticed that up to 3 days and before starting the selection with the geneticin, we were having a good number of cells, and we thought that the transient-3 transfected experiment, so measuring the creatine transporter, the creatine transport activity on transiently transfected immortalized cells could provide us, good data to prove our intention. So the utility of our assay also in supporting the classification of the variants. So here we have the results we obtained. So we test, as you can see, different cells. We had the normal immortalized cells, normal fibroblasts immortalized, and here we have the activity of the creatine transporter. Here we have CTD fibroblast immortalized with basically null activity. Then 2 CTD immortalized fibroblasts expressing a wild type CT1, and as you can see here, we had a very significantly increase of the creatine transport. And we also include in this experiment 2 well-known variants. One is the P544L variant, that is a well-known variant that is retaining a residual transport activity, and another well-known pathogenic variant, the P382L. As you can see here, we have these cells transfected with the P455L variant, the presence of a residual transport activity. So yeah, although is generated a small transport activity, but it is a significant increase. Comparing with well-known pathogenic variants, and compared to the our immortalized CTD cell lines. And so this is a very promising result, although it's just on transiently transfected cells. But with this experiment, we get the confidence that this assay that we developed could be used to distinguish between apomorphic and functionally null variants. So I will conclude here saying that we have developed a novel and non-radioactive assay to measure creatine transport, and we also developed immortalized cells lacking a creatine transport, which can be used together to assess the functional activity of missense variants identified in patients with CTD. So thank you everybody for your attention, and I'd like to thank first of all the ACD for the support, and University of Utah and ARUP, and my colleagues, so Shilu, Kumari, who is hardly working with me on this project, and Dr. Longo, Dr. Pasquali, and for their support, and also for their support and sharing general knowledge. Dr. Kent Lai and Vijina Balakrishnan. And of course, I would like to thank all the patients and their family. So thank you again.
Thanks to Filippo for pre-recording his talk. Filippo is not able to join the Q&A today, but I encourage folks to reach out to him by email if you have any questions. Please do put your questions in the Q&A box so we can share it with him. And for our former speakers, Amanda and MAAIKE, if you're in the audience, please continue to stay there. We'll bring you to the Q&A panel after our last talk in this session. Our last speaker for this session is Emily REINHARDT. I will wait for Emily to be brought up as a panelist while I give her introduction. Emily is ACD's patient registry coordinator. We're fortunate to have such a dedicated staff member working on our registry for the past couple of years. Our patient registry launched in 2021, and since then the registry has provided us with a path for engagement and contribution between CCDS patients and caregivers. It is also the way we collect information from our community, and it is an invaluable resource for new and existing CCDS investigators. And coordinating all of that is Emily. She'll be giving an update on our patient registry. With that, I'll hand it over to you, Emily, to get started.
Thanks, Sangeeta. Thank you, everybody, for being here today, taking some time to allow me to give an update on our registry. So we'll just jump right into it. Just high level, I thought since we've had quite a few new members of our community, it might be beneficial to just talk about what is a patient registry. I'll give an update on what's new, highlight how the registry supports clinical trial readiness, and then talk about what's next. So patient registry 101, what is our patient registry? You've heard us mention CreatineINFO. CreatineINFO, the patient registry and natural history study, it's a patient and caregiver reported registry that's used for the collection and dissemination of standardized information from CCDS patients. Specifically, our registry collects longitudinal patient information to help families and researchers better understand how CCDS progresses over time. So why is a patient registry important? First of all, it improves our understanding of the natural history of CCDS. It helps doctors and scientists and industry partners to develop treatments and improve patient community outcomes. It helps us at ACD as an organization to better represent the CCDS community. And finally, it shares community reported recommendations and standards of care. So I know that there's been quite a bit of discussion about registries in kind of the news and culture, especially here in the United States. So I just wanted to answer the question, is our patient registry a real research study? And yes, it absolutely is. We have approval from what is called an institutional review board. In other countries, it might be called an ethics committee or a research ethics board. And essentially, this institutional review board, or IRB, is a third-party entity that reviews and approves the initiation of and conducts periodic reviews of our registry. And their primary role is to ensure that they are protecting the rights and welfare of study participants. So in this case, CCDS patients. Everything that we do in our registry, from surveys that are released to social media, language that's put out, is all carefully reviewed by this third-party entity to make sure that we are conducting research correctly and keeping the safety and rights of the participants intact. Just for some transparency, Heidi Wallace, our executive director, she's our principal investigator, and our IRB protocol is here. We use North Star Review Board. You can visit their website if you have questions about our registry and the protocol and things like that. So our registry, we have a partnership with the National Organization for Rare Disorders, or NORD. Our patient registry is hosted on their registry platform. It's called I Am Rare. And I really love this quote from their website. By harnessing the power of data and patient-reported experiences, I Am Rare studies are informing clinical trials, new treatment methods, and transformative change in the rare disease community. And that's absolutely because of them, we're able to do what we can in our registry. So how are we protecting your data? How are we protecting patients and the information that they share with us? All of the data is stored on password-protected, encrypted servers that are hosted in Canada. These servers are compliant with US and international regulations, including GDPR compliant. Only ACD's registry team and NORD support staff have access to your personal information. We do not share information like your name or your birth date or where you live with researchers who might be requesting data. It's all de-identified. A really important thing is that ACD owns all registry data. So we are in control of that data, and through the use of our registry oversight board, which is made up of parents and clinicians and researchers in our field, they make the ultimate decision about what data are shared, how they're shared, and who we share those data with. Again, just trying to protect the patients at all costs. So now that we all know what is a patient registry and why it's important, I wanted to give an update on what is new. And the biggest, most exciting thing that's happened since I spoke with you last summer is that CreatineINFO has migrated. This has been a long process, but last month we migrated to an updated 2.0 version of our registry platform. If you were already enrolled in our registry, you should have received an email with an activation link. I will resend those links over the next day or two, so it's fresh in your inbox. So please, if you were previously enrolled, please use that activation link because it will then connect you with all of your previously recorded data, because all of that information was migrated over. You do not want to have to start all over, especially those who have been in the community for a while and have taken all of your surveys. Use that activation link and it'll pick right back up where you left off. Just to give you a little sneak peek of what the new platform looks like, when you, as most likely as a parent or a caregiver, you create an account, you'll see some sort of screen like this with an opportunity. If you're new, you would register and you can add a participant. A participant is the patient. So if you have a child with a creatine deficiency, you would add them as a participant. If you were migrated over, you would see those existing profiles here. So in this case, I have, in this example, I have 3 children with a creatine deficiency: Agent Arginine, Captain Creatine, and Green Glycine. If you need to add someone else, you can add a participant. You can find the individual profiles here. And again, just as a reminder, each CCDS patient needs their own participant profile. So if you had 2 GAMT kids, you would need 2 profiles, and so on. So let's say then I clicked on Captain Creatine's profile. You would see here that you are enrolled in the CreatineINFO registry. Technically, this is an account on the I Am Rare platform, so you could enroll in other registries that are on this platform. Say if there was an epilepsy registry that you wanted to enroll in, or an autism registry that was available, you could do that and you could click search studies. But if you want to look for our creatine specific registry, you're going to go to CreatineINFO here. As a reminder, you will need to consent prior to enrolling in the registry. Again, because we are a real research study, you need to consent to participate. So once you're into our registry, again, you're under Captain Creatine's profile. This is where you will see where all of your surveys are. And so you can see surveys that you've already completed. You can look at your previous responses. You can start and stop surveys if you need to step away. And then you can see that the other surveys that you have yet to complete. So really excited. This is a much better user platform, both on the front end and on the back end for us. So now that we've migrated, just as a reminder, we have been around since 2021 is when we launched. We have tons of information, tons of data. We have a very active patient community that have graciously shared their data. And we have over 200 participants in our registry from all 3 creatine deficiencies, with a wealth of data on demographic information, their diagnostic odyssey, their phenotypes and symptoms, genotype and variant information, like what Amanda mentioned in her previous talk. We have some specialized information about cardiovascular health, oral medication preferences, patient meaningful outcomes, with a lot more information coming in the days ahead. If you want to take a kind of a high-level look at what's available, at the end of last year, we did publish we put together a 2024 annual registry report. You can find that, the QR code here. And these are just some screenshots of it's a really kind of easy to digest way of just learning about our community. You can share it with your patients, take it into your doctor, new families. It's a way to kind of learn about these conditions. And so I really encourage you to check it out. It's a great resource that will continue to grow and expand every year as we collect more data from our community. So clinical trial readiness. One way that our registry has an impact is preparing our community for clinical trials. And Sangeeta mentioned this during her opening remarks, but clinical trials, getting ready for clinical trials is more than just creating a new drug or developing a gene therapy. There are a lot of different puzzle pieces that need to happen simultaneously and come together for an effective and efficient clinical trial. And some of the ways that CreatineINFO, our registry, is tackling that is through the use of a core outcome set, clinical trial measurement tools, which you will hear more about over the course of the next couple of days. And then, of course, helping us better understand the natural history of creatine deficiencies. So we really are focusing our registry to build a registry that supports clinical trials. So as a reminder, in ACD's first Parents Project, we developed a core outcome set for CTD and GAMT deficiency that includes these 8 outcomes that our community came together and said, you know what, these are the outcomes that are most important to our community and really should be measured in every clinical trial. So our registry is preparing for clinical trials by using this core outcome set to really guide the types of data that we collect in the registry. And so this way, there will be a resource of data for researchers to use as they develop their clinical trial goals and endpoints. And so they can compare clinical trial data with registry data. So we're kind of using this as kind of our compass to guide some of our decisions. And so I want to just take a step back and talk specifically to our patients and our caregivers in the community. At times, it probably feels really isolating to be a rare disease patient or caregiver. It's likely that you may be the only family with your diagnosis in your city or your state or even your country that you're aware of. But I really hope that at the end of these next 2 days of talks that you don't feel so alone. Because even though creatine deficiencies are rare in numbers, when you share your individual stories, you can quickly see that there is power in the collective. And so your individual experiences can have a huge impact on this community. And it's through the combination of all of these individual voices, like being active and participating in the registry and sharing your experiences with researchers, is how we can get ready for clinical trials and really advance the field for a treatment for these disorders. So what's the first thing that you can do? You can take a really easy, quick step today. We launched a new survey on the registry this week. It's called the Clinical Research ID, or CRID survey. The CRID is a patient-generated identifier that's used for clinical research. Basically, it's just an ID number, but it allows researchers to reuse, merge, and share de-identified research data sets with other research studies. So it allows us to be able to link up data that maybe you shared in the registry with perhaps a natural history study like Vigilon, or another clinical trial that you're participating in. If an example would be like the FNIR study, potentially down the road, we could link up all of these different data sets to really build a fuller picture. So it's really easy. Log into the registry, join the registry. You'll see a survey there. It takes 3 minutes to create an ID and then share that information with us. So what's next? CreatineINFO, we are going multilingual. Currently, the registry is only available in English, but French and Spanish translations are underway, which is really exciting. And just a heads up, German, Italian, and Brazilian Portuguese will be the next languages that are added to the platform. So this is really going to expand the accessibility of our registry and really drive us even further. And again, circling back to how we support clinical trial readiness, our first core outcome set focused registry survey is going to be the seizure survey, and that is coming out this fall very soon. It's being built as we speak. And we're going to collect information like seizure types, your first seizure, types of medications and diets and surgeries that you've tried and what's worked, your EEG history, family history, and this survey will be longitudinal. So you'll get follow-ups every 6 to 12 months of a new survey with just kind of checking in, again, trying to paint a broader picture of what do seizures look like in the creatine deficiency community. We're also teaming up with the Parents 2.0 project, which you'll hear more about over the course of the next 2 days. But we will be launching another survey this fall. It's the Parents 2.0 community survey. And in this survey, we're really it's specifically for patients and caregivers, but we'll be asking you to identify what does meaningful changes look like in each of those 8 core outcome sets. What would that look like, a change in those outcomes look like in a clinical trial? So stay tuned for that. And so finally, as I wrap up, just a reminder, like, how can you get involved? Well, patients and caregivers, first things first, if you haven't joined already, join our registry. And then once you've joined and you've consented, complete all your surveys. And then finally, uploading your genetic and laboratory reports. Like Amanda mentioned in her talk, we really are supporting multiple efforts. And one of those is the CCDS VSEP. So really focusing on uploading reports that you receive or that you had done when you were first getting your diagnosis, blood work, MRS, things like that, before you began supplementation is really beneficial to us. Clinicians and researchers, invite your patients to join. Request data from us. We would be happy to share data with you. Maybe you're working on a manuscript or you're submitting a grant application and would like to supplement those with community data. We'd love to help you. We've worked with several researchers in the community to be able to get them the data that they need, and it's been really beneficial for everyone. And then finally, collaborate with us. Maybe there's some data that you're interested in, but we don't have in our registry. Work with us. We have experience developing custom surveys with partners, and so we'd love to help and support you in any way that we can. So I'll just wrap up here. If you have any questions, please let me know, reach out. This is my email here. Again, this is the QR code to join the registry, and I will pass it back over to Sangeeta. Thanks, everyone.
Thanks so much, Emily. That brings us to the first Q&A of today. I think we have all of our panelists and speakers as well as our moderators ready. So for if this is your first time attending this conference, then I just want to note that our Q&A is managed a little differently. We have two moderators who will be asking questions of our panelists. One of our moderators is a community member. For the purposes of this Q&A, we have Dan Collar, our board chair, and then we have Jenny Goldstein. And I'll leave them both to maybe give a brief introduction before we dive into a Q&A. I request all panelists to keep their videos on if you're comfortable, and if not, just audio on would be great. Okay, over to you, Jenny and Dan.
After you, Jenny.
Oh, thanks, Dan. Hi, everybody. I'm Jenny Goldstein. I work at the University of North Carolina, Chapel Hill, and I've had the pleasure of working with many families with creatine deficiency syndromes when I was a genetic counselor working at Duke. And right now I'm working with the Clinical Genome Resource, work with Amanda on the variant curation expert panel. So just delighted to be here today and just really appreciate all of the efforts that the ACD puts into research and supporting everyone. So great to be here.
And hi, everyone. I'm Dan Collar. I live in San Diego. I have a 10-year-old son, Cadman, with CTD. I've been the board chair with ACD. This is my third year. And it's great to see everyone reconvene year after year and familiar faces and new faces. So great to have everyone here. So I think we'll start off with the first questions, if that's all right. Emily, there's one for you, and then I actually have one. So I think maybe if we do those, since it's fresh in our mind. The first one is, will it be helpful to take the seizure survey if your child has not had a seizure?
This is a great question. Yes, absolutely. And that's the case of whether it's a seizure survey or the cardiovascular survey or anything. There's an option. One of the first questions that's asked is, does your child have a seizure? And sorry, I'm getting some feedback somewhere. It's a yes or no question. And it's really important to hear from the no's, because either one, your child may not have a seizure and may never develop seizures, or it may change over the course of time. And like I said, the seizure survey and several other surveys will be longitudinal. And so we will send out surveys over the course of every 6 to 12 months. And it's helpful to us to see, maybe you don't have seizures during early development, but then we hear quite a few families as we approach puberty, we start to see an increase in seizure development. And so that's important for us to see. So yes, absolutely take the survey. If you don't have seizures, it'll be a very fast survey. I think you're muted, Dan.
Thank you. I can relate as a parent of a 10-year-old whose seizures seem to have changed over time. So that's good info. We're getting quite a few now. I'll read the next one and maybe then pass it off to Jenny. This is from James in England. Great to see the registry evolving. Question, it would be really helpful for me as a parent to see aggregated and anonymized results from the seizure survey to understand what medications other parents have tried. Would that be possible?
Absolutely. We want to be very transparent with the data that we collect, especially returning that back to our patients and caregivers who so graciously share their data. So it'll be updated in the yearly annual report. We'll share updates as we collect that information through the year. You can also, once enough responses are collected in the registry platform, you can actually view responses when you're in the registry. So you can see graphs. There's basic graphs, but there is some information in the registry that you can see. Again, there's a minimum number of responses that need to be collected. And then we've also just talked about the possibility of we, as families, reach out to us. If you have specific questions or are looking for a specific piece of information, reach out, ask us. We'd love to help and support you, especially if you have questions about specific medications or your doctors are needing some more information. We'd love to share that information with you. That's a great question.
Okay, actually, it looks like the rest are more for the other presentations. So if it's okay, I just want to ask the question I had real quick, which is, from your experience with people who are coming into the registry, is there a common thing that people have said, oh, this is why I haven't done it yet? And maybe the one thing you can say, it's really easy, like don't have any fear or anything. I'm guessing privacy is a big thing, which you might have to alleviate.
Yeah, privacy is can vary, especially depending upon where you are in the world. There's different perspectives on sharing personal data, which is completely understandable. And if you have questions about that, or you want to talk more in depth about, how are your data being protected, I'm happy to answer those questions, have a conversation with you. I tried to share some of that today. I think it's just also the biggest thing is just time and bandwidth. The parents and families in the community, you have limited time. Some days it's just, it's a struggle just to, get everybody dressed and out the door, and let alone sit down and remember my password and take these surveys. And it's a lot. And I know that we're asking a lot from you. And it's okay. It's okay if you can't do it right now. But I hope that even if you can just enroll, just enroll, and you're in the system and we know that you exist, and you can receive updates from us, and you'll get to it when you get to it. And if you ever have questions or if you need help, I'm happy to sit down and walk you through it. If it's a language barrier, maybe your language isn't available yet, we can work with you. We have worked with others in finding a translator to sit down and help you with that. So yeah, I think really, Dan, to answer your question, I think the biggest thing is just time. And you'll get to it when you get to it, and that's okay.
Well, my last plug is going to be anyone who hasn't been on the registry yet. Yes, it's a little bit of work, but if there's one thing you can do as a parent or caregiver that will help, no matter where you live, this really is it. This is part of the baseline. This is the foundation of a lot of the stuff we do. So I'd really encourage everyone. Okay, and I'm going to see if Jenny wants to pose the next question.
Yeah, sure. Thanks, Dan. And I completely agree. I'm kind of coming from the other side of things as a member of the Variant Curation Expert Panel. I mean, the data that the registry collects is just incredibly important and helpful, as Amanda's talk showed. And then also, as Emily was discussing, readiness for clinical trials and just increasing understanding of these disorders. So we so much appreciate your participation in the registry. And as you've seen, of course, Emily is just wonderful at coordinating the efforts and talking with you about how to participate. So just really wanted to put my plug in as well. And to thank Emily and all the great speakers this morning. So the next question is, I think, for Amanda. So how are new variants identified in the literature not coming from the patient registry? And is someone from the VCEP keeping tabs on all new CCDS papers? So Amanda, I'll give that one to you.
Yeah, and of course, Jenny, who's the co-chair of the VCEP, please feel free to, obviously answer as well. But so literature, yeah, we do survey the literature. Many of the kind of molecular experts are constantly reading literature. So I'm a molecular geneticist, so I do genome sequencing. So I'm constantly in the literature looking for other variants. And if I come across one, we get it from there. There's also an ongoing initiative. There are some papers with GAMT variants specifically that are in Chinese. And we have someone who's volunteering with us who are translating those papers. So that's a whole new group of variants. But yeah, when we come across them in the literature, we can take a look at them in the VSEP. And then I think in general, we're all interested in the CCDSs. So we're all keeping tabs on new variants, new papers, new functional studies, mouse models, all of these things.
Yeah, fantastic. Yeah, I agree, Amanda. It can be a little bit of time to keep up with things, but it's really, really great to just increase the number of variants that we're curating. And also because we submit those to ClinVar, which is the public database for variants, then they become available to everyone. So it may be that a lab hasn't yet submitted this variant to ClinVar, but through our work we do, and then that increases the knowledge overall. So it's really, really nice. So I don't see any other questions in the chat right now, but maybe another question for Amanda. I thought that Dr. Ingoglia's talk was really interesting, especially thinking about the use of functional studies for SLC6A8 variants. And I just wondered if you had any comments on that regarding variant classification and better understanding of these variants.
Yeah, it's really exciting what they're doing in the lab. And I think when that's published, that will give us a lot of additional weight for functional classification. With the number, so the way that we look at specifications for functional classification, it really depends on how many known pathogenic and known benign variants. And Dr. Longo and Dr. Ingoglia are very tuned into the VSEP. So when that paper comes out, I imagine it will be done in such a way that we'll be able to apply fairly strong evidence for pathogenicity for functional studies. But yeah, we are excited, and it will help significantly in variant classification. I do see a comment, X-linked variants with no phenotype in the Q&A. Yeah, do you see that one?
Yeah, that's right. It's Jenny, the questions are in the Q&A, not in the chat.
Oh.
Okay, I see now. Yeah.
Yeah, I just wanted to clarify. So I guess the question is that X-linked variants with no phenotype are not classified as benign, and kind of what the assumption is that they may be recessive. So I just wanted to clarify a little bit. So there's X-linked variants are incredibly complex for multiple reasons. So if we see an SLC6A8 variant in a male, so males only have one X chromosome, right? So they're considered hemizygous. So that means this variant is the only one they have. If we see an SLC6A8 variant in a male with one X chromosome who does not have biochemical evidence of creatine deficiency syndrome and does not have phenotypes, that would actually lean towards the idea that that variant is not causing CTD. So that would be benign evidence. In females, it's different, because in females, there's a whole nother layer of complexity. So first off, females will only have one copy of the variant and another X chromosome that's we got time's up. That's presumably has a normal copy. But in females, something else happens called X inactivation. So one of the two copies of the X in each cell is kind of randomly inactivated. And normally, under normal circumstances, it should be a 50-50 ratio, but in some cases, that's skewed. So it might be 60 to 40%, 30 to 70, so on and so forth. So in females, as I mentioned, there's a very large variety of, CTD phenotypes you can have. You can be completely asymptomatic, or you can be severely affected if you're a female. And we think the X inactivation has something to do with that, and it could be other genetic modifiers. But certainly in a female with two X chromosomes and one SLC6A8 variant, we don't count that as benign, because we know that there are asymptomatic females. So yeah, I just wanted to kind of clarify that. It could be in conjunction with other variants. So this would be like genotype variability. It could be. And I think that field we're still learning. But for now, with what evidence that we have, that's, I hope that makes sense. If it doesn't, feel free to reach out. I'm available through the CCD VSEP. My email's addressed there. Or Heidi can get in touch with me.
And that's actually another question, Amanda. Is it possible to get a personal consult for a greater understanding of
Yeah, so I mean, on a personal level, kind of outside of my work and my job, I'm happy if anyone wants to email Heidi like your variant de-identified. Of course, if you just want me to kind of explain what's happening, I'm more than happy to do it. It just just bear with me and give me some time, because all of this is kind of in my like free time. But yeah, if you want some more information, or if I can help explain what we think's going on with the variant specifically, sure, I'm happy to do it. So just shoot Heidi an email and she can forward it to me, and yeah, happy to.
And I don't know if we have time for one more, Sangeeta.
That's all right. I do want to get Micah's question.
Yes.
Yeah. So Micah, maybe we'll start from the first question you received, which was from Dr. Longo. If you could just share if you saw any improvements with the supplements and the dose of creatine that you tried. Jenny, back to you for the last one, and then I'll take over after Micah.
Well, that's a difficult question to answer, of course, because we don't know what the supplements are doing exactly, and what is his normal development, and what is all the extra effort we're putting into his therapies and attention we're giving him. The only thing we know is that the MRS did not show an increase in the creatine levels in the brain, and what we kind of objectively measure is his developmental milestones and the developmental age, and we see an increase, and a maybe slightly steeper increase than before the supplements, but that's pretty subjective anyway. So yeah, do the supplements work? We don't know, but as long as we don't have any negative side effects and he's happy to take them, then we are just gonna continue using them. And our pediatrician is keeping a close eye on the negative side effects and the kidney function, and that's why we're having blood tests done every 4 months, slightly readjusting the dosage of the creatine and the arginine and the glycine. And yeah, we do it because there's nothing else we can do.
So there was
Yeah,
go ahead,
go ahead.
Sangeeta.
No, go ahead, Jenny.
I was just going to say there was another question for Micah as well, about one piece of advice you would give to new CCDS families who are new to this community.
Yeah, I don't know if I'm there long enough to give advice, as I feel as one of the newbies in this group. But yeah, I think it has been of great help to me to just reach out to the ACD and to try to get involved and to hear other stories. And even if you're not willing to share your own story yet, just listen to other people's story, find people on social media, and yeah, see what it is that you need at that time, and go find that. But don't push yourselves to things you're not ready for yet. If I look back at the past year, where it has been really a roller coaster of getting the news and having to deal with all of it, yeah, just take your time processing it all. That's, I guess, the best advice I can give. And we're not nearly there yet, but we're doing much better than we were a year ago.
And I think having a forum like this, just for people to share their stories and just know there are other folks out there going through similar circumstances, is just really fantastic. And you gave a wonderful talk, and Oscar is so cute. He's adorable. So.
He is. Thank you. Thank you for inviting me to speak here as well. Indeed, it also helps. Just, presenting this story already, just summarizing the past year was already helpful for me in dealing with it. So thank you, and thank you for listening.
That's wonderful.
Thank you, Micah. Thank you, Jenny, Dan, and all of our speakers for this session. That was a great first session. We're going to keep things moving and dive into our next session. We'll be kicking off session number 2 for today, which is learnings from gene therapy advances in mouse models. The first talk for this session will be given by Laura Baroncelli. Laura is not able to join the talk live, but she will be live for the Q&A. So while her slides are being brought up, I'll give you a brief introduction to Laura. She's a senior researcher at the Neuroscience Institute of the Italian National Research Council, or CNR, in Pisa. Her research has focused on understanding neurobiological mechanisms, understanding of rare diseases, with a particular emphasis on CTD, Angelman, and Rett syndromes. Unique to to Laura is that she actively leads and participates in multidisciplinary projects to advance novel treatments as well as diagnostic tools in cross-country collaborations. Laura's been a member of the CCDS community for a number of years now, and it's really excited to hear from her lab on the progress towards gene therapy for CTD. And we can go ahead and hit start on the recording.
Okay, so good morning or good afternoon, everyone. Thank you. Thanks to the organizers one more time for inviting me to speak at this symposium. It's a real pleasure to be here, even in a virtual mode, especially because this event is particularly meaningful to me. Today, I'm excited to share some updates on one of the core projects in my lab, which is the development of gene therapy approach for treating transporter deficiency. My talk will be divided in two parts. In the first one, I'll present the key findings from our recent study, which is the first experimental gene therapy approach we tested for CTD, which has been just accepted for publication in Brain. In the second part, I'll focus on how we are building on these results to advance the project, refine our strategy, and move, hopefully, a step closer to a therapeutic solution. But first of all, I would like to acknowledge and thank my research team. None of the results I'm presenting today would have been possible without their passion and dedication. And I'm really grateful to all of them because we have been working together on CTD for several years by now, and it's their expertise and commitment that continue to push this project forward. So our overarching goal is to develop a gene therapy approach that addresses the root causes of CTD and ultimately improves the quality of life of patients and families. To this end, we are testing whether a single intraventricular injection of a viral vector carrying a functional copy of SLC6A8 gene can rescue the pathological features observed in the CTD mouse model. So we began by engineering a construct encoding the human coding sequence of SLC6A8 tagged at the C-terminus with an HA tag. And this sequence was placed under the control of a jet promoter, which is a synthetic element known to drive strong and widespread gene expression in the mammalian brain. Then the construct was packaged into an AV9 vector, chosen for its proven ability to target the central nervous system and administered via intracerebroventricular injection in newborn mice. To test our gene therapy approach, we rely on the SLC6A8 knockout mouse model that we have generated and fully characterized in the last 10 years, which serves as a primary experimental platform. This model shows a very strong phase validity with the human condition, displaying profound brain creatine depletion, along with the constellation of symptoms that mirror the clinical phenotype. Indeed, knockout mice display global cognitive deficits, stereotype behaviors, partial motor impairments, and epileptic phenotype. And more recently, thanks to valuable collaboration with Alessandro Gossi's team at the Italian Institute of Technology, we uncovered a widespread pattern of hypoconnectivity or functional hypoconnectivity in the brain of these mice. These findings suggest that the consequences of creatine transporter deficiency go beyond the alteration of cellular enemy metabolism and impact large-scale brain network organization and synaptic connectivity. So leveraging this robust mouse model and these outcome measures, we have initiated our first preclinical study on gene therapy. And in our initial experiment, we examined brain tissue at postnatal day 40 to determine whether the therapeutic gene was being expressed. Because there is currently no reliable commercial antibodies to detect the native creatine transporter, we use the HA tag epitope that I mentioned before as a proxy marker. As you can see from this Western blot analysis, we had very encouraging results. Indeed, we detected a robust expression of the HA tagged creatine transporter, so the transgene, in the brain of treated animals, confirming that the viral vector had successfully delivered the gene and enabled its expression. In a separate cohort, we also used immunohistochemistry to evaluate the biodistribution of the therapeutic gene expression across the brain. We observed strong and widespread expression throughout multiple brain regions, as you can see from these representative images. As expected, the HA signal corresponding to the transgenic creatine transporter showed a punctate membrane-associated pattern consistent with the native localization of the transporter. Here, I'm showing to you the data only from the knockout mice, but similar results basically was seen in both genotype. So strong expression was especially evident in deeper layer of the cortex and in some key subcortical areas such as the hippocampus or the striatum. And when we quantified the transduction efficiency of the viral vector, we found an average of about 8-10% across all animals. And while there was some regional variability with slightly lower levels in deeper structures, this pattern is consistent with prior studies using AAV9 delivered intracerebroventricularly in newborns. So to gain insight into which cell types were targeted by our gene therapy, we also performed double immunolabeling for the HA tag, so representing a proxy for our transgene, and markers specific to the major brain cellular populations. We used MAP2 for neurons, GFAP for astrocytes, IBO1 for microglia, and BCAC1 for oligodendrocytes. And what we found was interesting because the vector showed a very strong preference for neurons with a clear colocalization between the HA tag signal and the MAP2 signal. We also detected a significant transduction in oligodendrocytes and microglia. However, we did not observe any colocalization with GFAP, indicating that astrocytes were likely not targeted by this treatment. Most importantly, the transduction led to a sustained and stable increase in brain creatine levels, while total creatine in the SLC6A8 knockout mice remained about 50% lower than in wild types. Reintroducing the functional transporter after birth through our gene therapy resulted in a partial but significant restoration of cerebral brain content, and this increase was stable over time. And this is encouraging because it suggests that even a moderate level of transporter expression is enough to produce measurable biochemical improvement in vivo. These encouraging results prompted us to launch a multi-site preclinical trial, once again in collaboration with Alessandro Gozzi, to rigorously evaluate the therapeutic potential of our gene therapy in reversing CTD-related phenotypes. As illustrated in this timeline, the study included longitudinal imaging and behavioral assessments at multiple time points in mice treated at postnatal day 1. As an initial outcome, we observed that the treated knockout mice exhibited a small but clear increase in the body weight both during the juvenile phase and adulthood compared to control knockouts. And this indicates a general improvement in overall health of these animals. Secondly, we found that early gene therapy completely prevented the widespread hypoconnectivity seen in the brain of knockout mice. In fact, we observed a significant increase in functional connectivity across both neocortical and subcortical regions that are most affected in knockout animals. And here you have this graph displaying all these red lines and these red bars that basically shows that there is hyperconnectivity in treated knockout animals compared to control knockouts. So this means that gene therapy rescues brain connectivity. At the behavioral level, in addition, we saw a striking absence of autistic-like behavior in treated animals. Indeed, knockout mice that received the gene therapy showed a significant reduction in grooming time compared to knockout controls. And grooming is a very used proxy for assessing autistic-like behaviors and stereotype movements, motor rituals, and routines in mouse models. Moreover, we observed that the AAV treatment significantly improved the cognitive performance of knockout mice during the object recognition test, at least postnatal day 100, although no significant benefit was found at postnatal day 40. The object recognition test is a well-established test of declarative memory, that kind of memory involved in recognizing and recalling facts or events. And in this task, as you can see here, animals are first exposed to two identical copies of the same object, and then, in our case, 24 hours later, they must recall that familiar object and tend to spend more time exploring the new, never-seen object. And here we found that treated animals were able to partially recall the familiar object, at least at P100. Indeed, the discrimination index, so basically a number representing the time exploring differentially the new and the familiar object, is significantly higher in treated knockout animals compared to control knockouts that equally explore the two objects, not recognizing the new one compared to the old one. However, in the other cognitive tests, such as the Y maze, treated knockout mice perform completely similar to the control knockouts, indicating that gene therapy had limited effects in this aspect. The Y maze is a widely used test to assess working memory, which plays a key role in the adaptive everyday behavior. And in this test, we measure spontaneous alternation in exploring the different terms of the arena, which is a natural tendency of mice. So you can see here that the percentage of spontaneous alternation is totally comparable in treated knockout mice and controls. So just suggesting that the gene therapy we developed is not fully successful to rescue all the endopathological phenotype of CTD mice. Moreover, we also noticed a slight decline in cognitive performance of wild-type mice receiving the gene therapy at least at P40, suggesting that the overexpression of SLC6A8 might have unintended negative effects on healthy brain circuits. So taken together, our findings offer for sure an encouraging proof of concept demonstration that gene therapy could be a promising treatment for CTD. However, we strongly believe that further optimization of the viral vector is essential to enhance therapeutic efficacy and ensure safety before moving forward with testing in additional animal models, and of course, before moving to the clinics. So, thanks to a bilateral agreement between our institute, the CNR, and the Royal Society of Edinburgh, we were able to start a very fruitful collaboration with a group of Stuart Cobb, which is a leader in gene therapy studies in Europe. And together, we designed 3 novel genetic constructs to help refining our therapeutic strategies. As the literature shows that CRT expression in the brain is both very low and highly cell-specific, we explored 3 complementary strategies. And each of these constructs uses a different regulatory sequence to drive the expression of the exogenous SLC6A8, aiming to closely mimic the physiological expression of a CRT. We began preliminary in vitro screening of these plasmids using primary cells from our mouse model, but here I should mention that due to some technical issues in our institute's cell culture facility, these initial experiments have been limited to primary astrocytes. And moreover, you see that some experimental groups are still missing from certain assays, And also, we haven't yet assessed in vitro toxicity. But that said, the initial results are very promising. Indeed, Western blot analysis of epitope tagged proteins, as usual, shows that the FF351 construct leads to a significantly lower transgenic expression compared to the JET approach, while the FF382 construct gives an intermediate level, and this pattern was very consistent in wild type and knockout cells. Then we also find consistent results with immunocytochemistry, which basically confirm the differences in the expression levels between the two constructs. In contrast, the analysis of creatine uptake gave us quite different data. We first looked at non-transfected astrocytes, actually, both wild type and knockouts, and as expected, knockout cells showed no response even when simulated with creatine. However, wild type cells exhibited a clear increase of creatine uptake after the simulation with external creatine, indicating that the creatine transporter, a native creatine transporter, is functionally expressed in these cells. And while these results still need to be validated, in my opinion, I think that they are very interesting, especially in the context of strategies, on the context of studying strategies for restoring gene expression with a transgene. So we then evaluated our constructs in knockout astrocytes, and what we found is that while the FF351 led only to a modest increase in creatine uptake, an increase that was even lower than what we observed using the endogenous promoter of the SLC6A8 gene. Both the FF382 and the FF391 constructs showed uptake levels comparable to what we observed with the JET promoter construct. And this suggests that these two plasmids may be too strong, and further refinement of their regulatory elements could be needed to fine-tune expression and avoid potential overexpression. That said, I would like to stress one more time that these are preliminary findings because we need to replicate the experiments in other cell types, and the same regulatory elements might have very distinct results in different cell populations. And moreover, we still need to assess the cellular viability to get a more complete picture of what happens after the transduction with this construct. However, based on the encouraging preliminary results we obtained with the FF351 construct, we decided to move forward with both in vitro and in vivo studies in parallel. We produced the viral vector and began testing its efficacy in P30 animals, and focusing first on transgene expression and creatine levels in various brain regions, specifically the cortex, the hippocampus, the striatum, and the brainstem. As previously reported in the literature for intraventricular administration, the injection in the ventricles of the FF351 virus led to very good levels of transgene expression in the cortex, hippocampus, and striatum, as you can see from this Western blot, although the expression in the brainstem was lower. And we did observe some variability, especially in wild-type animals, likely due to the combination of physiological CRT expression that probably may affect somehow the expression of the transgene, but also injection-related differences. But more importantly, when we analyzed creatine levels, sorry if I spoiled the results a few seconds in advance, the data closely mirrored the transgene expression results. What is particularly promising is that the FF351 treatment led to a robust rescue of creatine level, as you can see here, here, and here. And this rescue is present across all the three brain regions, but is also comparable to, and in some cases even exceeding, the rescue we previously observed with the JET construct in the cerebral cortex of knockout animals at the same age, which is indicated here by this dashed line. So you can see that the red dots here, here, and here are even higher compared to the average rescue with JET. What we need to determine now is whether FF351 actually also improved the cellular pattern of transgene expression. And to address this, we are planning to analyze creatine levels in specific cell populations isolated from the brains of injected animals. But to move forward on this, we first need to set up and optimize a protocol that we do not know, which also involves purchasing reagents and some expensive instruments. So meanwhile, we started working on what we already know well, assessing the functional improvement at the behavioral level. And we designed an experimental timeline very similar to that one used for the JET strategy to evaluate the efficacy of the FF351 construct in rescuing the key pathological feature of the CTD mouse model. The main difference here is that due to very limited funding for this project, we did not include yet imaging or double-site studies. However, we plan to incorporate additional cognitive tests like the Morris-Water maze and EEG recordings to assess the epileptic phenotype. Please, starting from now, keep in mind that the results I'm about to share are very, very preliminary, and any positive findings will need to be validated with larger sample size. So, starting from the object recognition test, we observed an improvement in cognitive function in treated knockout animals that appears even better, larger than what we have previously observed with the JET approach, largely because the variability in the performance of treated mice here and here is reduced, and also because in addition to the already significant effect at P100, there is also a trend toward an improvement at P40, which we had not observed before. Interestingly, we also observed beneficial effect of the treatment in the Morris-Water maze at P100, which assesses, we didn't do a Morris-Water maze at P40, just P100, because this is not a test that you can easily do in a longitudinal way. The Morris-Water maze assesses a different form of declarative memory, specifically spatial memory, and in this test, animals are placed in the pool and must learn to locate an hidden platform using visual cues placed outside the pool as reference points. And this task includes two phases, a training phase where they learn the platform position, and the probe phase where the platform is removed from the pool and the animals are tested on how well they record the position of the platform. So while, as you can see, the performance in the probe phase did not show a clear improvement in knockout treated mice, there is a trend, at least a trend, toward better performance during the training with treated mice covering shorter distances before locating the platform. And this means that they learn faster where the platform is. Finally, we also observed signs of improved performance in the Y maze in knockout animals treated with FF351. Similar to what we saw in the heart, there appears to be an improvement at P100 with a trend already emerging at P40. However, one more time, these are still preliminary findings and further experiments are needed to confirm these effects. So to conclude, our results demonstrate that gene therapy is a viable and promising therapeutic strategy for CTD with encouraging preliminary outcomes in the animal model. However, based on our data, we believe that the JET strategy is not yet suitable to move forward to clinical trials, and for this reason, we designed and tested a new series of constructs, both in vitro and in vivo. Among these, the FF351 is emerging as a particularly promising candidate, but we still need to complete a full in vitro screening and in vivo efficacy studies to validate its potential in full. So, and with this, I'd like to thank one more time all the members of the team for their work. I'm also truly grateful to our collaborators and funding sources for their invaluable support throughout these projects. And finally, thanks to all of you for your attention and interest today. I will join you live for the Q&A session. So see you soon, and thanks again.
Thanks so much, Laura. That was fabulous. I see you on the panelists already, so I'm sure there'll be a lot of Q&As when we get to that session. I'll invite our next speaker, CHIARA, to bring up her talk. A brief introduction about CHIARA. CHIARA is a master's student in the Guanidinoacetate lab at Queen's University. She's been working on a Dosage study project for the gene therapy treatment of CTD for the past couple of years, and she'll be sharing those results from the Guanidinoacetate lab. I should maybe just briefly call out that our community is pretty lucky. We have not one, not two, but 3 gene therapies, and I think maybe 4 gene therapies that are actually have been attempted for our community, which is really, really great place to be in. Over to you, CHIARA. Looking forward to hearing from your study.
Thank you, and thank you again for allowing me this opportunity to share my results. So we'll just go right into it. We'll start with some background. I know we've heard a lot, and we all know creatine transport deficiency results in the dysfunction of creatine transporters. So this means that creatine is unable to enter cells, and within the brain, this also means that a precursor GA is unable to enter GAMT-expressing cells via this transporter to then be converted into creatine. And we know that creatine supplementation has been tried as a treatment, but it's often ineffective because creatine has trouble crossing the blood-brain barrier. And even if it could, there's no creatine transporters to allow creatine to enter cells. So what has often been used for these kinds of diseases is gene replacement therapy. So briefly, this is the introduction of a functional gene into a body or organs, and it's carried by a vector. So these vectors can be viral vectors, such as adeno-associated virus, which I'll refer to as AAV, or lentivirus, or they can be non-viral vectors. For this project, and we were using AAV, so that's what my focus will be on. So when you're doing gene replacement therapy, the actual therapeutic gene that you're introducing replaces the viral DNA. So if you look at this first image, it's an example of wild type AAV. So what happens is we will get rid of the unimportant stuff, the viral DNA, but we'll keep the ITRs, but we'll replace it with a promoter, the transgene, which in this case was SLC6A8, and poly-A tail or introns. So you can see there's 2 underneath that wild type AAV. There's a self-complementary AAV or a single-stranded AAV. Single-stranded AAVs are useful when you have larger genes as it has a larger packing capacity, but it does take a longer time for it to actually be expressed from the time of administration to peak expression. It's about 2 weeks. Whereas for self-complementary, it's a smaller packaging capacity, but it is able to be expressed sooner after injection. And for this project, we used self-complementary because SLC6A8 is small enough that it could be packaged in there, no problem. So just a little bit of how AAV actually enters cells. So it binds to receptors on the cell membrane, and it's taken into the cell via endosome encapsulation, and it's trafficked through the cell to get to the nucleus. Once it enters the nucleus, the vector uncoats and the viral DNA is released. When it's single-stranded, it then goes through second strand synthesis to become double-stranded. And then it goes through transcription, translation, to actually produce our transgenic protein. So for the case of CTD, we'll have SLC6A8 go through transcription, translation to create our creatine transporter. There are many different delivery methods that you can use for this. When you're wanting to inject straight to the brain, we often use intra-CFF deliveries. So these include intracerebroventricular, intracisterna magna, which are directly to ventricles in the brain, or there's intrathecal lumbar puncture, which is a direct injection into the spinal cord. This is the least invasive option and is often very clinically translatable, which is why we decided to go with this option for this dosage study. So our proposed treatment for CTD is the use of gene replacement therapy using a self-complementary AAV9 vector carrying the human SLC6A gene. We used a small ubiquitous promoter, and it was delivered via an intrathecal injection. Prior to my study, we had Dr. Troy Webster working on some other projects for this entire big scope of a project. So he did neonatal ICV injections and compared them to adult IT injections. And these are some results at 24 weeks. So he saw an increase in weight for both the IT group and the ICV group when compared to the affected mice, as well as a decrease in hyperactivity, which you can see on the left here. He also looked at the response in the brain, the amount of creatine that was restored, and there is an increase in creatine in both the IT and the ICV group. Neonatal ICV is not very clinically translatable, as these are done in day-old pups. So that's why for this dosage study, we went with the adult IT route of administration. So I'll just briefly tell you about the actual project and the study design. So we used a transgenic SLC6A knockout mouse model. This mouse model showed reduced body weight when compared to unaffected mice, decreased creatine levels in most organs, including the brain, muscle, and heart, as well as increased hyperactivity at nighttime since these are nocturnal animals. So what we did, we had 5 different cohorts, one cohort with unaffected mice, one with affected mice, and then 3 had affected mice given a low dose, medium dose, or high dose of treatment. And there was 2 different time points, so we could see the response to this vector at a short term of 13 weeks or a long term of 24 weeks of age. So to start, the mice began on immunosuppressants so that we could reduce their immune response to the vector, which was injected at 6 weeks of age. At 13 weeks, the mice did a grip strength testing and they were weighed, and this was the short-term endpoint. And then for the long-term mice, at 22 weeks of age, they went in a digital cage tracking system. So this was so we could monitor their hyperactivity during the nighttime. And they were in that for 2 weeks. And then at 24 weeks of age, they exited this cage tracking, did another grip strength and weight. And this was their then the long-term endpoint. So I'll move on to some results now. So we saw that the mice, all the treated mice, had an increase in weight when compared to the affected mice. And the 2 higher doses, the medium and high dose, also saw weights that were comparable to that of the unaffected mice. So here's just a graph showing the weights at both 24 and 13 weeks, so we could see how it compared over time, but can also compare it between the short-term and long-term cohorts. So we saw a sustained weight increase with them continuing to gain weight over this 3-month period between these 2 endpoints. Moving on to grip strength testing, we saw very similar results with all treated mice increasing in grip strength. And again, the medium and high dose mice saw grip strength levels that were comparable to that of the unaffected mice. And these increases in grip strength were sustained over the 3-month time period between the short-term and the long-term endpoints. Our hyperactivity data, as I mentioned, was taken at night, so we tracked how far these mice traveled during their night period. So the lower the distance traveled, the less hyperactive the mice were. We can see all of the treated groups again show decreased hyperactivity, with the medium dose being the least hyperactive compared to all the other treatments. which was very exciting to see. We then wanted to look at the creatine levels in the periphery, because as I mentioned, these were low in the periphery and in the brain of these affected mice. So in all of the liver, heart, and muscle, we saw increases at all our doses, with, in some cases, especially in the heart, we saw levels in the medium high dose that were actually higher than that of unaffected mice. So there was complete creatine restoration in the periphery of these mice, which was very exciting. So then I wanted to look at the brain creatine, because many of the symptoms we see in this disease are neurological. So what we saw was there was an increase in creatine levels in the brain of all treated mice. When we compare it to the unaffected mice, it's still about half that, but if we just take that bar out and just look at it compared to the affected mice, we can see this increase was actually about double the amount compared. So there was a significant increase in the amount of creatine, even if it wasn't significant compared to the unaffected mice. So this was at the short-term endpoint. So looking at the long-term endpoint to see if this was sustained, we can see again there was a slight increase, not the same as unaffected mice, but if we just look again at only the affected mice and the treated mice, there was again an increase in creatine to an amount that was significant compared to the affected mice. And this was sustained over the whole 6-month time period, with even slight increases in creatine between the 3 and 6 month endpoints. To take a look at the actual transgene expression, so how much of the SLC68 is being expressed in the brain, we used the delta CQ method. So for this, the lower the number on the graph, the more gene expression. We can see that it was sustained over the 13 and 24 week period. So not only was it there, but it didn't decrease at all, which was very exciting to see and very promising. So what can we conclude from this, and what are our future directions? We can see that there are therapeutic benefits when these mice are injected at 6 weeks of age, with clear increases in weight gain, grip strength, and decrease in hyperactivity. We can also see that there was increased level in creatines in the periphery and the CNS, with complete restoration within the periphery of these mice, and their stable transgene expression over the 6-month time period since the vector was first injected. So where do we go from here? We would like to see a greater increase in creatine delivery to the brain. So we're going to attempt to use techniques such as ICV to inject our vector, in hopes that there will be more widespread distribution throughout the brain, and perhaps we'll use focused ultrasound to increase this delivery. We also will need to do some toxicology studies to determine the toxic dose of this vector and monitor for any side effects. And in the future, we would like to look at the response of affected females to this treatment as well. As we know, this is a somewhat understudied area for CTD patients. With that, I'd like to thank my lab members, our collaborators, and our funding for this project. Thank you.
Thanks so much, Chiara. That was an impressive body of work, and I mean, I anticipate that there'll be a good number of questions at the end of this session. Thanks for bringing us back on track in a timely manner. We got some time savings there that I'm mindful of. All right, well, I'll bring up the next member of the WALIA lab, Robin Winsfield. It's my pleasure to invite you to speak on this next topic. Again, brief introduction to Robin. She's completed her PhD at Queen's University in the WALIA lab, which has been focused on developing gene therapies for actually all three CCDS, which has been phenomenal for us. She is going to be giving her talk on the treatment of GAMT deficiency. So over to you, Robin. You can bring up your slides and we can get started.
All right, can everyone see the slides? We're good to go?
Yes.
Awesome. All right. Well, thank you very much for that introduction and for ACD for having me. Some of you who have attended this conference over the years may have seen some of this body of work before from Jagdeep last year or from me at the virtual conference previously. So I really just wanted to take this opportunity to go through what has truly been the last 5 years of my life and of part of the lab developing this gene therapy for GAMT, creatine deficiency. So first thing I want to walk through is how do we approach making a gene therapy for a rare disease, which is really the big focus of the WALIA lab as a whole. And so we kind of go through this multi-step process where first we design our vectors and then we need to prove that they actually work. So we do that in vitro in cell models as well as in vivo in mouse models. And if we get confirmation that we are having some success there, then we go on to look at different dosages as well as, longer term durability, such as that CHIARA just presented here. And then in the case of this study, we also wanted to look at the immune responses to the vector and take a look at, how that might affect the ultimate efficacy of the gene therapy. And then finally, we move into larger scale toxicology studies with wild type mice. And of course, we always have the end goal in mind of clinical translation. So always keeping in mind our ACD families and keeping in contact with them to figure out how we want to end up designing those trials and how we want to get those therapies to these kiddos. So obviously, we've seen this diagram probably a few different times, a lot of focus on creatine transporter. So just kind of give what the problem is with the GAMT creatine deficiency more overall. And so there's kind of a two-sided issue with GAMT deficiency. And one is, of course, that there isn't effective production of creatine biosynthesis. And so there's low levels of creatine in the cells and in the brain. And the additional issue with GAMT creatine deficiency is that there's actually accumulation of GAA, which is the intermediate between creatine. Apologies, my dogs are barking in the background. And so there's an accumulation of this GAA, which actually has some neurotoxic effects and has been found to be the likely culprit of why GAMT creatine deficient patients have higher levels of seizures and more frequent seizures. And so, as CHIARA mentioned, our lab looks at using AAV delivery to deliver these gene therapies. And so here we're using a jet promoter with the human GAMT transgene package inside, once again, a self-complementary AAV9 vector to get that quicker level of expression. And one thing I want to spend a little bit of time on is how creatine biosynthesis in the brain specifically can really impact the efficacy of gene therapy. So what I mean by this is because we have this kind of broad biodistribution, there's kind of two things that it applies to gene therapy. One is really positive, and that means that GAMT is not expressed in every cell in the brain. It's not expressed in most cells in the brain. In fact, it's a fairly small percentage. And that's really good news for us because it's really difficult to get a gene therapy to every single cell in the brain, pretty much impossible at this point. And so When we hear that, we only need to get it in, 10% of cells or something like that, that's really beneficial for us. And it means it's kind of a good candidate for gene therapy. But the other issue is that because of this kind of varied distribution of the two different enzymes and the transporters, there is the possibility that we get our gene therapy to a cell where it doesn't really have an effect. And by that, I mean that if we get GAMT into a cell that either doesn't express any transporter to get GAA into that cell, or it doesn't express any AGAT to make GAA for GAMT to then turn into creatine, that cell doesn't really have a positive effect. So we kind of rely on transducing cells, hopefully, that we can get GAA, and we rely on cross-correction of creatine then moving to other cells, which is, a benefit we can have in GAMT deficiency compared to the transporter, because the cells do express the transporter to get that creatine. So I'm going to walk you through 3 different projects. Most of the data I'm going to show you, just for the sake of time, is simply going to be creatine and GAA levels in the brain and muscle and periphery. So we can kind of get a snapshot of each project, but there's far more included in these studies. But I want to really give everyone an overview of what this process is like. So this is first a proof of concept where we took the gene therapy in cell models and mouse models. And so what we found in the cell models is that there is actually complete reduction of GAA accumulation in these cells. So that was really exciting to see, since we know that this is one of the big toxic players in GAMT creatine deficiency. And then, of course, we also saw increases in creatine. However, the highest dose was most effective at restoring it. And it wasn't quite to the wild type levels of cells, but this is enough for us to say, let's move in and see what it does in vivo. And so here we're looking at the brain and muscle levels of creatine and GAA. And here we saw really beneficial data in terms of creatine restoration in the brain, especially to the point where we're getting fairly close to heterozygote levels. We use heterozygote mice just as a benchmark because they do have fairly similar levels of creatine and GAA to wild type mice. And what that kind of tells us is that we need 50% or less expression to have a normal phenotype. And so that's kind of our goal and why we use heterozygotes here, as we don't need to achieve 100% expression like we might have in the wild type mice here. And in the muscle, similarly, we saw a similar trend. This study was a little bit underpowered, just given the proof of concept nature, but I'll show some data with some more mice that is quite promising in the muscle as well in terms of creatine. And we saw full reduction of GAA in the periphery, again, a really good sign. So from there, we moved into this much larger scale dosage study. So we looked in the short term at 5 different doses, ranging from 2.7 E10 to 5.4 E11. And all these mice are receiving that intrathecal lumbar puncture at 6 weeks of age. And again, so the short term, we looked at 7 weeks post-injection, or 13 weeks of age, and in the long term, 20 weeks post-injection, or 6 months of age. So this is just the different cohorts here. So again, like I said, in the short term, we're looking at these 5 different doses, and in the long term, we looked at 3 different doses, and we'll get to why we kind of chose to do that as we get into the data. And so in the short term, just to zoom in on the brain and the muscles here, what we saw was pretty impactful. So we saw restoration of creatine in the brain and in the muscle up to the heterozygote levels, with no differences being observed between the heterozygote mice and the 3 highest dose mice. But the other thing that we also saw was kind of this plateau effect at these 3 highest doses. So between the 3 highest doses, we also didn't see any kind of extra effect. There wasn't more creatine, so we're kind of hitting what seemed to be a bit of a ceiling. And this is consistent throughout the entire study in GAA as well. But again, a very stark reduction in GAA from the untreated knockout mice in the pink bar here. And at every dose, even the lowest one possible that we did, what we saw this reduction and this increase in reduction in GAA and increase in creatine. So this was quite promising for us because, you know, the lower doses we can give, the more safe therapy can be. And so even not seeing the differences between those highest 3 doses isn't too much of a problem for us, because it means we can probably dose a little bit lower, around that between kind of 1 and 2 E11 vector genomes in terms of how they translate to mice. And just to take a look over time, we did do serum measurements of creatine and GAA throughout the duration of this study. So we took blood samples from the mice at 5 weeks of age, which was their baseline. So that means that was before they received the drug. They received the drug at 6 weeks of age. So in some cases, as early as 8 weeks of age, we were seeing quite significant improvements in terms of creatine. So you can kind of see they all start at this very low level, except for the heterozygotes. And you can see over time, these, especially the 3 highest dose bars, really do get up to the levels of that black line. And we see the same thing with GAA. And actually, what I like to point out here is that the 2 highest doses, medium and high dose, they actually had higher levels of GAA accumulation at the baseline, which is just normal variability for these mice. But what is really interesting to see about that is that even though they started at higher levels of GAA, they did get down to those normal levels. And so similarly, if we just look at the endpoint serum in the short term, we see creatine and GAA restorations. And so going into the long term, because we saw that plateau, we chose not to move forward with the higher doses in the long term and just kind of stick with that 1.1E11 as our highest dose. And we saw sustainable increases in creatine that were fairly impressive at both the lower medium dose and that medium dose. And these were consistent in the brain and the muscle. So in the muscle, we really did see complete restoration of the phenotype, including GAA accumulation stayed quite reduced. So that is really promising to see. And we also saw consistent reduction in GAA in the brain. However, of course, not down to the heterozygote levels, which seems obvious. But you know, the thing that we don't know necessarily about GAA is how much is too much, and what level of reduction we really need to get. So between these 2 short-term and long-term studies, we did see between 40 and 80% reduction compared to the knockout untreated mice. And again, just to give an idea of what that looks like over time, we can see a pretty nice dose response in terms of the lines with the low, medium, and high dose, kind of moving upwards in creatine as well as downwards in GAA over the entire study period. And something, again, that I'll point out is that there tends to be a decent amount of variability in these studies, and that's just the case. And an interesting thing that we saw is that, you know, these 3 mice, which I may point out that kind of tended to have a lower level of creatine and also a higher level of GAA, these were the same 3 mice, and they possibly just didn't respond as well to the therapy. And may go back to kind of what I talked about in the beginning in terms of brain cells that we are able to get the therapy to are going to affect how efficacious the therapy is in each of these mice. And so finally, to end it off on something slightly different, we also looked at the effect of immunosuppression on the therapy and immune responses against especially the GAMT protein, which is a foreign protein. And so it's pretty standard to use immunosuppression regimens of some kind, most often with corticosteroids in gene therapy clinical trials. And so we try and replicate that as much as possible in our animal studies, which means the previous studies that you just saw, the mice were immunosuppressed with a rapamycin and prednisone regimen. However, in this study, we're going to look at mice with and without immunosuppression. And so just a little bit of background on immune responses against gene therapy and how they can reduce their efficacy. There's kind of 2 big components, one being the capsid that the transgene is packaged in, so that would be, in this case, AD9 capsid, and the other part really being the transgene and kind of the components of that, and ultimately what becomes a foreign protein to the body. So in a lot of GAMT patients, it may be foreign because they don't produce it normally, and in mice, of course, we're using a human transgene, so it's certainly foreign to them as well. So we kind of have a couple problems that can arise in terms of our immune responses to that, and that is often antibodies that are made against these components and pre-existing antibodies, as a majority of humans have been exposed to AAV at some point in their life, so they may have existing antibodies against that capsid that reduce the efficacy. And additionally, there's other like T-cell adaptive responses that can be specific to the transgene or a capsid, and immune responses. So here we're going to mostly be looking at antibody and T-cell responses. So what this looks like is these cohorts. Some of them you may have seen before. So the vehicle cohorts are previously presented in the Dosage study, as well as this the immunosuppressed group that received a 4-month or 16-week immunosuppression regimen. And then we added 2 additional cohorts. So we have one that is not receiving any immunosuppression and one that is receiving a 12-week immunosuppression. So a little bit of a shorter schedule. You know, the less we have to broadly immunosuppress someone, the better. So if it can be just as efficacious with a shorter schedule, then that's ideal. So in the short term, all the mice receive the same level of immunosuppression. So it's either immunosuppressed or not. So you'll see the immunosuppressed mice in the purple here with the non-immunosuppressed mice in the green color. And to zoom in again on the brain, we actually did see an impact of this immunosuppression on creatine and GAA in the short-term animals. So mice that didn't receive immunosuppression didn't have as positive of an outcome in terms of creatine restoration in the brain or muscle and GAA reduction. So there did seem to be a short-term clinical benefit to using immunosuppression. And this was consistent over time as well. Again, looking at the serum, you can see the purple line is achieving a much better creatine restoration over this short period and a much quicker GAA reduction than those that didn't receive immunosuppression. So there does seem to be a short-term benefit to this. One organ that we didn't see this in was the lumbar spinal cord, which is the site of injection. So it may be that there is just more transduction happening here, and immunosuppression was not as effective. Or didn't have an impact here in this location. In the long term, interestingly, we didn't see this remain very much. So we saw actually that no matter what kind of immunosuppression regimen these mice received, they kind of had similar creatine levels, which again were approaching that of the normal heterozygote mice, as well as they had similar levels of GAA reduction, with one exception being kind of an outlier that we found was that the longer immunosuppression actually had less creatine restoration in the brain. So it maybe begs the question of, is a shorter regimen actually better? But this also could just be kind of an artifact of this cohort. So overall, though, in the long term, immunosuppression didn't have much of a difference. And this was consistent over time. And so looking more specifically at why we might see this, we wanted to really look at the immune responses against the GAMT transgene. And so we looked at anti-GAMT antibodies. And while you can kind of see there seems to be a peak around that 12-week point, there wasn't any significant differences at any point in terms of antibodies against GAMT. So this kind of says to us that this is likely a fairly well-tolerated protein in these mice. And the same case is for looking at GAMT positive, GAMT specific T-cell responses. So again, no differences between the immunosuppressed and non-immunosuppressed animals. And this is consistent in the long term. So again, you can kind of see there seems to be a bit of a peak with the non-immunosuppressed animals between 12 and 16 weeks, but again, no big differences here. Maybe a few mice that had higher immune responses than others. And again, no differences in T-cell response. But what I will point out is that in the short term, we do have about a 10-fold greater amount of positive T-cells that are anti-GAMT T-cells compared to in the long term. So getting kind of an overall reduction in the long term, which is something we would expect to see. And in terms of the immune responses against AAV, now this may be kind of what might explain there being a short-term benefit, because we have seen quite significant increase in anti-GAMT or anti-AAV antibodies, in the mice that did not receive immunosuppression. So again, there is likely a benefit to the gene therapy in using immunosuppression here. So just to kind of wrap up, so our AAV9 vector preclinical outcomes so far. So we found that this vector is a promising therapy for GAMT deficiency. It improves creatine and GAA in the brain and periphery of treated mice. It restores expression in the liver. And the human GAMT transgene seems to be minimally immunogenic in these mice. So that's a good sign for safety. And immune suppression does provide a short-term benefit, at least on clinical outcomes, is what we found. So what's kind of happening next? So the big thing that will be going on is a larger scale toxicology study in wild type mice, basically looking at even higher doses than we've tested before to look at any safety outcomes of the vector. We're in the process of drafting and hoping to submit a pre-clinical trial application meeting notes, and then looking, of course, to also start designing the clinical trial and drafting protocols for that translation. Now, we're not done in the lab. We certainly are still looking and exploring new vector designs that might even be more efficacious and doing preclinical testing on those. And of course, a big part of what we have to do is fundraising in order to fundraise and fund this research, especially at the clinical level. Clinical trials are very expensive. And so we are, actively always looking to fundraise in order to support those and this kind of translation. So I just want to end off with a big thank you. Without organizations like ACD, this kind of research wouldn't be possible. And our relationship with this organization has been, I think, really fruitful for all of us, and we're very grateful for that. And just a shout out to the lab and our collaborators at the Hospital for Sick Children in Toronto. Alona Chikova is an incredible mass spectrometrist who is responsible for me having the majority of the data I showed with you today in terms of creatine and GAA. And of course, I just want to say another thank you to the resilient CCDS families for their continued support. Thank you so much.
Thanks so much, Robin. That was great. Looks like you have a number of questions in the Q&A, so I'll invite our moderators for this session, Sarah and Chuck, to turn on their cameras. Quick reminder for everyone, please put your questions in the Q&A box. Sarah and Chuck, if you could do a brief introduction each, and then if you could go round robin on questions so we keep this moving, it would be really great. All right, over to you, Sarah and Chuck.
My camera's disabled. Otherwise, I would turn it on. So if you want to start, Chuck, go ahead.
Okay, thanks, Sarah. And thanks to all the speakers, and thank you, Sangeetha, and Heather, and everyone at ACD for organizing this. So I'm Chuck. I'm an ACD fellow, been an ACD fellow in the past. I really love this community, and it's always great to see new faces again and see old faces again. I'm a researcher at Purdue University. I work with Jonathan Schlebach. Our lab really focuses on sort of membrane protein folding in general, and how different variants can affect that. We study it in the context of a few different systems that are implicated in disease, including the creatine transporter. So I'm the sort of computational biologist in the lab, but we have people who do bench experiments and spend a lot of time thinking really hard about how different genetic variants can impact disease.
Great, thank you. And sorry, mine still says the host disabled my video, but my name's Sarah Brownstein. I am the parent of a 5-year-old girl with CTD.
Sarah, I don't know if you want to start with a sort of patient-driven question, or if I should just go ahead and...
Sure. I actually had a question for Laura around the risks of overexpression with CTD. What do we know about the risk of overexpression? I know you referenced it in some of the data, but I was curious.
Can you hear me well? Because my connection is pretty unstable. Okay, nice. Well, the overexpression, we didn't explore too much this aspect. We have seen that treating the wild type animals with the construct, at least with the jet construct, induced overexpression. Of course, we need to do a full toxicology study, because basically treating the wild type animals with the same construct we use for knockout mice is something that you can refer to as a toxicology study. We have seen this detrimental effect at P40 with the dosage that we used. And actually, we also tested multiple dosages of the jet construct, and we found toxic effects increasing the dosage of the jet construct. And this is why we decided to change the full construct in order to, let's say, not minimize, but try to replicate as much as possible the physiological pattern of SLC6A8 expression and do not see overexpressing cells. But for now, we still need to assess if what we meant to do is actually real or not.
Understood. But I guess in patients, what would you expect clinically would be the implication of overexpression of the transporter?
The analysis of heterozygous and wild type animals is important to understand what happens with gene therapy in patients with some residual function of creatine transporter. We hope that using different regulatory sequences, I cannot really disclose the exact sequences we are using for patenting reasons, but using these different regulatory sequences, we hope that physiologically relevant mechanism can avoid this problem of overexpression, basically.
Interesting. Chuck, you want to go next?
Sure, let's get into some of the questions that we have in the Q&A here. So Jagdeep asks Laura, he says, I think it's hard to say that jet promoter vector didn't work or is insufficient as not multiple doses have been tested. Your thoughts?
Yeah, actually, I didn't show the results, but we did test multiple dosages. And as I mentioned before, with the higher doses, we found toxic effects. And this is why we reduced the dosage of the jet construct. With this reduced dose, in any case, we found some positive effects, but still we were not satisfied. And this is why we decided to move forward, sorry.
Okay.
You want me to do another one from the Q&A, I guess? This one would, I guess, also be for Laura. What are there extra risks in administering gene therapy directly to the brain? Have there been other diseases, presumably where this administration was used, and was it well tolerated?
Yeah, of course, the intraventricular injection is pretty invasive. I know that there is currently there is an ongoing clinical trial for Rett syndrome, relying on intraventricular injection of AAV vectors. I do not know the data from this clinical study. I can understand the concerns of parents and family of doing such kind of treatment to the kids. And this is why we want to be completely sure that the treatment is reasonably effective and completely sure before moving to any clinical trial, because of course it's basically a surgery, it's a brain surgery, so the potential side effects can be risky, of course.
Yeah, I can just add to that, like to complement what Laura said. We're also using direct delivery to the CSF through a little bit less invasive way intrathecally, but I think there's actually a lot of evidence that there is potentially higher risk to delivering some of these therapies intravenously that are targeting the brain, because you have to deliver such high doses in order to get them to the brain that you end up with liver toxicity. And that's actually a really big problem in the field of gene therapy with AAVs. And so because we're delivering it locally, we have the option to give a much lower dose, which I think actually in general for AAVs has a safety feature to it. And I think that's something to keep in mind. And as far as other diseases, Zolgensma SMA gene therapy, which is the first AAV9 approved gene therapy, is being studied to be delivered intrathecally at the moment. And I think it's likely that it'll move more towards that over the intravenous administration.
Yeah, so on that note, can I ask kind of a question from me? We will hopefully obviously we'll get to more of these, but yeah, there's gene therapy is going through a little bit of a rough patch right now. I know that there was a therapy from, I think, Therapeutics that just, had some side effects and that caused some issues. And the FDA canceled a clinical trial from Ultragenyx, I think. And sort of as Robin alluded to, I think that those were due to problems with liver side effects. And, Robin, my question for you is, I thought it was really interesting that you guys sort of did some experiments on looking at the effect of immunosuppression. But in a realistic clinical sense, you're always going to have to do that, because even if, some cohort, maybe would receive a short-term benefit from not receiving the immunosuppression, when you're looking at a population level, even if the population is small because it's a rare disease, probably there's a good chance that someone might have like an acute effect from the gene therapy, just from exposure to AAV9 that someone might not have. So I would think that, gene therapy at a clinical level is always going to be accompanied by immunosuppression. Is that right?
Yeah, absolutely. I think that that's very much true, and you kind of hit the nail on the head with the population aspect. Not using the immunosuppressant. I mean, many mouse studies actually don't use immunosuppression. We do it specifically to look at this more translatability kind of aspect of it. But the kind of rationale to look at that was more to see the kind of immunogenic effects of this therapy when you don't have immunosuppression, because obviously with it, we're maybe not getting the fullest picture. And so that was more the rationale behind doing a non-immunosuppressed group. Then it will be clinically translated. But the rationale behind doing the shorter immunosuppression was more for clinical translation, because we've seen, patients who are have effects from the immunosuppression part of the therapy where they get sick or something and they don't have that capacity to then mount an immune response. And so the shorter period of time we can have them on those kind of broad effect immunosuppressants, the better.
Right. Yeah, I think it's really cool that you guys are really nailing down, in a mouse model, what other drugs are needed to do this. And like we're still in early days with gene therapy, and it's still extremely promising. And all I wanted to sort of say about that is that there's a lot of stuff in the media right now about how gene therapy is maybe dangerous, but highlighting to patients that the scientific community is really trying to nail down those things, and people are really paying attention to these things. I think that's really important to remember that this, science works, and in the long run, I think that there's a lot of promise in this approach. So Sarah, I don't know if you had a question.
Sure. I can give another one from the Q&A for Robin, since we're on her. In your long-term dosage study, did you collect data on levels of GAMT post-gene therapy in liver versus brain of the GAMT protein by IHC or IF? Also, is the construct tested for activity ahead of time? Does it have the same activity as wild-type GAMT?
Yeah, so great question. So on the first side of that, so we did look at GAMT expression post-gene therapy in the liver and the brain. In the liver, we can use a Western blot, and we actually get really beautiful data that shows GAMT expression. So I do have that, just for the sake of time, to present all of this to you. I didn't show it, but we do have durable expression in the long term in the liver. The brain, our expression is low enough that it's hard to detect with Western blot, and we haven't done IHC yet. We have the samples to potentially do that, but we do have vector genome distribution data on that does show that at least we know that there's vector copies in the brain, and so ultimately that combined with what we see in creatine and GAA in the brain is kind of enough for us to go, yeah, we think that there's expression here.
Were there clinical measures that also supported that? I think there was a Q&A question to that effect as well.
Yeah, like behavioral and yeah. So I think one of the big challenges with the GAMT mouse model, unlike the CTD model that has a little bit more of a behavioral phenotype, is that it's quite hard to detect. There's been a few extensive studies that have found very minimal differences between the wild type, heterozygous, and knockout GAMT mice. There's a few that have seen a couple behavioral differences, but it's very limited, which is kind of a limitation for us. And we did our own in-house verification of that. We kind of ran a few studies that hadn't been looked at before, like hyperactivity and some novel object recognition, like Laurel presented, and we didn't see any differences. So unfortunately, that is a limitation to this model. We do have muscular strength data, and that did show a positive trend towards improvement.
Thank you. Chuck, did you want to do the next one?
Yeah, so Troy has a question. Dr. Baroncelli, great data. If I remember your previous results with the JET promoter, you saw a decrease in efficacy for both total creatine and brain connectivity between P40 and P140. In this data with the construct FF351, you do not seem to see a decrease in expression. Do you have any insight into why, such as promoter silencing or greater immune response when expression might cell types?
Well, to be honest, with the JET construct, we found pretty stable increase in creatine levels in treated knockout mice, starting from P20 to P140, P150. I totally agree that with the hypoconnectivity phenotype, we didn't see a rescue at P140 with the JET construct. However, I cannot say anything about the FF351 construct yet, because for now we just measure creatine levels at P40 and we didn't do any imaging testing because of lack of funding for now. And so I do not have a comparison reference yet. I'm sorry.
I can ask one for Chiara. I was curious what your perspective is on what the advantages of using self-complementary vector.
For one, you can have quicker gene expression right away. As I said, the single-stranded takes about 2 weeks for complete gene expression, whereas self-complementary can be as short as like 4 days. So you can get the results sooner. It's also, we don't need the single-stranded for the size of this gene, so it just benefits us in that way, I would say. Sorry, I'm a little sick.
No, feel better. Thank you for still participating.
So I can ask a question also for Chiara. Are there extra risks in administering gene therapy directly to, we sort of talked about some of this.
Yeah.
Why don't we move on to, so Robin mentioned that toxicology studies are on the roadmap. Can you give us a timeline about that?
Yeah, so I mean, there's a little bit of a different approach when you're doing a toxicology study for like clinical translation, because you have to get the vector produced a little bit differently at like a higher grade and things. So there's a bit of a delay in terms of when we are actually going to be able to start this study, but hoping within the next 6 months. And the study itself is kind of a 6-month runtime as well. Essentially, mice are sacrificed at various time points to look at the toxicology over a period of time. So hoping to kind of have those studies done within the next 2 years. And I think I'll answer it too. I think I saw a question about if there was plans for NHP or poison studies like planned within that clinical translation, and that would be definitely something we're like looking towards. NHP toxicology studies are the ideal, but they're also very expensive and difficult to run, and so it will kind of depend on our conversations with regulatory agencies on what they want from us, and whether mice toxicology studies will be enough. Ideally, we would be able to run them in NHPs, but it really depends on access, fundraising, and regulatory requirements.
There's another question here for Dr. Baroncelli. Did you do any in vitro work to ensure that the HA tag is not impacting transporter function?
Yes, actually we did. We measure creatine uptake in immortalized cells, cell line, and we transfected this cell line with the creatine transporter with or without the HA tag, and we found that the creatine uptake is completely unaffected by the HA tag itself. So we can be sure that our construct was working properly. Even if, of course, for the clinical work, we wouldn't use the HA tag because we don't need to detect, we don't need it to detect the transgene expression. This is something that we just exploit for preclinical studies, of course.
I guess for all three of you, I was curious, I think someone referenced funding, and obviously gene therapy development is expensive and time consuming. So I was curious what sources of funding you've all been able to use to support kind of the work that you're doing.
Well, if I may go first, I'm based in Italy, and so in Europe we have some private foundations, such as Teleton Foundation, which is present both in Italy and in France. And there is also another private foundation called Jérôme Lejeune Foundation, where we got this year some fundings. The problem is, at least in Europe, but I guess that in the US, with considering the current situation is pretty the same. The amount of money is currently decreased a lot. For example, for the last project I presented, I don't know if this is meaningful for you, but we got just 40K for getting the project done, which is, of course, well, not enough. We can do a part of it, but not the full project for now, of course. But we are applying to all the possibilities we have.
Yeah, I think I'll just second that. We apply for anything and everything that we might be able to get. We're here in Canada, so CIHR is a big government granting agency that we have been applying to and recently received fund for creatine transporter. So that's very exciting for us. And ACD has also been a big help. We've had 2 fellows. There's a lot of different things that go into funding the research. There's obviously the direct research costs, but also even just funding for graduate students and research assistants that work on these projects is huge. So I know ACD really helps out with that. We've had 2 students go through that, and it's been, so we get what we can get, and I'll second that, fundraising has kind of been decreasing overall. So it's, we try to get anything we can,
Let's get a question from Dr. Britz. Is there still time? Professor Britz. So I think this is an interesting question. So you mentioned that you're giving treatments at 6 weeks, Robin. How does that translate to a human patient? Does this mean that maybe some of these gene therapies could be effective in older children? And Professor Britz also asks, besides GAA and creatine levels, were any behavioral measures done on the mice, such as memory, hyperactivity?
Yeah, great. So I think I touched a little bit on the second part of that question already, that we kind of did look at behavioral measurements, that there's limited differences in the GAMT mouse model, which makes a limitation. But as far as the age, that's a really good question. And I think there's still a lot of, debate and question on the field of like how translatable is mouse age to human age, because mouse age is kind of, they generally have a lifespan around 2 years, and it's more in developmental stages. So we talk about, CHIARA talked about Troy doing neonatal injections, and that's more equivalent to what the fetus in utero would be in terms of developmental stage, which is why it's much less translatable in terms of time of administration. And so 6 weeks we consider an adult mouse. So this would, be the equivalent of someone who is older getting the therapy. And part of the reason we do that is just feasibility, but the other part is that we want to see, could we have an effect if we treat older patients? Because the reality is that, many patients who are going to want this gene therapy are not going to be newborns. Hopefully we have lots identified by now, newborn screening available for GAMT. Very grateful to that. But I think the other issue we have with neurological therapies like this is we don't know the exact dosing window. The overall idea is that the earlier the better, because kind of once you start missing some of those developmental milestones, it can be really difficult to get them back. So what the actual impact on some of those behavioral outcomes may ultimately be, I don't think we can quite say in terms of later treatment after symptom presentation. But we're hopeful that there's still a significant quality of life impact, even if it's not fully a cure at the later stage.
There was another question in here that kind of asked about how you think about this. I think was for all the participants, testing your gene therapy in mice versus in kind of human cell lines.
Yeah, I've seen the question in the Q&A panel. Course would be very important, and again, this is something that we have in mind. Again, this is something that requires a lot of fundings, but of course, using iPS cells, for example, would be very, very useful for testing, or the gene therapy in addition to the mouse models. This is something that I think we should do. Again, my plan is to refine as much as possible the gene therapy strategy in the mouse model we have, and then try to expand the testing to human models and also additional mouse and animal models to understand if this is something really working or not.
So Heidi actually has a really important point of clarification. I think I misspoke earlier based on, perusing some headlines about Ultragenyx. So maybe she could shed some light on that.
I think that was posted by Andrew, and he commented that there has not been a gene therapy that was canceled by Ultragenyx. I think that the gene therapy that did not move forward recently was related to manufacturing and not the application in a human or anything clinical like that.
I think that brings us to the last few seconds of the Q&A. We'll be hearing next from Amy Perry. Amy lives in Ohio with her husband and 2 kids, Ben and Will. Her younger son, Ben, has creatine transporter deficiency. Amy recorded this story to be able to share something about their lives and about a means to raise awareness. Colleen, I can see you bringing up the video, so thank you.
Sometimes when Ben laughs, it fills the whole room. There's a light in his eyes, pure joy, totally unfiltered. I want to start there with that laugh, because it's at the heart of who my son is, even as we navigate this complicated journey called CTD. Hi, my name is Amy, and I'm honored to share our story with you. We live in Powell, Ohio, just outside of Columbus. My husband, Jeff, and I have 2 boys, Will, who's 14, and Ben, who's 12. Ben was diagnosed with creatine transporter deficiency when he was 4, but our journey started much earlier. Ben was born healthy and happy, but as the months passed, we started to worry. He missed milestones, simple things like sitting up, crawling, babbling. Around the same time, he was diagnosed with infantile scoliosis with a 30-degree curve in his spine, 4 full torso casts, 8 months, and then a year in a back brace. At first, we thought, of course he's delayed. He's been in a cast. But even after his spine was stable, his speech, gross and fine motor skills lagged, and the gap kept growing. Therapy started after his first birthday, and by age 2, he was in physical, occupational, and speech therapy every week. He worked so hard, but he still wasn't catching up. We saw specialist after specialist. It wasn't until Ben was 4, after whole exome gene sequencing, that we finally got the CTD diagnosis, and blood and urine tests confirmed it. But getting that diagnosis was bittersweet. I mean, we finally had a name, no cure, no targeted treatment. And I remember sitting in the parking lot after that appointment, feeling both relief and heartbreak. I had to let go of the life I imagined for Ben, myself, and our family. But then we found the ACD community and realized we weren't alone. There were families and researchers out there fighting for answers, and that gave us the strength to keep going. Ben is a social, easygoing, and joyful 12-year-old, but his abilities are very, very different than kids his age. He can't write his name or tie his shoes. He's nonverbal. He has a speech device, some sign language, gestures, and a bunch of different facial expressions. We've learned to listen to all the ways he tells us what he needs. He loves people, especially adults who take the time to connect with him. He's got a collection of stuffed animals he tucks in every night, and he could spend hours lining up his toy cars and trucks. He thrives on routine. And he surprises us. Sometimes out of nowhere, he'll notice something nobody else did, and you realize he's taking it all in. Caring for Ben at 12 is a lot like caring for a toddler. I help him dress, bathe, brush his teeth, comb his hair, change his pull-up, and make sure he eats and stays safe. It's nonstop, and sometimes it's exhausting, but there's so much reward in small wins. But I want to be honest about what day-to-day life can look for Ben and for us. The truth is, Ben's world can be unpredictable and overwhelming for him and our family. When routine changes or when something unexpected happens, like misplacing a favorite toy or even a small transition, it can set him off. He can become dysregulated, and sometimes that means tantrums, hitting, breaking things, even hurting himself. Not badly, but hurting himself. And there are moments when communication breaks down and frustration takes over. And sometimes there's nothing I can do except keep him safe and wait for the storm to pass. It's not easy to talk about, but this is part of our daily reality too. We love Ben fiercely, and we celebrate his progress and his joy. But we also live with the hard parts, the exhaustion, the worry, the heartbreak of watching him struggle and knowing I can't always fix it. Still, we keep showing up for Ben, and he keeps showing up for us. Even on the hardest days, there's always a moment, big or small, where love breaks through, and that's what keeps us going. Ben's medical care involves a whole team at Nationwide Children's Hospital, which we're so thankful is really close by. He sees a lot of specialists right now, but mostly we see cardiology for his long QT, neurology for the possibility of seizures. He doesn't currently have them. Genetics and metabolics for daily care, gastroenterology because with not being potty trained, he has a little issue with constipation, and then behavioral health for all of the behavioral type issues that we deal with regularly. So each of these specialists help us manage a different piece of the puzzle. He also receives multiple therapies to support his development. Occupational and speech therapy weekly and private sessions. During the school year, he gets occupational, speech, and physical therapy through his IEP. But one of the most meaningful therapies for Ben is equine therapy. The movement with the horse helps balance and core strength, and it also calms and centers him in a way that's really hard to describe. But on a horse, you can see his whole body relax, he smiles, connects, and truly enjoys the experience. We've also tried supplements, arginine, glycine, and creatine, the usual suspects for CTD. For years, we weren't sure they were helping, so eventually we stopped them. But then his behavior started escalating, especially at school, episodes that could last up to 2 hours. His medical team recommended reintroducing the supplements, this time adding betaine, and the change was dramatic. He was more emotionally regulated, more present, and less distressed, and this regimen is now a critical part of his care. Research has also given us hope too. Ben was a part of the CTD Natural History Study at the NIH. He also participated in a dysmorphology study for children with CTD, looking for similar features in the body, the face. And most recently, the FNIRS study in Boston for brain creatine monitoring in a less invasive method. And then I'm also part of Parents 2.0, helping define what success should look like for future treatments. And being a part of research makes us feel like we're doing something, not just for Ben, but for the families who will come after us. So it's very important to us. And school is one of the best parts of Ben's life. He started at 3 in special needs preschool and is now in a specialized classroom with some integration. He loves the structure and cannot get enough of riding the bus. He just loves it. And he is very attached to his teachers, aides, therapists, and they're attached to him too. They've gone above and beyond to really help him meet him where he is, constantly adapting, recommending new approaches to help him succeed. And we're truly beyond grateful for their partnership and, more importantly, the way they love and care for our son. Ben's story isn't just about challenges in medicine and school and everything else that I just mentioned. It's also about moments where we show up for a community and the community shows up for us. There were 2 very specific examples that I want to share with you that were just very special moments to our family. In 2018, Ben was chosen as a patient champion for the Columbus Marathon, representing my employer, Nationwide Insurance. He represented mile 12. His face was on banners, ads, and even on the side of a building downtown. And that fall, our son and CTD were front and center in our city. We got to tell our story to people who'd never heard of CTD, and we felt seen. It was an amazing event. And then just last September, Ben won a drawing for a Grand Marshal Prize at our school's annual fun walk, where he got to ride in a golf cart with the principal and everything, and Tyler the Tiger, who was our mascot. They did the drawing at a class assembly, and when his name was called as the winner, his classmates started chanting, and the whole school started chanting his name. It was loud and wild and beautiful. And just for a moment, Ben wasn't just the kid with special needs or with CTD. He was just Ben and loved by his community. It was truly special. I wish I could say every day is easy, but it isn't. I see my friends' kids growing up, gaining independence, and we're still in full-time caregiving mode. We don't have family nearby, and finding reliable help is hard. Sometimes I just want to feel like a normal mom, but being in public can be tough. Ben needs my full attention, and even a quick conversation can trigger distress. In other words, he just wants me to focus on him instead of others. So it's cute sometimes and very annoying in others. But self-care has become my lifeline, but I'm human. Some days I don't have it in me, and some days I fail, but what really keeps me going is love in the moments big and small. When Ben reminds me that joy is always within reach, and even when life looks nothing like I had imagined many, many years ago, he has this way of grounding me, of showing me what truly matters. And in those moments when I hear him laugh or see him completely lit up in joy, I remember that this is what life is about, just being fully present and finding happiness in the simplest things, because Ben is so good at just finding joy in the simplest things in the moment. So I don't know what the future holds. We've learned to take it one day at a time, but I do know that we'll keep showing up for Ben, for CCDS families, and for progress. To the researchers and doctors in the room, thank you. Your work matters more than you may ever fully realize. Because of you, we have hope. Because of you, we can imagine a future that looks brighter for kids like Ben. Thank you.
Thank you, Amy, for sharing your story. I think reading about it was just so amazing. The video of Ben receiving his award, as well as what you shared about his smile. And you're right, it's one step at a time, every day, incremental, and showing up matters. So thank you for showing up. Thank you for your support. And we hope to show up as well. We hope to be there and continue this journey together. Okay, we'll switch into our next session. Next up, we have a poster session with 2 speakers. These are pre-recorded sessions. In the past, we've not done a poster session for our virtual conferences, so this format is a little bit new. So bear with us while we try to beta test this today and tomorrow. We will have a Q&A, like Q&A after, where we'll invite the speakers for a question and answer session. Okay, so first up we have Emmanuel's talk. Dr. Bartolini is a clinical neurologist and researcher with special interest in epileptology. He has worked for a while at the United Kingdom, as well as in Pisa and Florence in Italy. And he is experienced in emergency neurology, as well as the management of chronic neurological disorders in children and adults. He recently published a paper, and his talk today focuses on the same topic. It is on the dynamic electro-clinical features of GAMT deficiency familial case series. All right, Colleen, we can go ahead and drive.
Hello, and thanks for the invitation. I'm Dr. Emanuele Bartolini. I'm the head of the Epilepsy and Clinical Neurophysiology Lab at the REINHARDT Foundation Stella Maris, Pisa, Italy. Our institute is devoted to the care and research of children with rare neurological disorders. They especially take care of children with epilepsy. I will talk about the dynamic electro-clinical features in Guanidinoacetate N-methyltransferase deficiency. Well, this is a form of cerebral creatine deficiency disorder, and especially it's in the intermediate part of the metabolic chain. And like the other cerebral creatine deficiency disorders, children may suffer developmental delay, cognitive impairment, and a speech-language disorder, and some kind of movement disorders that may include hypotonia, myopathy, and a movement disorder as well, proper movement disorders, behavior abnormalities, and of course, seizures and epilepsy. Epilepsy is a common manifestation of GAMT deficiency, with very good features at different ages. In the vast majority of reported papers, children initially suffer febrile seizures. During follow-up, generalized seizures have been largely reported in about 68% of cases. Instead, focal motor seizures have been very rarely reported and described. Generalized seizures are mostly convulsive, which is tonic-clonic, but myoclonic seizures, tonic seizures, and atonic seizures have also been reported. There is no electroencephalographic hallmark of GAMT deficiency. There are records, but these are really anecdotal, of Lennox-Gastaut syndrome. It is an epileptic encephalopathy with typical EEG features. Ideal for the patients with GAMT deficiency are resistant to anti-seizure medications. However, creatine, ornithine, and sodium benzoate supplementation, as well as arginine-restricted diet, can improve both the development of children and epilepsy. Further reduction of GAA levels, mostly. So, to catch an early diagnosis is crucial because it can modify the natural history of the disorder and can avoid the disease progression or at least limit the disease progression. In our experience, we aim to describe a familiar case series of three siblings that are currently aged 7, 5, and 3-year-old, born to consanguineous parents originating from Pakistan, with a disease onset with global developmental delay, absent expressive language, ataxia, and microcephaly. The diagnosis was by serendipity because patient 2 exhibited low blood creatinine levels during an intercurrent infection at 2 years of age. Such findings in a child with developmental delay prompted to perform plasmatic and urinary creatine dosages, which were found to be abnormally low. Thereafter, next-generation sequencing of the genes involved in cerebral creatine deficiency disorder revealed a homozygous truncative variant in the GAMT gene. This was classified as likely pathogenic because the truncating effect on the protein yielded a loss of function of the protein itself. The siblings came to our attention at the age of 5 years, 31 months, and 16 months, and we immediately introduced supplementation of creatine, ornithine, and benzoic sodium. Urgent restriction was not necessary because the family followed a low-meat diet. The first patient, the older one, is patient 1 that suffered. He was male. He is male, actually. He was male at the epilepsy onset at 18 months of age with prolonged febrile seizure with left head turning. Later on, at 3 years of age, he suffered a similar seizure, but this time without fever, still with focal fissures, left head turning. Levetiracetam was introduced. 2 years later, when he came to our attention, we captured multiple atypical absent seizures by long-term video EEG monitoring. There was something unclear, and we wanted to look better into it, so we performed long-term EEG monitoring. After this diagnosis, we introduced ethosuximide on top of levetiracetam and supplementation as well, of course. After 2 years, he's still seizure-free, so we obtained complete seizure freedom. His EEG was peculiar. At the time of diagnosis, he exhibited long ramps of slow spike weight discharges that were diffuse. And this corresponded to waxing and waning state of consciousness that are indeed atypical absent seizures. On therapy, he stopped exhibiting seizures, but some abnormalities that still persisted, the initial abnormalities, albeit ameliorating. But focal onset left temporal spikes started later on in spite of seizure freedom. Patient 2, she is a girl. At 31 months of age, she had no over seizure, but in light of the experience we had with patient 1, we decided to perform a long-term monitoring. And we also captured in her multiple atypical absent seizures. We decided to introduce valproate, and we managed to obtain a seizure freedom that is still enduring. But in this case, similarly, at the beginning, the EEG was still showing very long rounds of slow spike weight discharges with this fluctuating state of consciousness. And during follow-up, the EEG improved in terms of these discharges. Atypical absence disappeared, but focal left temporal discharges started with no seizures yet. Patient 3 is the youngest of the family, and we managed to introduce supplementation at an early stage of the disease, 60 months of age. It never exhibited any seizure, and the EEG was normal, actually, it was a bit slow, but she didn't exhibit any type of epileptiform discharges. So, in our experience, as in most formally reported cases, you obtain seizure freedom with supplementation diets and anti-seizure medication. However, this is quite unusual, and this struck us very much. Clear-cut focal EEG abnormalities arose and persisted during follow-up, despite clinical improvement and neuroimaging normalization, prompting us to maintain a stable drug regimen. In fact, at disease onset, the MRI was normal, but the spectroscopy detected no levels of cerebral creatine. Levels of cerebral creatine were restored after supplementation. However, the EEG still showed these focal abnormalities. Former works have described seizure reduction and possibly freedom, even when the EEG was still showing generalized discharges. Other papers reported a complete reversal of EEG abnormality, so complete disappearance after supplementation, allowing to discontinue anti-seizure medication. However, our experience suggests, first, to carefully monitor short and insidious atypical absence seizures in GAMT deficiency. This type of events cannot be easily identified by caregivers of children with intellectual disability and might be underestimated. Thus, video EEG monitoring is recommended in these children. And of course, follow-up studies after the downtaping of medication are still lacking, so we don't know what the epilepsy course will be a long term. So we recommend to be cautious in suspending, interrupting anti-seizure medication. It can be done, but with caution and follow-up, especially neurophysiological follow-up. Well, thanks to the Association for Creatine Deficiencies, as well thanks to the patients, of course, and their families, and Dr. Zini, the neuropsychiatry specialist of the children. Thank you very much indeed. It was a pleasure.
Thank you to Dr. Bartolini for the recording. He is not able to be here today, but if there's any questions directed towards him, please do post them in the Q&A, and we'll capture those and share it with him. And at this time, we do have our other poster speaker here today, Maria Porel-Bechot. Maria is a resident in neurology with a special interest in rare diseases as well as movement disorders. She collaborated with the epilepsy unit at Hospital de Saint Paul and published a research article on refractory epilepsy in a patient with CTD. We'll go through her talk today and her poster presentation today, and she will be there for the Q&A session live. All right, Colleen.
Hi, my name is Maria Borrell. I come from Barcelona, from Hospital de la Santa Creu i Sant Pau, and together with the epilepsy team and Dr. Carmen Fons from Sant Joan de Déu, we reported this case of an excellent response to CBD and CLB of a patient with drug-resistant epilepsy with creatine transporter deficiency. Let's get started with the case. This is a 29-year-old male without any relevant family history that we received for consultation because of poor seizure control since childhood. Let's see a bit of his timeline. He had no complications during pregnancy or delivery, but however, before 1 year of age, he started showing some developmental delay and mainly language problems. And already at 2.5 years old, he started having seizures. They were tonic seizures and generalized tonic-clonic seizures at a high frequency, at least once a week. He started follow-up in San Juan de Déu Hospital, and at 3 years old, he was diagnosed with autism spectrum disorder. Soon, he had 2 episodes of status epilepticus that were quite severe. The phenomenology was prominently motor, but he needed barbiturate coma. After some years of stabilization, at 15 years old, he stopped having generalized tonic-clonic seizures, and instead new seizures appeared. They were focal motor with a dystonic position of the left hand and impaired awareness, and they lasted between half a minute and 1 minute. After many years, he arrived to our clinic at 27 years old. He was having at that time between 10 to 20 seizures every month, and he was dependent for eating because he couldn't use cutlery, and he needed a wheelchair to use outside the home. So what were the imaging that was made to this patient? He had 1 brain MRI from 2011 that showed some non-specific hypomyelination, and the spectroscopy, which is the most important, showed decreased creatine levels in the thalamic. He had a genetic study that at that time he was 9 years old, so it had to be sent to the Netherlands, that showed a pathogenic X-linked mutation in the creatine transporter gene in SLC6A8. We diagnosed him of creatine transporter deficiency this way, And regarding EEG, we don't have really any previous EEG image available. There are some reports that show different things, but there are some slow and spike wave reports and a diffuse slowing at 5 Hz. So what do we know about creatine disorders and epilepsy? I will not introduce to you all the things that you already know about the main features of every creatine disorder subtype, but epilepsy is prominent in GAMT deficiency, appearing in more than 70% of patients, usually before 2 years of age. And it is also important in creatine transporter deficiency, appearing in almost 60% of patients at an average age of 4 years. The seizure types are very heterogeneous, not to say that almost half of them, as you can see, are not specified. So you can see a bit of predominance of febrile seizures in both deficiencies and also generalized tonic-clonic seizures, but they are very unspecific. The EEGs are also very non-specific and heterogeneous. The suggested mechanisms in the literature for epileptogenicity in these disorders are several, but the main ones are neurotoxicity of guanidinoacetate because of oxidative stress and because it is a GABA-A partial agonist. That means it doesn't have the whole effect of GABA-A, but it has some, and it also leads to receptor desensitization. And also the deficient creatine phosphocreatine energy shuttle, and the fact that creatine is also a neuromodulator, acting also as a GABA-A agonist. Regarding drug-resistant epilepsy, the treatment recommendations are not very specific because we don't have many patients and proper studies haven't been conducted. So the main recommendations nowadays are the standardized treatment with anti-seizure medications by an experienced neurologist. And yeah, many anti-seizure medications may be effective, but none has demonstrated specifically effective for this disorder. Education of parents and caregivers is very important. So around 200 cases of creatine transporter deficiency have been reported. And in a deep literature research, you can see that less than 10 cases have been reported drug resistant. With my team, we made a table where we summarized most of these cases. You can see the age and sex of the patient, the prior treatment, and the treatment of the intervention that was reported in that specific article. Most of the treatments, as you can see, were supplements with creatine, arginine, or glycine. And in most of the cases, the specific anti-seizure medications are not mentioned. The outcomes were quite variable, but most of them showed no improvement or partial improvement, also explained in a non-specific way, like reduction in frequency, mild improvement, and only one patient went seizure-free with creatine, arginine, and glycine supplementation. So it's quite heterogeneous. What did we do to our patient? So in the first medical visit, he came with levetiracetam, lamotrigine, phenytoin, and clonazepam, and he had tried all this prior anti-seizure medication, and also he had tried supplements with creatine, glycine, and arginine for a long time without any difference, so he wasn't taking them anymore. After, he was having 10 to 20 seizures a month, as we mentioned. After the first visit, we changed brivaracetam for levetiracetam, and we started a ketogenic diet that he followed for at least 3 months and was not effective. There were no changes. That's why we decided to remove clonazepam and start CLB and CBD. That's clobazam and cannabidiol. After 1 month of starting these medications, he started being completely seizure-free. And in September, it's going to be exactly 2 years from then. He has been seizure-free since then. He also has been shown some behavioral improvement as reported by his caregivers. He has a bit better attention, and he is collaborating a bit more in group activities, and he didn't show any adverse effects or lab test alterations. This is his EEG that we managed to make 3 months after treatment, which is completely normal, and you cannot see any epileptiform activity. So what is CBD? Let's quickly review its mechanisms of action. CBD is a phytocannabinoid that is non-psychoactive And it has many uses outside of epilepsy because it also is anti-inflammatory, analgesic, and anxiolytic. And mechanisms of action are multiple, and many of them are unknown. Regarding epilepsy, the specific anti-seizure medications of this drug, in the literature, there are mostly three. The extracellular adenosine levels are elevated, which modulates neuronal excitability and inflammation. Also, the reduction of intracellular calcium release because of GPR55 antagonism, and the desensitization of TRPV1, which reduces extracellular calcium influx. Why was this patient so responsive to CBD? Does creatine deficiency disorders have anything to do with the CBD mechanism of action, anything special? Well, we researched a bit on the literature. We could see that CBD has some effects in mitochondrial function and energy systems because it is an antioxidant agent, and it also has some GABAergic properties that could compensate and higher a bit the seizure threshold, but nothing that we could see that was really specific for creatine function. The most common side effects of CBD are somnolence, that is the most common. Usually it's associated with clobazam and brivaracetam. Hypertransaminasemia, usually associated with valproic acid, and this is generally transient and dose dependent. Diarrhea, that is the most difficult adverse effect to manage, and also encephalopathy that can be also associated with valproic acid. Our recommendation is to make a slow titration and to monitor interactions with other drugs. So in Spain, and I believe in most of the European Union countries, the current indications for CBD in epilepsy are three scenarios. You need a patient that is drug resistant and is more than 2 years old and has Lennox-Gastaut, Dravet syndrome, or tuberous sclerosis complex. For Lennox-Gastaut and Dravet, it has to be associated with clobazam. This is a bit of a problem because the criteria for Lennox-Gastaut are quite strict. For example, in the case of our patient, I put here in green the criteria that he did fulfill. Here you can see the tonic seizures as mandatory. Well, this is a bit strange because we know that he had tonic seizures when he was 3 years old, that is more than 25 years ago, because his mother remembers and his mother could tell us, but especially in the adult clinic, the retrospective history is very difficult. Also, there's a problem with EEG. I already told you that we don't have previous EEGs available, and in one report we could read that there was a slow spike wave. It's a requirement. But well, if we could not have had this information, we could not have checked all the boxes, and probably this patient couldn't have benefited from this treatment and he would not be seizure-free. So we hope that the criteria for CBD indication expand a little bit within the years. I want you to take home these messages. We reported an excellent response to CLD and CBD in this patient with creatine transporter deficiency and drug-resistant epilepsy. He has now been seizure-free for 2 years. Regarding epilepsy and creatine deficiency disorders, 70% of patients with GAMT and 60% of patients with creatine transporter deficiency have epilepsy, but there is a non-specific anti-seizure treatment that has been established for drug-resistant cases. This is due to the small sample and also because information from case series is heterogeneous and usually incomplete. The current indications of CBD are Lennox-Gastaut syndrome, Dravet syndrome, and tuberous sclerosis complex that have refractory epilepsy. Its mechanism of action is multimodal and it is not completely known. Antioxidant and GABAergic properties might benefit patients with creatine deficiencies. And a slow titration is recommended to avoid adverse effects and to monitor interactions with valproic acid, clobazam, and brivaracetam. Lastly, about future directions, we do encourage the creation of a collaborative database for neurologists that have patients with creatine deficiencies and drug-resistant epilepsy. That this way we can track the medications and hopefully identify those that might be especially beneficial. We would like to acknowledge the family members of the patient for all their cooperation and support throughout the course of this case study. We also thank the Spanish Creatine Deficiency Association for their continuous efforts to improve the quality of life of creatine deficiency patients and their caregivers. And we are deeply grateful to the Association for Creatine Deficiencies for their unwavering advocacy for patients and for connecting professionals all across the globe. Thank you so much for your attention. These are my contact details in case you have any question, any option of collaboration, and I'm now open to your questions.
Thanks, Maria. May I ask you to please switch on your video, and if you could go off mute, we can get our Q&A started. That was a great talk.
Thank you.
So I think we do have a couple questions, so I can go ahead and get started. So one of the questions is about CTD patients' liver. So I think CTD patients are typically on a lot of different types of drugs, and some of these do have toxicities for the liver. Could you speak a little bit about this particular combination, or if liver enzyme levels were measured during treatment? If there's anything you can share, that'd be helpful.
Yeah, specifically for this patient, he didn't have any liver enzyme elevation. That's the only patient with CTD that we have in our clinic, so I cannot speak for others, but he didn't have any problems with that.
Okay, that's great. I think this, so let me know if this question applies to you. When was creatine introduced, and what effects of discontinuing creatine seen on symptoms other than seizures?
So when was creatine introduced, and what else? Sorry?
Your patient was on creatine throughout?
No, he was on creatine many years
creatine.
ago, not now.
Okay, so there was no creatine really present in this. I
No.
think the other question is just more on maybe wide availability of CBD, right? So CBD, particularly maybe in other countries, available in many different forms. Can you speak to whether in your country and maybe elsewhere, like does CBD always need to be prescribed by a doctor? Or would you have an opinion on CBD that's available, other mechanisms? What is your opinion?
Well, I think it's getting more and more trendy. As I know, CBD in Spain can be only prescribed by a doctor, at least the pill. I know there are some like creams and stuff that people use, but I don't know if you can buy that in the supermarket. But the medical one, it has to be prescribed by a doctor, and especially in epilepsy, the indications are very narrow, and we are being supervised so that we cannot do it freely.
Of course. I guess I'm curious about, having tried this protocol in a CTD patient, and I understand that there's restrictions for exactly how this protocol can be prescribed, right? Because it sounds like they don't meet the criteria necessarily for widely utilizing this. But what is the mechanism of testing something like this in other seizure disorders? Would you have you seen any GAMT patients? Would you approach treating them in a similar manner? What are your thoughts?
I think so. We really researched if there was any connection between CBD mechanism and creatine deficiencies. We didn't see anything specific, but with the amazing response of this patient that he had tried so many other medications without any benefit, I think I encourage myself and other doctors to give it a try. As soon as the patient is refractory, I would give it a try, because who knows, maybe we can reproduce this pattern and we discover something new.
Yeah, no, thank you. That's great. I think there are a couple questions that are here now are for Emmanuel, and I will capture them and maybe share them with him. While we're waiting, maybe, and because we have a little time left, maybe I'll ask folks, maybe Dr. Longo or Dr. Stockler, if you'd like to chime in on this area, since this aligns with your area of expertise. If you would raise your hand, then we can make you a panelist. And while we wait for that, I don't know, maybe Heidi, do you have a perspective on CBD availability in the US and whether that is utilized for treatments?
I have read the same requirements of LGS and the other disorders for CBD as well in the US. So I think it's consistent. But I will say that my daughter takes CBD. She takes Epidiolex, and I think it's because we checked all the boxes for LGS as well.
In the United States, every state is slightly different depending on who might prescribe it. In Utah, it must be a neurologist. But in different states, it is different. In California, you can buy that anywhere. But
yeah. And are there, to both of you, are there like dosing specifications associated with CBD? I imagine there must be for the treatment of epilepsy, right?
there are, but again, they are usually managed by neurologists, not by a geneticist. And in fact, I assume that Dr. Pichot is a neurologist.
Yes.
Yeah. It looks like we have a question here. Could the many seizures in the month be contributed by many ASM or any specific ones on board? I heard that Keppra may introduce more seizures, but not sure if research supports it. And let me maybe find out what Keppra is for. Keppra is levetiracetam. Thank you.
Well, I haven't heard of this theory actually, but I can say that this patient had been having a lot of medications, switching, trying. The list is very long. And I'm sure there was periods where he was on Keppra and periods off Keppra. So at first I would say not, but maybe I should review more about it because I had never heard about it.
Okay. Well, thank you so much, Maria, for your talk and for being a part of this symposium. I don't think there's any additional questions at this time, so we will go forward to the next session. This next session will be our last session for today, and we're moving on. We heard earlier today from the molecular insights of CTD as well as GAMT, the patient registry. Then we heard from gene therapy advances in preclinical models, clinical cases of CTD and GAMT. And now we're moving on to a biomarker focused session. So measuring what matters, different biomarkers that are available or being worked on for CCDS. And so while we bring up Dr. Mabonzo for his talk in this session, I'll just provide a brief introduction to Dr. Mabonzo. Dr. Mabonzo joins us from Paris-Saclay University in France. He's the research director of the neurovascular unit of Research and Therapeutic Innovation Labs at CEA-Saclay in France. He has been working on CTD for a number of years. We've also had the pleasure of supporting several members of his lab through our fellowship program. He's also the co-founder of Ceres Brain Therapeutics that is working on therapies associated with CTD. His talk today is titled Toward Identification of Creatine Transporter Deficiency, Circulating Biomarkers, and Brain Dysfunction. I believe Dr. Mabonzo was actually not able to be here by himself, but he has someone else speaking in his stead.
So hello everyone, so I'm a researcher in Dr. Mabonzo lab at CEA-Saclay in France. And so I'm very happy to talk on his behalf today and to talk about what we are doing in the lab, especially about how we try to understand the pathological mechanism of creatine transporter deficiency and how we try to identify biomarkers to be used in preclinical studies, but also during clinical trials. So as you well know, CTD is a genetic disorder that disrupts the normal transport of creatine into the brain. and into the cerebral cells. So the non-functional transporter is present at the blood-brain barrier level, preventing creatine circulating in the bloodstream to enter into the brain parenchyma, but it's also present at the membrane of all the cerebral cells. So even the creatine synthesized within the brain is unable to reach the cytosol of those cells. Creatine normally play a role in cellular energy buffering. So in mitochondria, creatine is phosphorylated by creatine kinase to form phosphocreatine. Phosphocreatines and shuttle high energy phosphate groups to generate ATP in the cytosol via cytosolic creatine kinase, and in CTD, the lack of intracellular creatine impairs this phosphocreatine-creatine shuttle system, leading to chronic energy deficits in all the cerebral cells. However, the role of creatine in the cells, it's not only linked to energy. Emerging evidence suggests that the creatine may also stabilize mitochondria membranes, reduce the production of reactive oxygen species, so it acts as an antioxidant, acts as a neuroprotective agent, and also as an anti-apoptotic agent under stress conditions. We also demonstrate in the labs that as synthesis of creatine within the brain use methyl donors, creatine deficiency has some epigenetic consequences, meaning that nucleic acids, typically DNA, are modified, and that can lead to modulation of many gene expression. Altogether, creatine within the brain is very important through those mechanisms, but it's also implicated in maintaining ionic gradient, synaptic transmission and plasticity, neurotransmitter recycling, neuronal excitability, etc. All those alter mechanisms at the cellular and molecular levels in CTD are thought to underline many of the neurological symptoms observed in patients, including neurodevelopmental delay, intellectual disabilities, and seizure. In addition, some morphological changes in CTD brain have been described, such as delay myelination of neuron axons that can explain the white matter atrophy and thinner corpus callosum. In the lab, we have been working on understanding all those pathological mechanisms happening in the CTD brain, and we try to link those cellular and molecular signatures to clinical outcome. We also intend to use those signature or biomarkers to monitor therapeutic efficacy. So the question is, do these alterations in the CTD brain are restored by a treatment such as the CBT-101 treatment? That is the treatment that is initially developed in the Mabonzo lab and now carried to clinic by Ceres Brain Therapeutics. To do so, we have used preclinical models, including CTD mice models, and compare with or without CBT-101 intranasal treatment for a month. The brain of those mice after 1 month of intranasal treatment were samples separated in the different regions, and we choose to apply a global approach by proteomic to identify biomarkers related to cognitive improvement after the treatments. This analysis generated a database of more than 4,000 proteins in the brain samples, and comparison of the abundance of those proteins across the different brain regions and with versus without treatments identified 14 biomarkers. These biomarkers, including KIF1A and PLCB1, are modulated by the CBT-101 treatments and also correlate with cognitive tasks. They were described in the literature as related to ID disorders, including autism spectrum disorders, and are at the cellular level implicated in neuronal migration and in synaptogenesis. While the mice models appear to recapitulate the phenotype of human with CTD, there is still an important translational gap between rodents and humans. So to address this need, we generated human brain organoids from induced pluripotent stem cells of 3 male CTD patients. Brain organoids are 3D tissue models of the human brain that are derived from iPSCs and culture in vitro. They have enabled studies that were previously limited by the inaccessibility of the human brain tissue, and also allow to fill this gap, this translational gap between rodent and human. So we demonstrate that the brain organoids from CTD donors had reduced creatine uptake, and we deeply characterize them. We also use the same approach as previously in mice, using global proteomic analysis, comparing CTD organoids to healthy organoids. And we select 510 protein for which abundance was altered regardless of the patient mutation. Among those, we selected 32 proteins that are listed here, and those are the most significant. And among those, 21 proteins have been already related in the literature to autism spectrum disorders, ID, neurodevelopmental disorders, and epileptic encephalopathies. We also showed some interaction between those proteins. Putting together those alterations described by the proteomic analysis plus validation using more conventional techniques such as Western blotting, will describe alteration of molecular pathways that are related to clinical outcome in CTD patients. As example, we demonstrate alteration of the expression of proteins that are associated with microtubules that play a role in the structure of the axons, so in the synaptic transmission and neurogenesis. We also used the CTD organoid to look if the CBT-101 treatment ameliorates the alteration of such biomarker. So as example, we have demonstrated that the expression, and more precisely the phosphorylation of another protein associated with microtubule, which is the Tau protein, was altered in CTD organoids. So you have here an increase of the ratio of phosphatase to Tau. And so compare to the control here in orange. And so the CBT-101 or DCE treatment seems to restore the normal phosphorylation state. While promising, this biomarker identified in the mice brain or in the brain organoids remain to be assayed in brain tissue. And so the need has now moved to demonstrate their utility in clinical practice, meaning in biological fluids, such as cerebrospinal fluid, or even more easily accessible blood. So to answer these questions, and thanks to the biocollection at the Hospice Civil de Lyon here in France, we were able to conduct a study on 12 CTD patient plasma. So for those patients, the diagnosis of CTD was confirmed by identification of the variants. They all exhibit developmental delay and intellectual disabilities. We have access to their clinical description, including age, if they have epilepsy or not, their IQ, etc. And at this stage, we have 3 male healthy controls and 12 male CTD patients. With those samples, we conduct the proteomic analysis, followed by the bioinformatic analysis as previously. The proteomic database was generated, and the data were filtered and normalized to select the protein with differential abundance between CTD patients and healthy controls. The quality of the data were checked to evaluate if we were able to nicely cluster the two groups and to look for reproducibility and other parameters. The quality evaluation at this stage makes us exclude 3 CTD patients, so we had 9 patients for the next step. From here, a total of 68 proteins were identified as significantly altered in CTD patient with a p-value threshold below 0.05 and 27 proteins with a p-value threshold below 0.01. To investigate the biological significance of this differentially expressed protein and their potential link to the clinical characteristic of CTD, we conduct a disease-related pathway analysis using ENRICH, followed by a frequency analysis. This reveals that 9 of the differentially expressed proteins were associated with neurological disorders, including neurodegenerative disease. You can see here it's mapped with the 3 healthy volunteers on the left and the 9 CTD patients on the right, so with the dominance of red for healthy volunteer and the dominance of blue for CTD patient, meaning that all those 9 proteins were downregulated in the CTD patients compared to controls. When we try to see if there is a link or interaction between those 9 proteins, nothing was found between the 9 proteins, but when we use the 68 protein here with the significance below 0.05, we were able to construct what we call a string graph that depicts the predicted protein-protein interaction. This mapping reveals indirect interaction between 3 of the 9 proteins, which were alpha-synuclein, CA2, and PRDX6. So I will start with CA2, which is the carbonic anhydrase 2. It is a metalloenzyme that facilitates the reversible conversion of carbon dioxide into bicarbonate, and that is expressed in astrocytes and neurons. This enzyme is useful to maintain pH homeostasis, metabolic coupling, and play a role in synaptic plasticity and neuronal coupling also. And so the expression of the CA2 is decreased in plasma of CTD patients compared to controls. To go deeper, we came back to the 2 other proteomic database generated with previous studies, the one in CTD brain organoids and the one in CTD mice model. We demonstrate a consistent decrease of CA2 in both models, and also a restoration of the CA2 level after CBT-101 treatment in the mice model, significant in 3 regions, the cortex, hippocampus, and cerebellum. The second protein we focus on is PRDX6. In fact, on the interaction mapping, we had the PRDX6, and among the 9 proteins here in the heat map, we had both PRDX6 and PRDX2. So the PRDX family are a group of enzymes that mainly serve as antioxidants. They are playing a key role in neutralizing peroxides and participate in the regulation of multiple signaling pathways. In the context of CTD, downregulation of PRDX suggests aggravation of the mitochondrial oxidative stress and inflammation. Same as with CA2, we demonstrate a consistent decrease of PRDX6 in the plasma of CTD patients, in the CTD organoids, and in the mice model. And the CBT-101 treatment seems also to have a positive effect on the level of this protein. Finally, the last protein we focused on, it is the alpha-synuclein. So it is a protein that regulates vesicle trafficking, so important for neurotransmitter release. It's also important for maintenance of synapse, and it's stabilized the electron transport chain in the mitochondria, so important for energy metabolism. The alpha-synuclein is well known to be related to Parkinson's disease because if it's upregulated, if it aggregates, or if there is a misfolding of the protein, it yields neuronal toxicity due to several mechanisms, including inflammation, oxidative stress, mitochondrial dysfunction, and lysosomal dysfunction. In the plasma from CTD patients, we have a significant decrease of alpha-synuclein. In the analysis in the other two models, we are upstream here, and we were expecting to see an accumulation of alpha-synuclein in cerebral tissue. Consistently, we confirmed significant increase of alpha-synuclein in brain organoids and in the cerebellum of CTD mice. So the CBT-101 treatment also seems to improve a little this deregulation. The next step with those data will be to increase the control group size to at least 12 healthy volunteers, also to increase the CTD patient group to 24 male, and with all those samples to validate biomarkers by ELISA. We also intended to correlate this data with clinical data, for example, age or IQ or other data we can have. So in summary, we have identified potential biomarkers, circulating biomarkers for therapeutic monitoring in the plasma of CTD patients. We have demonstrated the potential of CBT-101 treatment to modulate those biomarkers level in clinical model of CTD, which will be useful, we hope, for the clinical phase 2 trial for CBT-101 coming in 2026. And finally, we validate the relevance of preclinical models, mice models, and human brain organoids we developed in the Mabonzo lab. For now, we have potential circulating biomarkers, and we need to know if they will be modulated by the CBT-101 treatment in a relevant model. So to answer this question, we intended to develop a new organoid model. It will be a choroid plexus model producing CSF in vitro and derived from CTD patient iPSCs. Body fluids such as CSF or plasma are considered as important sources for biomarkers research, and they are accessible during clinical trials. So this model producing CSF will give us access to such biological fluid. For this project, our PhD student, Lola Gillette, will implement a guided protocol to establish a choroid plexus organoid model from CTD patients. Cells and including also both male and female patients. So in the lab, we already have such model producing in vitro CSF, as we can see here on those vesicles. But here they are derived from healthy cells. So we deeply characterize this model. And among other things, we have shown a great similarity between the proteome of CSF produced in vitro and the pediatric and embryonic CSF, human CSF. So this new CTD model will allow us to discover neurological correlates between choroid plexus, in vitro CSF, CTD mice, and also plasma clinical samples. So we will also evaluate the proteomic changes after the CBT-101 treatment on those organoids. We expect that translational in vitro tools such as organoid derived from CTD patient cells will be a great asset to predict therapeutic efficacy of drug candidates such as CBT-101 before and during clinical trials. So I would like to thank you for your attention and also thank all the persons involved in this project. So the patients and family, the American and French Association for CDS, the LENIT lab, so Mabonzo lab at CA, particularly my mentor, Aloïse Mabonzo, and also Dr. Aurore Curie at Hospice Civil de Lyon to give us access to those very precious plasma samples. So thank you very much.
Thank you so much, Clemence. That was great. Stay on for the Q&A that we'll get to at this session. Our next speaker is Israel Abebe Admasu. He is currently at the Boston Children's Hospital, where he's working as a postdoctoral researcher. He has a background in primary care and specialization in basic and translational neurosciences. In particular, he has an interest in identifying functional brain signatures that inform diagnosis and tracking of disease progression as well as therapeutic response. He's currently an ACD fellow. He is working on a project evaluating the utility of functional near-infrared spectroscopy, or FNIRS, and developing physiologically grounded biomarkers for us to enable monitoring treatment changes. Israel, it's my pleasure to welcome you here today. Please go ahead and bring up your slides and we can get started.
Thank you, Sangeeta, for the good presentation. Hi, everyone. I've been presenter, so I will be talking to you about how establishing a novel functional biomarker for creatine deficiency syndromes. So going to the background, you have been hearing a lot about it today, so I would just go very fast on this one. So as you know, creatine deficiency syndromes are metabolic disorders caused by mutations in genes involving either creatine transportation or its metabolism. And creatinine is in the body. We get it through diet, and we also get it through biosynthetic pathways. So the biosynthetic pathways, Sangeeta was also mentioning it before on her presentation. So the kidney, the liver, and then all those things. I wouldn't be going through that, but it kind of targets the brain and the central nervous system because these are high energy demanding tissues, and the role of creatinine is in energetics. So it leads to all kinds of symptoms, including developmental delay, intellectual disability, speech and motor impairments, and seizures. So what we've been hearing until now is there are promising emerging therapies, but we need to see if those therapies objectively make a difference. So to objectively quantify the severity and response to treatment, our project aims to answer that question. So for the methods that we will be using, we chose an FNIRS because there are preclinical models showing that the energetics, the hemodynamic coupling in the brain is affected. So this kind of captures that in humans, and also it is very portable, it is non-invasive and very safe to do, and it is can be very well integrated with other modalities in order to increase its utility. So here you can see a patient looking at a monitor. With a monitor, we give a 10-minute cartoon movie that is in a very low contrast, so it won't be invoking any kind of activity, but then there comes a checkerboard appearance once in a while, so that we would see the response to that checkerboard. So what the ones that you see on the back side of this patient is the cape, the FNIRS cape. So it emits laser, it emits the functional near-infrared spectroscope. That's red light, and that's detected by detectors. And then it will tell us how much of oxygenated and deoxygenated hemoglobin are in the brain tissue. And that would inform us of the activity of how the cortex is being stimulated or inhibited, or its activity towards this checkerboard appearance. So to start with the method, then the checkerboard simulation evokes distinct hemodynamic response, as you can see here, but it's kind of difficult to put it all together. And then but as you can see here, you can see there is a distinct kind of response that we get by giving out this checkerboard. And as you can see on the gray, it's a mock, and then on the red, it's a checkerboard appearance. So it's different. And if you see it in the control group, so we had 17 controls already in this data, 17 CTD patients and 8 GAMT patients. So in the control, as well as the CTD patients, as well as the GAMT patients, you could see the checkerboard is significantly different from the mock stimuli. So that's what we try to quantify as stimuli. I'm saying the checkerboard and not the cartoon. So the visual stimulation, when you put it all together, like initially I would be covering for CTD, and then later on I'd come back to GAMT. So the visual stimulation, the checkerboard stimuli, produces a greater and sustained cortical hemodynamic change, particularly in CTD patients, as can be seen. This is an average, group average of the controls in the CTD. And as you can see this exaggeration of hemodynamic response in the oxygenated hemoglobin, deoxygenated hemoglobin, as well as the total hemoglobin that's reflected. So in order to say this is just an average, so we need to go and then to see statistically how is it significant, or how does it put up. So in order to do that, I took the oxygenated hemoglobin here as an example, but we quantified for everything. So the first thing I quantified is the latency. It takes it to reach 10% of the maximum. So how fast does it reach 10% of its peak? So the lower the number it is, the faster it has attained the 10%. And then the 50%, it kind of tells you when is the half-life, or when do we attain the 50%. And then obviously the peak, we also quantified the peak. So also for the peak, it's a latency. So the lesser it is, the faster that it gets it to the peak. And then 2 more parameters that are the maximum amplitude that is reached, and then also the amplitude at 20 seconds, just to see how it would return to the baseline. And you could see some here, but is it significant? And as you could see here, CTD participants exhibit early onset hemodynamic response during visual stimulation, and that is seen, even though here it's not significant, then the trend is towards an early attainment of that 10% of the peak. And that goes also for the 50% of the peak amplitude, which follows similar trend, and it attains that 50% of the peak in a relatively shorter amount of time. So this is the time that has taken it to attain that 50% of the peak. And if you go and if you see the latency to peak, you could see that there is no significant difference, or even a difference, because you could see that all the oxygenated hemoglobin, the total hemoglobin, and the deoxygenated hemoglobin reach to the peak at about around the same time, and it's not statistically significant. But so the thing is, we have previously relied on picking the peak amplitude. So the peak amplitude, as you could see, is significantly different. And here, it's not the lower is the higher, it is the higher is the higher. So the peak amplitude, as it gets higher, then it means there is a greater activation. So as you can see here, the oxygenated hemoglobin, the total hemoglobin, and the deoxygenated hemoglobin all have a higher peak. So if you see the deoxygenated hemoglobin, it's always on the opposite side of the others because it kind of decreases. So you could see that it is significantly different. If you go and if you look at 20 seconds after the onset of stimulation, then you would see that the control gets back almost to zero, to the baseline on each situation. But for the CTD participants, they have a higher than zero amplitude, meaning that they don't get back to baseline. So once stimulation hits, then at 20 seconds, they won't go back down to CTD, to the baseline, whereas for the controls, it does. In terms of ages, we tried to plot the amplitude against age, and on the first row you could see the control, and on the second you could see CTD participants, and for everyone, it shows that with an increase in age there is a decrease in amplitude, maybe due to skull thickening or any other thing, but we are not particularly focused on the pathophysiology and mechanism of these things for now. We just want to say if this could be used to pick out how treatment is affected a patient. But it's interesting to see, and a lot of data comes out of this, which could be used in a future research. And if you see also the rise time, there is something here. So in the control group, the younger individuals kind of respond early, so they have a fast, an early onset dynamics, whereas for the old ones, for the old participants, for both control and CTD, for CTD it's kind of lost, but for control specifically, it kind of goes up. So this could also be telling us about the maturity of the central nervous system, how it's processing, but this is just a speculation, and we would be not focused here for now. Now, what I want you to take as a key takeaway so far is CTD participants show a hemodynamic response that is marked by early onset, faster rise, higher peak, and slower return to baseline compared with controls. And this is the distinctive. And the next one is peak amplitude for specifically controls, but for both controls and CTD kind of decreases with age. I say kind of because it's not statistically significant, as I showed, but with aging controls shows a similar but non-significant trend in CTD patients. So among controls, the oxygenated hemoglobin rises more rapidly in younger participants, whereas the oxygenated hemoglobin shows a quicker rise with advancing age. So this was a story for the CTD participants and control participants. Now turning back to patient participants with GAMT. Before I show any data, I want to say that we only had 8 patients, which is half of the CTD and controls. So any statistical significance is not as much dependable, and so it would be more of showing trends. So the checkerboard stimulation evokes distinct hemodynamic response across groups, similar with the CTD, similar with control. Now, if you put the CTD and the control graph, and if you plot on top of it the GAMT group, then you would see that it lies intermediate between these two groups, between the CTD and between the control. This is quite interesting, as also at a speculation level now also, it could be that the GAMT is somehow improved with creatine supplements, and it better responds to treatment. So maybe could it be a matter of treatment? We don't know, but this is quite interesting to explore also for later. So when you come to GAMT and when you try to see the numbers, even though I say that it's not the numbers doesn't allow us for much of a quantification and significance values. When you come to GAMT participants, they show similar trends with CTD. So if you see here, this is a time that's taken them to reach 10% of their peak, and it shows consistently lower than controls, as it was shown with not so much here, but as you could see on it, the oxygenated hemoglobin and the deoxygenated hemoglobin, you could see a trend that they have a faster rise as a patient with CTD, but not as much strong, so not that much significant. And also for the rise time goes the same story. So for the oxygenated hemoglobin, total hemoglobin, deoxygenated hemoglobin, it shows similar pattern, but not significant for at least total hemoglobin and deoxygenated hemoglobin. So latency to peak remains non-significant, but on the oxygenated hemoglobin, it kind of shows. And you could also see on the previous graphs that I showed that the peak comes a bit earlier than the control ones, but this is yet to be tested with more patients and more participants. And the peak amplitude also follows the same trend. It's higher than controls as in CTD. It's higher than control as in CTD, but we fail to attain the significance level. Same goes with amplitude. It's non-significant, so it's not as strong as the CTD. It doesn't go up, and we have fewer patients, but the trend, as you can see, is here, and it is above 0 for most part. Now, in the case of GAMT, this is at a group level, so a group average level. But if you look very closely at the data, and if you try to go back and see the history, and if you dig further, you would see that there are some outliers that makes it closer to the CTD than the control. And interestingly, these 2 points, this point particularly is a participant who was diagnosed late and who started treatment late. And the second point is a participant who is young, but still who have not yet started treatment. And it would be so much interesting to record this participant again and see how it would affect it. So we can see how treatment is affecting the progression in terms of all the parameters that I have shown you before. And same goes for these same participants. You could see them as an outliers also on the hemoglobin, on the total hemoglobin. But on the deoxygenated hemoglobin, they kind of lose their outlier level, and they just mix with the group. We had 8 patients, as I said, for GAMT, and we had 3 female patients and 5 male patients. So this is not as well that much meaningful, but when you compare the peak amplitude, which is established, when you compare them, then you would see that there is no statistical difference between these two. But the outliers still remain. We have 1 female outlier and 1 male outlier, which follows the same kind of story. So for the future directions, what I've shown you until now is the latencies and the amplitude, but I was also looking at other ways of quantifying and other ways of discriminating this. And if you could just focus on here, enlarge it on this area, even on the first 3 seconds of stimulus presentation, you could see there is a clear distinction between groups. So if we could integrate this together with all the measures that I've shown you to be significant, then we could say we will have a good rater that will tell us the classification of patients. So for the next steps, we need more participants, definitely. The ones that I'm just showing you. If you further subclassify them into male and female, if you further classify them based on also skin color and hair color also affects FNIRS as it is light, so absorption and diffraction are different. So we want to see everything. And when you want to see everything, it's not possible with our current number of participants, especially for GAMT. And we would like to see a second visit for already enrolled patients with special attention to patients who are outliers and who started treatment. So we want to see how it goes. We want an in-depth analysis with clinical correlations, and then we want to do some analysis with machine learning. It's a lot of data, and we just measured what we can measure, and just by eyeballing it and then saying, if we quantify this, when we would get a difference, but that would be totally out, and we would have a fairly unbiased analysis. And we need to also, this is a multi-site study, so it's not only done in Boston Children's Hospital, but it's also done elsewhere in Europe. We have collaborators all over the world, and our patients come from all over the world, even as far as Australia. So we want to make this a good collaboration, and we want to see and integrate the data of what we have here together with other sites. All in all, the take-home message from my presentation is that cerebral creatine deficiency syndromes exhibit distinct hemodynamic patterns that are useful for monitoring patient progress as new treatments become available. So we can objectively say, we can objectively see if there is any difference that is being brought to the patient. So while GAMT follows trends similar to CTD, early intervention appears to normalize hemodynamic features towards control levels, because this is more of speculative, as I said, because those two outliers are more of outside, because one is with a late treatment and one is not on treatment, and that would give us that kind of clue. And incorporating quantitative features such as latencies and initial oxygenated hemoglobin responses together with amplitude measurement may enhance diagnostic specificity, supporting more accurate evaluation. So if you see only peak amplitude, then you wouldn't be open to the other features. But if you try to integrate all the features that are different in this group of participants, then you would really have something that could potentially have a higher sensitivity and sensitivity and specificity. So, which brings me to my last slide. So in terms of acknowledgement, I would like to acknowledge ACD, because this work is funded by ACD, and we are very thankful to let us do this. And even I want to thank the participants very much, because you would see that it's very enriching interaction and experience that we have with each other, and I'm very happy to be part of this. Adeline is a research assistant working with me, for which she does most of the powerlifting for organizing how patients come and taking care of them, and also getting things in order for the travel, for the stay, and also for consent and so much. So, and she also actively engaged in the analysis part as well. So I would like to thank Professor Michela Pagiolini and Professor Laura Baroncelli, my supervisor and my mentors, who are amazing at mentoring us, who are showing us ways, and whose dedication means a lot to us. And Dr. Raffaele Maseotti from University of Florence, we have been using his pipeline, and also he's been active in setting up our analysis and helping us in such. And also Professor Takao Hensch for helping us with acquiring of analysis software, and Camilla Simoncelli for helping me with the data analysis as well. So this is not a single person who was doing all this thing. And thank you so much for your attention. And that will be it for me.
Thank you, Israel. That was great. Yeah, I do agree. I think it'd be great to see some more numbers on GAMT patients. So this is a call out to the community, call to action. So if you'd like to learn more about FNIRS, or if you have any particular questions on whether it's invasive and what the protocol involves, then please get in touch with us. We don't want any of those fears to be a roadblock to getting the data, because it's only when we do get this data we're going to advance in our understanding of the disorders. Okay, at this time, I'll introduce our next speaker, Jerry Lipschutz. So my pleasure to introduce Jerry. Jerry is a California native. He got his medical degree and completed his postgraduate training all in California, UCLA and UCSF to be specific. He's a professor within the Department of Surgery and Department of Molecular and Medical Pharmacology at UCLA. He's also a member of the Intellectual and Developmental Disabilities Research Center at UCLA, along with the Broad Center. He's a physician scientist, and his lab has focused on developing an improved understanding of the neuropathophysiology of rare metabolic diseases. We've been fortunate to have Jerry in our community. We also support a member of his lab through the ACD Fellowship, and you'll hear from Alex tomorrow. All right, Jerry, the floor is yours.
Can you see
can
my
go ahead
slide?
and get started.
Thank you.
Yes,
Thank
we
you so
can.
much. You can. Thank you for the nice introduction. I appreciate it very much. I'm going to talk a little bit about PET imaging, positron emission tomography, reporters as biomarkers for CTD. And I'm going to spend the first 4 slides giving a little bit of background so that hopefully we're all on the same page from the beginning here. So we need to talk a little bit about glucose transport, or how sugar is transported into cells. So glucose can be transported into cells by sodium-dependent glucose transporters. That is, they require sodium to help drive, an ion to help drive glucose into cells, or it can be done independent of sodium. So there's 3 steps for glucose transport into neurons or nerve cells. Glucose must be transported through the endothelial cell lining of the capillary, the blood-brain barrier, using the GLUT1 transporter. There's more than 5 of these different types of transporters. This one was called GLUT1. It is then taken up by GLUT1 on the surface of astrocytes. So sugar crosses the blood-brain barrier, then taken up by the supporting cell. Some people refer to a supporting cell called astrocytes. And then the astrocytes pass that glucose with the GLUT1 transporter into the intracellular space around neurons, so that neurons can then take up that glucose through a different transporter called GLUT3. And this is sort of a depiction of these transporters on the left side of the screen, where glucose comes in. It's basically a channel that allows glucose to come in, either be independent of sodium, or when it's going up a concentration gradient, often sodium will be transported across the gradient to drive glucose into cells. So you have these different transporters, and depending on what organ we're talking about or what tissue we're talking about, there can be different types of those glucose transporters: GLUT1, GLUT3, GLUT5, different types of transporters. So that's how glucose gets into cells. In the brain, glucose gets in really independent of insulin driving it in, but in other tissues and other organs, insulin is needed to drive glucose in. So several decades ago or more, people had come up with this concept of imaging based on important molecules in your body, one of them being glucose. So if you have tissues or organs that are highly metabolically active, needing a lot of energy, they need glucose. And so someone had this concept, someone here at UCLA, in fact, Michael Phelps is one of those people, had this concept that maybe you could do something with glucose. You could modify in a way so you could use an imager to see that the activity of that glucose. So how this was done is this technique called PET, or positron emission tomography, computed tomography imaging. So in this case, here's glucose in the center, the molecular structure of glucose, and you can bind to it a radioactive molecule. In this case, fluorine, like fluoride for tooth protection, but fluorine. And it's a radioactive form of fluorine called fluorine-18. The fluorine that we encounter in our lives every day is fluorine-19, but this fluorine-18 is unstable, and it will eject off a positron as it degrades. And its half-life is about a little less than 2 hours. So what can be done is this glucose analog called 18F or 18-fluoro, fluorodeoxyglucose, can be generated, and this can be administered to an animal, to a human, and it will be taken up into cells just like glucose normally is. And inside those cells, it's phosphorylated. A phosphorus is put onto it, just like glucose normally is. But instead of generating 6-glucose phosphate, you generate 18F fluorodeoxyglucose 6-phosphate. And that glucose as fluorodeoxyglucose with the phosphate doesn't lead to the production of energy in your cells. It stays trapped in the cells. So you can, if you have a highly metabolically active tissue requiring a lot of energy, you can see those cells on an imager and see the organ on the imager and how much energy it needs. And so people have applied this technology to look for cells in someone's body that is highly metabolically active. So for example, cancer. Cancer's looked at this way because cancer cells are very metabolically active. They're dividing, they require a lot of energy. So someone will get a PET CT of their entire body to look for a tumor somewhere. Let's say if they have colon cancer, and you're concerned that it went somewhere else in the body, you get a CAT scan after being given intravenously 18-fluorodeoxyglucose, and you look for highlighted areas in the body. And so you might be able to apply the same type of technology to creatine transporter disorder or GAMT deficiency. So in this case, or in the other cases too, you'd have an instrument called a cyclotron, which would generate the radioactive isotope. You have a radiochemist who will take that fluorine-18 and link it to glucose, and then you administer that intravenously to someone, and then you lie down on a table, and you're imaged here, and you can have an image of someone's brain, and you can look at the different colors can mean different things based on activity. So we thought about this for creatine transporter disorder, and we decided really to look at in mice only. We decided to look at 3 different tissues or organs that we know are important in the disorder. Now, the creatine transporter is in more than just those organs. We're looking at the brain, we're looking at the heart, looking at skeletal muscle, but creatine transport is also important in many other places like the retina, the back of the eye for vision. It's important in the skin, in the gastrointestinal tract. I think probably many of the parents know about this for their children. In the placenta during child growth, delivery, red blood cells, white blood cells, and in the kidneys. So it has much more activity than and importance than the 3 tissues and organs I'm going to talk about here. But this is to give you that's the 4 slides of background. So we have the creatine transporter deficient mice that Matt Skelton at Cincinnati Children's developed, and we decided first to take those mice and see if they might have any abnormalities in glucose metabolism in the blood. So we collected blood from these mice, and we just measured their glucose. And in fact, all the colors are correlating between groups here, but in black we have wild type, in blue we have creatine transporter mutant mice, and the glucose is lower in the blood in creatine transporter deficient mice, whether they're not fasted, they've recently eaten, or fasted, it's lower. And the insulin is lower also in creatine transporter deficient mice. So they have lower glucoses and they have lower insulin. And you can think about this a lot of different ways, and you probably can come up with a theory as to why they might be lower based on this issue, or why they might be lower based on this issue. But we had some thoughts about this, and we performed PET CT imaging on the mice themselves. And what we see here in these 3 panels right here on the bottom where you see my laser pointer moving, these are wild type mice. So normal mice, they're looking with their side and profile on these anesthetized mice in this imaging. And the red oval represents a circle around the brain. And what you see is different colors. And if we look at this as a scale here from it's a pseudo scale from 0 to 10, where red is a lot of activity, black is really no activity, blue, light blue, green, and then yellow advanced, like in a rainbow. So if we look at the brain of wild type mice, they have this level of activity, a mid-level of activity. And we know the reason that they have that level of activity is because they have two sources of energy. They have glucose and they have creatine phosphate from creatine through the creatine transporter. But if we look at the top 3 mice, these are mice that have creatine transporter deficiency. They receive the exact same treatment, were tested the same day at the same time. And we see there's much more glucose-based metabolic activity in the brain, much more. Some ways it makes sense. You don't have the creatine transporter that's functional, so you don't have any substantial amount of creatine phosphate. So the energy source that's exclusive here is glucose. And that's what we're seeing in the top row. We're seeing that the creatine transporter deficient mice, their brain relies on much glucose metabolism. That's represented here quantitatively. Here's wild type mice. Here's the creatine transporter deficient mice. We have a largely statistically significant difference between these two groups of mice. And so this is really the model drawn as a cartoon here. So on the left are normal mice. We have two sources of energy. We see glucose here in red. We see this size of an arrow. We see over here the creatine transporter, CRT1, bringing in creatine. You get creatine phosphate. It's used to regenerate adenosine triphosphate, or ATP, which is the source of energy. When the creatine transporter is mutated and not functional, essentially this half of the system is no longer working. So the entire system for producing energy is based on glucose, represented by this much larger arrow, to produce adenosine triphosphate or energy. So we see this difference in the brain based on glucose metabolism. And clearly, at least in mice, PET-CT shows a difference. Now, we looked in different areas of the brain to see if these differences occur in one location, in several locations, or all locations. In fact, we see the differences. We detect the differences in metabolic activity based on glucose in all areas that we examined in the brain, whether it's the hypothalamus, the brainstem, the cerebellum, the globus pallidus. You find it everywhere, except for whatever reason in the olfactory bulbs, which are related for smell. I can't tell you why that is. Here are the data points. They cross some. It's a trend towards significance, but it's not significant. But there's a significant difference in all the other regions of the brain based on metabolic imaging for glucose. So we were trying to understand why this is, and if the brain has the capacity, because you might ask yourself, well, if all the energy is then based on glucose, is there a way to get more glucose into the brain to overcome the absence of not having absence of creatine or creatine phosphate? And it does not appear so by the data that I'm going to show you now. It doesn't appear that the brain, it modifies enough to take in more glucose to change itself to overcome this problem. We looked at different transporters in brain cells. We looked at GLUT1, the ubiquitous glucose transporter. It's in the blood-brain barrier. It's in the brain also. We looked at GLUT3, which is the very high affinity transporter I mentioned earlier that's on neurons. And if we compare them to wild type or normal mice, which is the red line, the amount of RNA or genetic component that will lead to production of the transporter is essentially the same for GLUT1, the same for GLUT3. So the transporter amount doesn't appear to be modifiable. That is, you can increase the amount of transporters to get more glucose in. But then we looked at other molecules or other proteins. We looked at kinases that are involved in examining if there's energy stress or a decrease in energy. One called LKB1, another one called calmodulin, a kinase that are activators of this AMP kinase, and neither one of these are upregulated either. And we also looked at AGAT and GAMT, and they're not increased either to produce more creatine. So for whatever reason, there is a limitation in the amount of genetic material, RNA, that can be increased to produce more transporter or to increase the signaling to say that energy is low in the brain to somehow increase energy. This mechanism does not seem to exist, at least that way that we studied this. Doesn't seem to be that this can be modified. So we went on to look at other tissues and organs. We looked at the heart here on the left, and we looked at skeletal muscle here on the right. And the position of the images are based the way the mice are lying. On the side here for the heart, lying on its back here for the muscle. So in the heart, we see at the bottom, wild type mice. We see the heart represented by this black circle here. We see mostly it's in green. But then when we see the mice that are creatine transporter mutated, we see a little more yellow and at least red in this mouse. And there is essentially a statistical significantly difference between the creatine transporter deficient mice and the mice that are wild type. Again, because of the absence of creatine phosphate or minimal creatine phosphate, we have more metabolism in the heart based on a glucose. And we see a similar finding in skeletal muscle. We're looking at the tricep, upper arm. Mouse is shaped in this position here. And we see again a difference in the muscle, a signal between wild type mice here and mice that are creatine transporter mutated. There's more energy metabolism based on glucose than any other source for energy. Now, as we looked at this, we were also studying these mice to see if we could gain a greater understanding of the brain abnormality that occurs in this disorder. So we performed some behavioral testing. Some other people can make presentations about behavioral testing, and one we did was this test called Open Field. You can see it here. Mice that have minimal anxiety, they'll run around in this box, and the surface, it can detect they're moving around, and they'll spend time in the center and time in the periphery. But mice with anxiety will spend more time in the periphery than they do in the center. Like this mouse doesn't have any anxiety. It spends more time in the center than it does in the periphery, or at least equal times. And so what we found in the creatine transporter deficient mice is they seem to have some anxiety phenotype at least. If you look at in blue are the mutated mice, they travel more in a shorter period of time. They travel faster. They spend less time in the center. They enter the center less frequently. So these are all findings consistent with some level of anxiety in the mice. But we did some other testing, and it's a little bit. This was touched on a little bit with part of this earlier. That we looked at a test called marble burying. You have a cage. In the bottom is basically sawdust. You put some marbles in it. It's done a little more sophisticated than I'm describing. You put a mouse in, and mice will always go in. They'll typically bury those marbles. Or if you take a piece of cotton, we call it a nestlet. Looks kind of like a piece of flat cotton. You put it in a cage. Mice will tear it up and they'll make a nest from it. But if we look at these activities in creatine transporter mutated mice, they're abnormal. The marble burying is greatly decreased. The number of marbles that are buried, the time it takes for them to start burying a marble is delayed. The number of times they try to bury a marble is small. They just don't bury marbles well. They don't really tear up the nestlet. They don't make a nest out of it. It takes much more time for them to do this. When you see these types of abnormalities in mice, it makes you think that they have a serotonergic signaling abnormality in. And serotonin is a neurotransmitter, just like GABA and other neurotransmitters, dopamine that exist in the brain. And these findings together suggested to us that maybe there's this enhanced serotonergic activity or enhanced serotonergic tone that's going on that's causing the abnormality in the performance of these tests. And so what could all this mean? So marble burying and nestlet shredding, these are spontaneous, they're normal, they're repetitive digging type behaviors for mice. They're thought to reflect exploratory drive or anxiety-like behavior or compulsivity if they're done too much. Serotonin reuptake inhibitors reliably induce marble burying and nestlet shredding in mice. So if you give certain types of agents that are used to prevent depression or to treat depression, mice will then have alterations in marble burying and shredding. And what could decreased performance like we're seeing here mean? Well, it could mean they have this reduced repetitive or compulsive drive. They could have this increased serotonergic tone that I've touched on, and that maybe if we lower these serotonin levels, maybe they could have a better behavior. And that's something that we want to consider. Now, we can't think of this in a vacuum all by itself, because the neurotransmitters and the neurons interact together. And it's a very complicated system. And so the behaviors that I'm describing are not completely pure serotonergic. There's some effect from the dopamine system, from the glutamate system, from the GABA system, but in aggregate, the abnormalities that we're seeing with marble bearing, nestlet shredding, some evidence of anxiety, suggest that there's this serotonergic abnormality that's going on. So we decided that we wanted to come up with a hypothesis that we thought might work, that might put this together, and maybe we could test it later on based on how could this occur in creatine transporter disorder. So we know that there's as I described earlier, there's somewhat of an energy shortfall in the brain with creatine transporter disorder. So you'd have less ATP around that's buffering the Raphe nuclei, that's buffering the target circuits that exist. And that could lead to less serotonin or 5-hydroxytryptamine, two names for the same thing, release from synaptic vesicles. And there could be some form of compensation that exists, that receptors upregulate because there's less present. So in order for those cells to detect it, they upregulate their receptors. And some receptors may desensitize. And the net effect would be that those receptors then could, quote, feel more serotonin at baseline, and that could lead to this increased tone of serotonergic activity. This is our hypothesis. So in our preclinical imaging facility here, there's a radiochemist, and he synthesized this compound that has a very long name, but we abbreviate MPPF, and labeled it with 18F, just like the 18-fluorodeoxyglucose that I mentioned, 18F-fluorodeoxyglucose. So again, another radio tracer here being tested, but this time not looking at glucose activity, looking at serotonergic tone or binding to the 5-hydroxytryptamine 1-alpha receptor, a receptor for serotonin. And what we find here is this. In the bottom are wild-type mice, again, profile. The oval represents the mouse brain. We see this imaging here, the different colors, the pseudocolor scale here. And then if we look at the mutated mice, we in fact see more activity. It tends to be more focused towards the frontal lobes in all three of these mice that were tested here. And if we look statistically, we're approaching statistical significance. 0.05 typically or less. We consider statistically significant 0.07. So certainly there's a strong trend towards significance, suggesting that there is an underlying mechanism for possibly this anxiolytic, anti-obsessive compulsive like behavior in these mice that may be related to serotonergic activity. Now, Matt Skelton in these mice previously did demonstrate by taking the brains out and examining for certain compounds that there was some increase in biochemical marker for this also. But these findings further confirm that there in fact may be some serotonergic abnormalities in these mice that may lead to potentially some evaluation, at least in mice, whether a pharmacologic therapy may change the behavior, may improve the behavior in the mice. So in conclusion, what I've discussed here in the last 18 minutes or so is that brain glucose metabolic activity is increased in mouse creatine transporter disorder, that regulators of AMP kinase and expression of GLUT1 and GLUT3 are not increased, but the plasma glucose is altered and the insulin levels are decreased in these mice, that 18F-fluorodeoxyglucose appears to be appropriate radiopharmaceutical brain biomarker for CTD and could be useful for assessing a change in brain glucose metabolism with gene therapeutic approaches. Serotonergic tone appears to be altered, at least in the mice, and that 18F-MPPF may be useful to assess changes in brain serotonergic activity with pharmacologic or gene therapy approaches. And finally, the use of specific radiopharmaceuticals for imaging, in my opinion, should be considered in therapeutic clinical trials for CTD. And with that, I'd just like to acknowledge the other members of our lab group who've worked on this project and others, Andreas Scholze and Aloni Takakova at Hospital for Sick Children's, and ACD for their support in our work in this field. Thank you.
Thanks, Jerry. That was a great talk. There's some really interesting implications there for change levels of serotonin if those are found to be confirmed in humans and CTD patients as well. Although it's maybe still a bit of a, I don't know if it's what your thoughts are about that, but we should get to that in the Q&A. I should hand off to Kim and Matt, who will be our moderators for this session. As before, Kim and Matt, there's a ton of questions in the Q&A box. Please take a look at it. It'd be great if you could give a brief introduction to yourselves and do round robin for questions, just so we get to everyone. And I'll hand over to you.
Hi, everybody. I'm Kim Cecil. I am a magnetic resonance spectroscopist. So I use clinically and in research settings MR spectroscopy to diagnose, find children with creatine transporter deficiency and GAMT disorders.
Hi, everybody. My name's Matt Henley. Been active with ACD for about 6 or 9 months now. I'm a new board member. I have a 4-year-old son with CTD. Enjoyed the presentations, excited to get to the questions.
Yes, they're excellent. We can just alternate if you want to, Matt, and hit the questions. We may jump a little bit so we get all 3 speakers some questions. So I think from our first speaker, Dr. Distier, this was great research. Could you see this research also inform other treatment and supplementation other than the CBT 101, which you've shown?
So of course, so for now, the circulating biomarkers, it's in the plasma from patients, so no patient receive any experimental treatments. So we don't know if the CBT 101 have any effect, but we have in the data in clinical model, and as we work very closely with CNS brain therapeutics, of course, we look if the CBT 101 has an effect. We have the tool to evaluate other treatments, so it's definitely something that we can do, but for now we have no data.
And let me follow up one more from that about the variability of the plasma biomarkers you saw. How big is that variation?
So in fact, we selected those biomarkers in plasma because there is very low variability between the patients. So we have for now 9 patients. So we selected those biomarkers because all over the patients group, it's significantly decreased. So it was our approach to select those biomarkers. And of course, the next step will be to validate those biomarkers by ELISA. And so it's why we want to increase the healthy volunteer groups and the CTD patients group number.
Thank you.
I will ask a question of Israel. Question here is the CTD data males only? Is there female data analyzed? And I guess I would kind of add there if you found any differences.
Sorry, I was muted, sorry.
It's all right.
So we had 17 CTD patients, 16 of which were males and 1 was female. So for the sake of numbers, we just analyzed it all together, similar with GAMT, except that I specifically mentioned males against females. Then we analyzed it all together because if you take it apart, it wouldn't make any sense comparing 16 with 1. But yeah, we are looking forward to getting more participants.
Can I do a follow-up on that? You mentioned for multi-site collaborations, how much variation across sites? So if we did this at Cincinnati, do we have to calibrate our scanners, our FNIR system? How difficult is that?
Not so much difficult, but so the graph that I showed specifically regarding the amplitude was produced also in Italy. They also did some of that, and then they could see similar kind of peak amplitude elevation. So that was it. But for the latencies, it's kind of a new analysis that I have added, and so we don't know, but we should be able to reproduce it wherever it is, and that will be seen when the data from all over the sites integrate all together and we see how it goes.
Thank you.
You're welcome.
So for Jerry, the question, how much FDG is needed for PET, and does administration perturb normal cellular metabolism?
I think it's about 5 to 15 microcuries, so it's very small. It doesn't change normal metabolism because the 18F FDG glucose that's phosphorylated remains in cells and does not go down the glycolytic pathway, so it doesn't alter the metabolism.
And how safe for families and their children? Yeah.
Yeah, it's an interesting question. You know, people undergo PET CTs for all different types of reasons, and in general, people would say, you know, no amount of radiation is safe, but there are guidelines on how much radiation people should have or allowed to have in a year. So it's hard for me to answer the question specifically. The half-life for the isotope is about 109 minutes. So typical precaution is, at least with mice, that once they have the procedure, we can do anything we want with them a day later. So it's not around very long. It's short-lived. That's about the best that I can answer that question.
Yeah. Speaking as someone from a radiology department, Cincinnati Children's, we do PET CT a lot. For answering a question like this, if we got to the point, I don't know, we would have to address how frequent for monitoring, but at this stage, in animals, we can do this type of research to help really answer the questions we're trying to answer.
Thank you.
All right, so I have a question for Clemence. I hope I'm pronouncing that good enough. Have you done any IHC experiments with these biomarkers? Do you think any of these could be used as a proxy for SLC6A8 expression?
So we didn't do yet IHC. What we plan is to do ELISA in the plasma of more patients and more healthy volunteers to validate the relevance of those biomarkers. And we can, of course, we can do IHC in the mice brain or in organoids. So we can also do Western blotting, etc., to really quantify the protein expression. We didn't do yet. So we compare all the database we have by proteomic to answer for this question. And can we use it as a proxy for the SLC6A8 expression? It's a very good question. I think we're going to need to first validate, like correlate the functionality of the transporter with the level of those biomarkers to really answer this question.
Okay. Israel, are you still doing these studies? Is there funding available for them?
That's a question. That's a good question. So funding, we are running low on funding, especially for patients travel and stay at Boston and so on. Also, I'm a fellow of SED, and I will be running out of the funding in the coming months. So it would be an amazing to have a funding source, but we are very much open to participants, and we would really appreciate it if we can get more participants. Usually participants are worried that their kids might not tolerate it well and so on, but it's a 10-minute of cartoon, and some of the participants were already refusing to leave, and some had issues, but it's okay. Like they can sit on the parent and watch the movie and so on. So it's so much flexible. So we are very much open, and we would very much like to have more participants, but on the funding, we are a bit on the low side, and it's kind of tough when you see that promising result and then when you want to spring, but funding is an issue.
And how reproducible are those line shapes? And does the CTD and GAMT line shapes look different than other affected other disease populations?
Yes, they kind of do. The thing is, if it was just an amplitude, then you wouldn't see any difference in the latencies. And if it was just latencies, it would just remain flat, and the amplitude, you wouldn't see much difference, but it's everything. So like the latencies are affected, the amplitude is affected, the return to baseline is affected, everything is affected. So if you place them all together, as I've shown, then you would see that they are different for sure. And it also looks kind of broad and multiple picked, the CTD ones. So like I tried also to analyze it in terms of number of picks that it has and so on, but it's more of exploratory for now. So I did not want to put that up, but once we kind of go through a machine learning process and so on, then we would have something that we would say, okay, this thing must be affected and so on. But in terms of the pathophysiology, it's quite interesting what you could squeeze out of all these graphs, because these are all independent, unlike the functional MRI, which just gives you an indirect reflection of the oxygenated and deoxygenated hemoglobin and so on. These ones are independent by themselves, and the level of data and the richness of data that you could squeeze in is a lot. And if you follow the same, the pipeline is already available. So if you follow the same pipeline of pre-processing and so on with the same parameters, I believe that it's reproducible, but we need to do a test and retest or on the already recorded patients to see how it holds up.
Thank you.
All right, the next question's for Jerry. How did you get rid of the peripheral use of FDG bias? Did you normalize the data on a specific region, such as the STEM region as an example?
STEM region, that mean the brain stem?
I would think so.
No, we didn't do it that way. We did it on the liver.
Okay. Sorry, there's so many questions here. So for Clemence, are any of the biomarkers you identified surprising, and what do they teach us about the biology of CTD? I know that's very broad.
So they are not surprising because we choose them because they are linked with neurological disease. So we just like pave the way to those biomarkers because we look only into to proteins that are altered, described as altered in other neurological conditions. So no, they are not surprising at all. And what it teaches about pathological mechanism, maybe all those regulation of all those different biomarkers like make sense all together because we can see a link with neuroinflammation, with oxidative stress, with mitochondrial dysfunction, also a link with a neurodegenerative disease such as the alpha-synuclein, the tau protein, etc., etc. So all together, those make sense. Those are linked to the cognitive impairment in CTD. So it describes a pathological mechanism that is impacted by the lack of creatine in cerebral cells.
Okay, I'll ask another question for Jerry here. Have similar PET studies been done in humans with CTD? And just in case, well, actually, I'll let you answer that one before I ask a follow-up.
Yeah, this is something that I, discussed briefly with Heidi and Sangeetha. I'm not aware of, there's probably someone out there who's had a PET CT who has CTD, but I've not found any reports on it. Could exist, but I've not seen any, and it's a question that I've had, and we've actually, myself with Heidi and Sangeetha, have discussed a little bit about seeing there might be an opportunity for this in the future, but I don't know that there is. So I don't know where it's being done. No.
Right. It probably could have happened for some other
Right.
pathology
Right.
they were going after, and then they probably had an abnormal result and overinterpreted what it meant and
Yep.
may not have looked at the, the right system. So, yeah. So another one for Jerry. Thanks for an interesting talk. I concur. Would you predict that the increased glucose demand you're seeing in your mice could explain why so many CTD patients report very low body weight, even when they are meeting or exceeding their recommended calorie count? Essentially, they're burning all of their energy to compensate for the lack of creatine.
Yeah, that's a really interesting thought. I don't know the answer, but I think it's possible that it's that. But also, if we look at the data presented, I think Robin presented some this time, maybe, or her colleague. And I know that other people have seen this when they perform gene therapy on the GAMT mice and the CTD mice, they gain weight. And I think it's muscle mass. And there's this concept, you hear, for those of us who, or for people who weren't in this field till recently, or maybe weren't in it before at all, you always hear of creatine supplementation for people who want to gain weight, muscle mass. And, I think when you supply the creatine transporter to muscle, like in these gene therapy studies, the transporter or more enzyme, biosynthetic enzymes, you produce more creatine, you have more muscle mass. And so I have a feeling it may be a combination that possibly that maybe it is you don't have enough energy and you don't, so that may be a reason that muscle mass is smaller, but also it's the lack of the transporter or lack of the biosynthetic function that's leading to less muscle mass. That's, I think, probably more likely, because probably not the same concept, but why do the children with CTD have gastrointestinal abnormalities? It's probably because the small intestinal, the smooth muscle myocytes are not functioning well. And that's because of the lack of creatine, the muscle in the intestine. So I think it probably has more to do with just dysfunction of the myocytes. They're smaller in the mice. If you look under a microscope, the muscle cells are smaller. I think it just has to do with the fact they don't have creatine more than the overutilization of energy would be my suspicion.
And are there any PET creatine markers?
I don't know of any. There could be one, but if someone's thinking about that for the brain, they can do what you do,
Yeah.
right? MR
Right.
spectroscopy.
Without radiation.
Yeah. So people might, in the audience, the families might not know about this, and you can do a magnetic resonance MRI, but do it to look at electrolytes or compounds that Dr. Cecil and Dr. Henry, who's on this call, also do, or on this talk also do as an observer. So that can be done that way with no radiation, as you said.
Right. Yes, glucose is a much safer and more, well, now that I say that, it would be interesting to see how creatine and glucose go across the whole body with PET. It's just a theoretical experiment, but yeah, but glucose is probably the better option.
Jerry, I'll ask another one here. PET scans are expensive and already quite less available. How do you see this for wider applicability or even for clinical trials when MRIs are even hard to recruit?
Yeah, you're right, because everything has to be paid for. But I think this would be my suspicion. If someone has a clinical trial, they'll have it at one site, or they'll have it at a couple of sites, and you for the parents, plan these things in advance and make a pre-gene therapy PET scan as part of the study, and then a post-gene therapy, some X number of amounts of weeks or months later, a PET scan, and you make it part of the trial. So it's paid for by the trial. Because I think what you're raising is a very legitimate issue. No insurance companies really in the United States is going to pay for your child to get a PET CT now. They're just not going to do it because there's no, how's it going to change the treatment? It's not going to change the treatment, so they're not going to pay for it. That's how I would see it. So I think you'd have to do it as part of the trial and do it before or afterwards. Or if a trial just was on diagnostic markers for creatine deficiency disorders and you get a grant and it pays for those studies to be performed. But yeah, I don't think you're going to be able to get it, unfortunately, just as a person, not part of a trial.
I have a question for Israel. From Jerry's work showing that the frontal cortex had more activity than potentially the visual cortex, do you look at other regions for stimuli? So I know you used a checkerboard. Could you use sound or some other paradigm for your activity? And would you expect us to have a difference?
Yes, in terms of flexibility, the FNIRS gives us that much more flexibility to also check with other modalities. Although this is just an upcoming, and we are establishing it only in the visual system, but we have plans to also record from other regions of the brain, as well as to integrate it with EEG and see how to improve the temporal correlation of the signal together with the hemodynamic response. I don't know if that answers your question.
Right.
Yeah. Okay. Thank you.
So I know we have 1 minute remaining. So could I ask you to maybe summarize? There was one question here on summarizing the research in this session, key takeaways for parents.
Me?
And Kim, you're this was more for the moderators, but if you want to bring it together
Oh, okay.
for the audience, that would be great.
Right. I think there's a lot of advancements in moving the field. So when we get those gene therapies, we're going to be able to tell what's going on from a relatively, we'll have all these tools and we won't have to experiment on your children. We'll have all these models. And then when we do have to go into those first trials, we will have really targeted questions that can be addressed in smaller sample sizes.
Thank you so much, Kim and Matt, for moderating that session. I know there were a lot of questions and we didn't get to all of them, but it's a great discussion and a great series of talks. Thanks so much for your time. I will go ahead and bring up our last speaker for the day. Heidi is our executive director. She needs no introduction to this group, so I will let her talk about all of the interesting things in the past year and close for today. Heidi, the floor is yours.
All right, great. Hopefully you can see my slides and hear me. Thank you to everyone who attended and participated today by asking questions, by sharing your research, the families who shared their amazing stories and really, I think, set the tone for the day and inspired all of us. Today, I chose to just do a quick look back over what has happened in our community, in our research, in our ACD community, in our family community over the past year since we last spoke during the 2024 conference. And I will be quick because I know it's been a long day, but bear with me. I have a really important message to share, I feel. But first, I will share the polling results that we conducted last week during our caregiver session, which was well attended. And I think that they really highlight the eagerness of our community to find a cure, to be active and participate in research, and to speak on behalf of their children in this process. 50% of the parents who attended had donated their child's biosamples. 93% know their child's mutation. 93% have joined the patient registry. And of the CTD caregivers, 89% said that they would consider gene therapy. And maybe somewhat surprisingly, 100% of the GAMT caregivers attending said they would consider gene therapy. 92% of patients that were diagnosed under the care of these caregivers were diagnosed by a clinician with no other patients with a creatine deficiency. And I really like to highlight that because I feel that we are lacking in centers of excellence and really widely available treatment protocols, and that's something that is a priority for us going forward. In the last year since the symposium at ACD, we've added an ambassador, our ACD China ambassador, Kai, who has been very helpful in supporting patients in China who do not have the same access to the resources, the YouTube recordings and whatnot, and also in assisting our VCEP in translating papers about patient cases that were only available in Chinese and allowing them to be able to curate some of those mutations. And our affiliate ACD Deutschland was launched in the last year. We added 2 new board members. We added 3 new part-time staff and 1 part-time volunteer. And looking at our public platform in the last year, where have we been? Where have we been out representing and educating? We've given talks and posters and attended at meetings, including but not limited to ACMG, ASGCT, NINDS, CZI's, Science and Society Fall and Spring 25 meetings, CBER, Rare Disease Day at Cornell, the Chicago Mono Epilepsy Summit, and others. We've recorded podcasts, and of course, I think everyone is familiar with the huge spotlight that our board member and CTD dad, Jeff Allen, has really shined on our disorders. In the world of advocacy, we've seen more and more adoption of GAMT newborn screening, and we are very near 50% of all babies being screened. By the end of this year, 100% of babies in Australia will be screened, and we are preparing a UK nomination for GAMT. And 2 infants with GAMT have been identified, diagnosed, and treated from birth in the last year since we met. We have submitted 2 ICD-10 code applications to, again, help find patients, help advocate for learning more about the natural history of these diseases and being able to have a larger network. And we've joined the Rare Epilepsy Network. And in supporting research in the last year, we've mentioned some of these. We have 5 new research fellowships, 1 Uplifting Athletes Co-Award to Dr. Chuck Kuntz at the Schlabach Lab. 2 larger research efforts have been ongoing in the past year by Dr. Ingoglia and Dr. Stockler, and we have migrated our patient registry to a new, more friendly platform. And on that note, data from our registry has been used in a publication. And lastly, we launched the Race for a Cure, a call for researchers around the world to submit to us a letter of intent for their projects that they believe are keen to move forward towards clinical trials. And carrying on with research, we've had 2 publications that Sangeetha mentioned earlier. We've helped to recruit patients for the FNIRS study at Boston Children's, and we've conducted a Sarah's Town Hall to share information about phase 1 of their clinical trial. And we've launched a wonderful research paper repository, which I strongly recommend both researchers and families check out on our website. You can search and filter and really drill down into a large collection of research papers on creatine deficiencies. And please do reach out if you find that a paper of importance is not listed. In fundraising for research, so all of this, like we've talked about several times today, is dependent on being able to have funding to move things forward. We had a great 2024 Holiday Heroes in the last year. Our first Ruck for Rare was led by Jeffrey Allen, and we had our 10th Walk for Strength. We had really creative fundraising this year. We had Hope for Oscar, a mom in the Netherlands running 68 kilometers in a single day. A bake sale that went wild and raised over $1,000. And we have Jeff walking 68 miles at the end of this month in a day and a half. We have launched a Momentum League, which is our monthly giving club. It's people saying, I'll give $5, $10, $20 a month just to give you all some momentum and get your minds off of fundraising and on to working on research and what really matters the most to us. And then recently we attended the NASDAQ closing bell ceremony where we officially launched Race for a Cure fundraising initiative. So the component that will fund those grants. And in the last year, in our Parents 2.0 project, we are just a couple of days away from finishing that first year. And we've recruited 24 caregivers, 4 parent mentors, 4 scientific partners, 4 subject matter expert groups. And we have spun up a GAA project that you will hear about at the end of the day tomorrow to study the fluctuations in GAA over the course of a day. We have conducted 8 workshops with these caregivers. And in our community has seen a surge in families finding our support group, in patients getting diagnosed. We have a bi-monthly webinar to welcome the new families and kind of give them an orientation on what this is all about and what's happening. And we've had 47 new CTD families join us since we last met. 19 GAMT families, 1 AGAT, so 67 in all. We've had, like I mentioned, the 2 GAMT newborns that were screened at birth. And then we've had 10 community chats where our community has met together to learn about advocating for their child and many other things. And we've celebrated our CCDS Day. And then lastly, I want to just publicly make my own commitment to this cause known. I've celebrated my 10 years with ACD, which is bittersweet because I think, we would all love to see a treatment for CTD and a better treatment for GAMT by now. Since a year ago, I've had 57 seizures that I've logged for my daughter. We've made 21 adjustments to seizure medications and used rescue meds 9 times. We've given our children 3,340 doses of their supplements, many, many kilos of supplements to help reduce their GAA, but of course not completely normalized. And so my heart goes out to our patient community and everything that they do each day to keep their children living the healthiest life they have the power and ability to do at this time. And ACD could not be more committed to finding a cure and helping all of these pieces come together. So I just want to call out again that this has all happened in 1 year. What can we do in a year? It's amazing. And if we can rally together, what could we do in the next year? What will we be talking about at the 2026 symposium? I'm excited. I hope that it's things we couldn't even expect today. And I just want to remind the families that your story is important. It's valuable, it's impactful, and it's how we connect with the world. It's how we take a little small rare disease of CTD or GAMT or AGAT and we put it on the side of Times Square. We attract people to say, I want to help these families. They're committed. They're great partners. So if we all step up together and we tell our stories and we fundraise and we do everything we can, I just want you to know that we have a strong team here and you're a part of it. And we'll see you all tomorrow for some really amazing talks. Thank you so much for being here today.